Good morning, Mukesh sir. Hello.
Morning.
Uh, uh, yeah. Good morning, sir.
Morning. We are start at your lecture will start at 10.
Oh, yeah, yeah, sir.
Okay.
Yes. Right.
Good morning, Richa ma'am.
Good morning. Good morning, Rakesh. How are you?
फाइन मैम आप बताओ कैसे हो
बस ठीक चल रहा है सब कुछ।
चलो
यहाँ वेदर थोड़ा ऐसे ही है।
चलो अच्छा है ना
हाँ जी। साइक्लोन की वजह से थोड़ा कोल्ड है।
हम्म।
So, starting the technical session 5 of today's program, we'll quickly have a recap of yesterday's lectures. So, meanwhile, other participants will join. And so, yesterday, to begin with, the first session of the technical session 3B, the first lecture was dealt by Dr. Satyendra Singh, Principal Scientist Nematologist. He exclusively flagged upon issues with related to IPM in protected cultivation with special reference to nematodes problem, which is being a major, massive threat in protected cultivation. And also has highlighted upon the impact of IPM on various other pests, including insects, pests, sucking, with the special reference to sucking pests and diseases. So thereafter, Dr. Revana Siddha, scientist who is working on storage insect pest management from IIPR Kanpur, delivered a lecture on good agriculture practices and integrated approaches for monitoring and management of post-harvest storage insect pests. In the afternoon, we had a very good productive panel discussion on role of ICTs in plant protection and constraints faced for implementation of IPM in field level. So renowned entomologists, renowned plant pathologists and plant protectionists in general involved in this panel discussion. Almost everybody has given a way forward for taking up the IPM for effective dissemination in the country. Like to quote with Dr. Dandapani, principal scientist who had tremendously contributed to the development of ICT for NCIPM in last decade. He has discussed regarding the ICTs and its way forward in IPM, and followed by Dr. S. Vanilla, principal scientist who is taking care of ICT in ICT program, is a team leader of ICT program in our institute. Also discussed upon various aspects of ICT's utilization and its future perspectives. Thereafter, Dr. Navindu Nayar, Dr. Dr. Adesh, and likewise many from our institute, Dr. Anup Kumar, have flagged upon various constraints which they are been facing in dissemination of IPM technology. And after that panel discussion, there was an excellent lecture which was delivered by role of pollinators in IPM and the conservation and the importance of pollinators in crop production was dealt by Dr. R.K. Thakur, Head of the Department of Entomology, Dr. Y.S. Parmar, University of Horticulture and Forestry, Solan. So yesterday, that's all for yesterday. And today we have a lecture which will be dealt with the role of predators and parasitoids in IPM, which will be delivered from our own sister institute ICAR-NRIIPM Bengaluru under the leadership of Dr. M. Nagesh, director. And Dr. Mukesh Segal sir will be dealing with IPM in cereals. Thereafter, we have a very important lecture on role of ESAP and ICT application in smart agriculture with the various case studies by Dr. A. Prabhuraj, head of the Department of Entomology, US, Raichur, Karnataka. And last but not the least, role of IPM in vegetable crops by Dr. M.N. Bhatt, who is a team leader for horticulture crops from ICAR-NRIIPM. So without taking much time, I request Dr. Rekha Madam to start today's session. Rekha Madam, please.
Thank you, Dr. Raghavendra. Good morning to all. Today, as Dr. Raghavendra said, we will start with a very important session in which the role of predators and parasitoids in IPM, which will be dealt with actually three very competent scientists, Dr. Richa Vashni Madam from NBAR Bengaluru, Dr. Ankita Gupta and Dr. Gracie. Dr. Richa Vashni will be delivering the lecture, and Dr. Richa Vashni.
Yes, Rekha. Yes, Dr. Rekha. Yeah, I'm here.
Yes.
Morning.
Good morning, ma'am. Dr. Richa Vashne, ma'am, she is a very competent scientist and she has a experience in working in biological control from from 7-8 years. And for her contributions, she has been awarded with many awards like Young Women Scientist Award and Dr. B.S. Bhuvanwar Team Research Award. Along with, she has presented her research in various international conferences, for which she is awarded, of course, for for Best Poster Award and Best Oral Presentation Award in the International Conference on Biological Control. She she has received oral awards in many national conferences and national symposium as well for her contributions towards biological control, and she received many awards from various, and she has published her research in various national and international journals. And without taking much time, I'll request Madam to please deliver her lecture.
Yes. Thank you, Dr. Rekha, for introduction. Now I'm sharing my presentation. Is it visible?
Yes, ma'am. Kindly go to the presentation mode.
Yes, yes, yes. Yeah. Now I hope it is visible.
Yes, ma'am. Yes, ma'am.
It's visible.
Okay. Yeah, so thank you, Dr. Rekha, for introduction. Further, I would like to tell that I'm working as a scientist at ICAR-NRIIPM. My forte is rearing and mass production of the insect pest and natural enemies. Particularly, I'm working on bugs, anthocorids, predatory merits, and geocorids bugs, their identification, and their, if possible, we can multiply them in mass number and their evaluations. So the topic which I will be dealing today is role of predators and parasitoid in IPM. So we know that these two are the major players, that is the predators and parasitoid in biological control, which is further a very important, or I would say the key component in IPM. So in India, many natural enemies, especially trichogrammatids, chrysopids, coccinellids, anthocorids, and many other bugs and different other parasitoids have been identified. Their rearing protocol has been developed. They have been evaluated and recommended for the field release in different crops against different insect pests. So for the convenience, I have divided my presentation in three parts. So first part, I would be talking about the world scenario, like how many species they are using, not only in open condition, in field condition, but in greenhouses also. And what is the market, the global biocontrol market stands. Then I will, in my second part, I would be dealing with the Indian perspective. How many predators and parasitoids do we have, and how we can mass produce them, and what is their demand means farmers, which parasitoids and predators they are in more demand. And third, I would like to give some example of case studies and the success stories through our own trial, which we have conducted at NBAIR, and through AICRP biological control, which is the extended arm of NBAIR, where we are validating several biocontrol agents across India. So if we talk about world scenario, there are approximately 350 species of the biocontrol agents. It is a data which has been collected during 2016, which are being used against 100 pest species in different crop ecosystem. So these biocontrol agents not only includes predators and parasitoids, it includes mites and nematodes as well. So if we talk about the market of biological control agents, so in 2015 and 16, the global pesticide market was about 58.46 billion US dollar. And at that time, the biocontrol market was just 1.7 billion US dollar, which was approximately 2% of pesticide market. But what happened in last decades, there was a rapid increase. There was a rapid growth in the biocontrol market, which was 15% per year. And if we see this newspaper report that they have expected, according to that report, the demand for biocontrol agents market in North America and Europe particularly, it was valued approximately, as I told, 1.7 billion or 2,359 million US dollar in 2018, but it may expected to generate around 7,591 million US dollar by 2026. So it is a huge leap for the biocontrol agents companies which are producing different predators and parasitoid. It is an opportunity, it is a hope in this area. So as I said that there are 350, there is a huge list of that, but I have here shown the first page of the table. So in that they have mentioned that was reported by Lentron in 2012 and later he updated in 2018, he published also. So they have given the natural enemies and the region where they have been used and the target against the target insect pest, year of first use and the market value. It is medium, small and large. So if we talk about large market value, it means they are selling 100,000 to million individuals per week. If we talk about the medium market, it means that they are selling 10,000 to up to 100,000 individuals. I'm talking about individuals per week. And if they say the small market, that means the demand is very less, up to 10,000 per week they are selling. So on the basis of, there are many, most of them are from the ICAR order, that many predatory mites are there, trichogrammatids are there, neuropterans are there, polypterans are there. So this is the n number of list which I have given only the first page here. Then if we see the world scenario, what kind of the augmentative biological control programs are happening, and it happened between 2003 to 2016. The major biocontrol agent which was used was trichogramma species in different countries like the USSR. Approximately 10 million hectare area was covered. In China, approximately 4 million hectare area was covered. I'm just giving, I have highlighted trichogramma species. Besides that, the Crotacea species was also used against sugarcane borers in approximately 3.6 million area in South America and China. So this gives a complete picture where these biocontrol agents are being used in which countries and how much area is being covered by that. So now what this table shows that out of the 350 biocontrol agents, these are the 25 species of the biocontrol agents which are being used in more than 20 countries altogether. So majority of them are being used in greenhouses, especially in European countries. For example, Amblysia swiski, it is being used in more than 20 countries against whitefly, thrips and mites, and it was introduced in 2005. Many predatory bugs are there like macrolophers, aureus, these are the bugs which are being used, and Cryptolemus montrouseri is also being used in more than 15 countries. Similarly, these nematodes are also there, Heterorhabditis, and they are also being used against Polypteran insect pest, and Trichogramma, of course, it is being used in more than 10 countries. So again, NAP 2018, they further extracted some major players. These are the major players which are contributing 90% of the biocontrol market, which I told you about 600 million US dollar. So these are the biocontrol players. Majority of them are being used only in the greenhouses. So for example, many predatory mites, amblyopsis, phytoceos, and oreas, and this is the predatory bugs. So they constitute around 80 to 90% of the world market. Rest 20% biological control market is being used by is being generated by use of Trichogramma cotaceae and other species. So if we see nowadays, earlier there was a trichogramma which was holding, even now also it is holding a major upper hand in biocontrol program. But since the later on the protected cultivation came into existence and many biocontrol agents were explored during that time, so it was amblyopsis whiskey which was introduced in 2005. So before 2006, the major market was covered by Trichogramma and Encarsia formosa. And if we talk about particularly in greenhouses, the Encarsia formosa was the major one because it was being used against the whitefly. Since the introduction of the amblyopsis whiskey, because it is very effective against whitefly, thrips, and mites, it is a general predators, the major market has been captured by this particular predatory mite. So besides that, he has also given a list where the predatory mite are being used against different insect pests, thrips, whitefly, and different species, which I am not going in detail. Just I am giving you a glimpse where which are the major players, predators, and parasitoid in biological control. Now it was the world scenario. Let's come down to Indian scenario. What do we have in India? So at NBIR, since we are bureau, we are supposed to maintain, nucleus culture of different parasitoids and predators. So we are having cryptolemus, Chrysoperlagis strobissalami, and several anthoporids and predatory mirrid bugs, which I will be discussing one by one. These are the parasitoids which we have been rearing at bureau. This is the Trichogramma, Anastatus, Telenomus, Gonoides, many parasitoid, egg larval parasitoid we are having, which I will be dealing one by one. So now coming to the trichogrammatid, which is the most extensively used biocontrol agents, not only in India, but across the globe. Let's talk about this. We have in NBIR approximately 26 species of the trichogramma and the 4 species of the trichogrammatoidea. So out of 26, 12 are indigenous. These are the indigenous trichogramma, which we can get from the field. And out of this, there are, there are 14 exotic species also, which we imported long back. But if we say that out of all the trichogrammatid species, the major demand is for Trichogramma chelonis, Trichogramma japonicum, Trichogramma acci, Trichogramma pretiosum, Trichogrammatoidea bacteri. So these 4, 5, and sometimes Embryophagus and Kaposi also, especially in temperate area. So these are the major Trichogramma species which are in great or huge demand. And besides these Trichogramma species, the NBIR has also developed some strains of Trichogramma chelonis. For example, the high searching ability strain. So it is having the high host searching ability, and it can parasitize up to 95% of Porcella cephalonica eggs even after 30 generations. Then multiple insecticide tolerant strain has also been developed by earlier Endogramma was a major strain which were used when the endosulfan was there, but still now we have multiple insecticide tolerant strain of Trichogramma chelonis. We do have one Trichogramma chelonis and Trichogramma embryophagus strain, which we have, which we are rearing on Iricilpom X, because the size of the Iricilpom X is huge. So whatever the individuals we are getting from the Iricilpom X, they are having the larger size and which better and faster searching ability. Besides that, we have high temperature and low temperature tolerant strain of Trichogramma chelonis, which can sustain up to 40 degrees centigrade at the higher side and 15 degrees centigrade at the lower side, with approximately 90% parasitism. Now, if suppose somebody wants to produce these biocontrol agents, how they go ahead? So first, we do have one, Coursera cephalonica. This is the factitious host which is being used extensively for multiplying different parasitoids, predators. So if we see the Coursera X, it can be used for the multiplication of trichogramma, trichogrammatoidea, chrysophids, anthoporids, geoporids, and predatory merids. While larvae can be used to multiply goniages, which is a larval parasitoid, and Bracon bravicornis, which is also the larval parasitoids. So first, if suppose somebody wants to produce, they have to have the Coursera cultures with them. How to go for that? Is that this is the cages which we are using for multiplication of Coursera cephalonica. It is a wooden cage of size 15 into 30 into 45 centimeter, and it is having mesh. There are the 6 holes are there, and it is covered with the 100 mesh size just for the aerations. So before starting the Coursera culture, it has to be cleaned thoroughly, sun-dried, and if possible, then it has to be sprayed with the formaldehyde and the sulfur dust if you have mite problem. After once your this Coursera cage is ready, then take approximately broken bajra or sorghum, depending upon the availability of the base material, that is 2.5 kg. Then add approximately 75 grams of groundnut seed powder just to enhance fecundity, and then yeast powder, it's streptomycin sulfate, little bit, a pinch, and then add just 0.125 cc of fresh Coursera eggs, or approximately 2000. Because 1 cc is equals to 1 ml if we do the volumetric measurement. So 1 cc is having 16,000. So we are adding just 1/8 of 1 cc, that is the 0.125. Then after just to mention the dates over on the cages and put it in the rack. So after 35 to 40 days, the moths will start emerging, and then it has to be collected through the vacuum cleaner. It is simple devices, semi-automatic devices in the oviposition cages. These are the oviposition cages. And once the moths are there, it has to be fed on honey, that is the 50% honey. Take 50 part of honey, 50 part of water, put some vitamin E capsules just to enhance the fecundity, and hang it through this vent. Here we have cut it and then put the mesh, and through this these are hanged inside that, and they can feed. And because the moths are nocturnal and they lose eggs on the paper, it can be collected like this. These are the Coursera cephalonica eggs. Now, this Coursera cephalonica eggs can be used to replenish, means to sustain your Coursera culture as well as, but if you want to go for the trichogramma, it has to be UV treated. So keep in UV chamber for 30 to 45 minutes. So they are UV treated just to arrest their embryo development, because the trichogramma, it is an egg parasitoid. It parasitize the eggs of Coursera, and then it completes its entire life cycle inside the Coursera eggs. So for better multiplication, the embryo arrestment should be there. That's why we are going for the UV treatment. So once you have the UV treated eggs of Coursera cephalonica, take one postcard size of card with the thick it is the size of 16 into 12 centimeter. Then just sprinkle Coursera eggs over the card, and then expose with the already previous card in 1:6 ratio for 24 hours. After 24 hours, take out the cards which have been exposed with the parasitoids. Say if you want to go for the Trichogramma chelonis, then expose with the Trichogramma chelonis adults. After 24 hours, take out these cards and keep it. Then on 4th day, you will see that they have turned black. That means they are parasitized. And on 8th days, the adults of that particular species of Trichogramma will come out. So recently, NVIR has developed this improved mass rearing things. It is having the 3 tiers. So at a time, we can expose 150 to 200, even sometimes more than 200 if you are using high searching ability strain. Then, but to the safer side, on an average, approximately 200 cards can be exposed at a time to cover huge area. So that was all about the Coursera cephalonica X for trichogramma multiplication. I also told you that on Erysilpom X also, we tried to rear few species, especially Cleonis and Embryofagum. Here we are using Erysilpom X and exposing with these 2 species. So as I told you earlier that the individuals which are coming out from the Erysilpom X, it is having it is sturdy in size, and it is having high searching ability. So this table give you a glimpse about the different trichogramma species against different insect pests in different crops. For example, in coconut, sorry, in paddy, and their Trichogramma chilonis japonicum are being used. Even in maize, Trichogramma proteusums are being used in other countries, but in India now we are using Trichogramma chilonis. Similarly, for sugarcane, also chilonis and japonicums are being used. So once you have your tricho cards ready with you, how to release in the field is that the tricho card is having 16 bits. So it has to be cut. There are bits are there. So all the 16 bits have to be taken out and they can be stapled on the lower surface of the leaves. Okay. And make sure that tricho card release should be done either in the morning hours or in the evening hours, and avoid, avoid pesticidal spray if you're going for the tricho card release for at least 4 to 5 days. And so this is the release methods, and these are the different doses of the different trichogramma species against different target pest. And for example, Trichogramma chilonis against these many pests, 50,000 per hectare can be released. And for the fall armyworm, which is a invasive pest now in maize, which is a major problem nowadays on maize. So there the recommendation is 1 lakh per hectare, and 3 to 4 releases in maize can be made. Similarly, Trichogrammatodia bacteria against the Plutella xylostella can be released at the rate of 1,50,000 per hectare, and against the pink bollworm, 1 lakh per hectare can be released. So here I would like to give you one example at the village level. This is from the Gadchiroli, Maharashtra, and there is a self-help group of 250 farmers. They are particularly growing rice and vegetables. So they have started, Coursera production in not only in wooden cages, but in the plastic cages also. And they have developed their sort of collecting mechanism of moths. And you can see here they are producing the tricho cards. So they could produce total 4,000 tricho cards in both the season Kharif and Rabi, and which fetched them around 4 lakh rupees. Besides that, they have supplied 1,160 cards into the State Department. So I will link later with the commercial insectary. This kind of activities we should start at least to meet the local demands, which I will come later in my end part. So this was all about trichogramma, on how to produce on Coursera, how to produce on Eddie silkworm and others. Now coming to the Anastatus acuronchi and Bangalore, which are the egg parasitoid of lychee sting bug. It can be reared on again Eddie silkworm eggs. So now the Telenomus ramus. Natural parasitization was observed on fall armyworm eggs by this parasitoid. So this is the target pest of this parasitoid, like Exigua, Helicoverpa. These are many. But how we are also producing this both on Spodoptera litura as well as Spodoptera fujiparda. So here what we are doing, we are taking just a plastic container and covering with the rough paper, any rough paper, and then release around 20 pairs of the Spodoptera litura or the fall armyworm moths inside that. Then they will lay eggs. It has to be cut and pasted on the thick card, and it can be exposed to the parasitoid in 1:60 ratio. So 1:60 ratio, and exposure time should be 24 to 48 hours, depending upon, if suppose you have the parasitoid in less number, then expose for longer duration, and then take out this card and put the fresh card. So repeat this procedure for 4 to 5 days. After that, the parent Telenomus ramus fecundity is reduced, so we are avoiding that, just to expose up to 4 to 5 generations. And then on 10th day, the parasitoid adults are emerging out, which can be released into the field at the rate of 20,000 to 25,000 per hectare, and 3 to 4 releases can be made against the fall armyworm. Then coming to next, egg larval parasitoid, which is Chilonus blackburni. It is a parthenogenetic parasitoid, and this we are rearing on again Coursera eggs. Here we are not going for UV treatment because it is an egg larval. The adult, it parasitizes the eggs, and complete its life cycle inside the egg and the larvae of the Coursera. And finally, the adult of Chilonus blackburni will come out from the larvae of the Coursera cephalonica. So here what we are doing, we are making card with the fresh Coursera eggs, and it is expose with the Kilunus black bunny adult for 24 hours. After that, take out the card and keep in Bajra medium. Okay, so the larvae of Coursera cephalonica will hatch out. Inside that, the larvae of Kilunus black bunny is also there. It will also complete their life cycle within the larvae of Coursera cephalonica, and finally the adults will come out, which can be released against potato tuber moth at the rate of 50,000 adults. Two releases should be made in the field, while in go downs, 5 adults per kg potatoes can be released. Besides Kilunus black bunny, recently we got Kilunus formosanus and other 2 species of the Kilunus which are parasitizing fall armyworm in natural conditions. So we collected and we tried to rear in the lab. So we could rear it on both Coursera cephalonica as well as its own natural host, that is the Frugiperda. So here the Frugiperda eggs are pasted on the card, and then they are exposed to Kilunus formosanus adults for 24 hours. Then after that, take out this card and keep in ventilated container till they hatch out. Once they hatch out, rear those fall armyworm larvae on castor leaf for 4 to 5 days, and after that transfer each larvae individually in the semi-synthetic diet. Here you can see the larva of Kilunus formosanus is coming out from the larvae of fall armyworm. So it's the developmental period is about 25 to 27 days. Then cocoon will be formed here that can be collected from the diet and then keep in ventilated container. After 8 to 9 days, adults will be emerged and it can be released into the field. As I told you that besides Kilunus formosanus, we also got 2 Kilunus species which we have been rearing on Coursera cephalonica, just like the Kilunus black bunny we are rearing. Now coming to Gonius nephritides, which is a larval parasitoid. It is a gregarious larval active parasitoid of black-headed caterpillar in coconut. So here we are rearing on the Coursera cephalonica, and both BSC also you can rear. So what it does, it just paralyze the one larvae like this. One larvae in individual vials have to be released, and then one female has to be released inside this vial. So first it will paralyze the larvae, and then it will start laying eggs. So every time they take out the adults from these vials and again expose with the new larvae of Coursera cephalonica. And you can see here, this is the larvae of Gonius, which is being developed on the BSc or the Coursera if we are taking Coursera. And these are the pupae cocoons of Gonius. Collect it and then send it like pack it like this, and you can ship it also. And after some time that adults will come out, which can be released on the trunk at the rate of 10 adults per coconut palm against black-headed caterpillar. Total 4 releases can be made. You need not to release these parasitoids in the crown area. So because they prefer walking, if you release on the palm here itself, it can walk and reach up to the crown area and parasitize the black-headed caterpillar. Then again, the next larval parasitoid is Potatia vestalis or Potatia frutelli. Earlier it was known. It is a solitary larval endoparasitoid of Lutella xylostella. So here, this mustard seedlings is used to rear the Plutella xylostella larvae. So what there we are taking one cage, that is a wooden cage of 1 feet, and then 200 cocoons of Potatia frutelli can be kept. After some time, when they will be emerged, they can be kept in a cage for approximately 24 hours to complete their pre-ovulation period. Then in that cage, provide mustard seedling carrying approximately carrying second instar larvae of Plutella xylostella and expose these larvae for 24 hours. After 24 hours, take out this mustard seedling and replenish with the fresh mustard seedling carrying second instar larvae of Plutella xylostella. Like that, it can be provided fresh batches of second larvae for a week at least. So each female can parasitize approximately 100 and 150 larvae, then collect the cocoon. Once the cocoon, the adults are emerged, it can be released into the field. The next larval parasitoid is Bracon bradycornis. Everybody must be aware about that. It is a gregarious ectoparasitoid again. So it has been evaluated against black-headed caterpillar and fall armyworm. It can be reared very well on Coursera cephalonica larvae. So we have done we have evaluated this Breadcorn brevicornis against the fall armyworm. So we could notice the 54% reduction in infestation by the fall armyworm. And once we have made 3 releases, we could record 10 to 22% of parasitism by this parasitoid against the fall armyworm. Now coming the papaya mealybug parasitoid. So everybody knows that the papaya mealybug problem was there in 2000. It was reported in 2008. Then the efforts were made to import 3 parasitoid, Acidophagous papayi, Pseudoleptis mexicana, and Anagarus loci. So out of these 3, the Acidophagous papayi proved to be very efficient against the papaya mealybug. And this parasitoid can be reared on potato shoot. So what can be done, that infest that once the potato shoot is ready, they infest this potato shoot with papaya mealybug population. So once they will colonize the potato shoot, it will take approximately a week to colonize the potato shoot. That time release approximately 100 parasitoid of Acidophagous, and then allowed them for oviposition and other things. After 12 and 14 days, you will see the mummified mealybugs. That means they are parasitized. And once after 14 days, the adults of Acidophagous papayi will be emerged, which can be released into the field at the rate of 100 to 200 per infested plant or tree. So that was all about parasitoids. Now coming to predators, the most famous predator is Chrysopids. So there are 2 family, Chrysopidae and the Hemerobidae, green lacewing and the brown lacewing. The green lacewing, the majority of them feed on aphid scales. They are the general predators and caterpillar spider mites. And Hemerobidae, they could feed on spider mites, but they are also mainly aphidophagous. So if we see Hemerobidae, the Micromus timitus and the Micromus igorotus, both are very prominent predators. So Micromus igorotus, it is a very good predator of the sugarcane woolly aphid, and it has been evaluated against this particular woolly aphids, which I will be giving in my table. Now, the second, the most exploited one is the Chrysoperla gistrovisalim. It is also a general predator, and it has been evaluated against different insect pests in cotton, green gram, sorghum, sunflower, pigeon pea. And how to mass produce this is that just this is the oviposition container, release some adults of chrysopids there and provide diet, which is having protein X plus fructose plus honey plus powdered yeast in small quantity of water, and mix pollen, castor pollen also, just to increase its fecundity. So it will lay eggs on the paper, this brown, on this black cloth, and then it has to be dislodged because they lay stocked eggs like this. You can see stocked eggs like this. It can be dislodged with the help of the brush, and then collect the eggs. After 3 to 4 days, they will hatch out, and then initially there will be a group rearing of these grubs. Corsera cephalonica UV treated eggs can be provided as a feed. After 3 days, because they are cannibalistic in nature, after 3 days, one grub can be released into this partition. This is a lower which is having 192 partitions. So one grub can be released in one part, and Corsera cephalonica eggs can be sprinkled every alternate day with a shaker. Any salt shaker is there. With the help of that, it can be sprinkled. So there is a 14 to 15 days is the grub duration. After that, they will become pupa like this. And then pupal period is about 7 to 8 days. After that, the adults will come out, and further you can multiply your chrysopids. So, but in this Chrysoperla gistrobi, the release, the predatory stage is grubs only. So either the eggs can be released into the field, it can be mixed with the sawdust and sprinkled, or if possible, to avoid predation, release first install grub against the insect pest on the crops. So here the paper set early 2011, they have given a list in which the Chrysoperla species have been evaluated against different aphids, for example, Apis gossypi, and different other insect pests, Vemisia tabeci, Mysis persici. So many insect species were evaluated for this Chrysoperla species. This is the Indian scenario. This table shows what work has been done on chrysopids in India Malada boniensis or the Malada desters dinsi, it has been evaluated against associated. They were found associated with many insect pests in many in many crops. For example, it was associated with Mysis persica in tobacco, Sporoptolitura in tobacco. For example, Pseudomalada asteris was with the white fly in guava. Similarly, Chrysopella gestrovisilemi, it was found associated with the Lepephis erythema in different crops in mustard, Memisia tevici in cotton. So this list gives a glimpse of different chrysopids associated with different insect pests. Now coming to predatory coccinelids, which is a major, after Hymenoptera and Echeri, the coccinelids is upper hand in terms of biocontrol agents. So they are aphid feeders, for example, Philomenus, Sex maculatus, Coccinella, Harmonia, Coccinella transversalis. They are millibug feeders, Cryptolemus and Ischemnus. Right now at our bureau, we are rearing Cryptolemus montrouseri on pink millibug. There are scale feeders also like Chiloporus and others, silage feeders and general feeders like Brumoids and Ischemnus latimiculatus. So I will be giving, though NBIR has developed rearing protocol of others, but to cut short, what we are rearing now, I will be giving the rearing protocol of Cryptolemus montrouseri. We are getting a huge demand for this predator, especially in mulberry, against the Tupra disease, that is the pink millibug problems. And besides that, in orchards also, we are getting huge demand for these predators. So these predators just procure the medium size of pumpkin, which is having the grooves, and then make it ready. If suppose any crevices is there, cover it with molten wax. Once the pumpkin is ready, it has to be infested. This is a Fericia, but if you have millibug culture, pink millibug maculatus, it can also be released. Approximately 40 to 50 sac can be released on the one pumpkin. After 25 days, it will be fully covered with the millibug. This is the time this predator has to be released with 10 pairs. So it will lay eggs on the sac of ovisac of the millibug, and then finally the grubs will come out. This is the grub of the predator, Cryptolemus. It will feed on the millibug, and finally after 30 days, the adults will come. Adults will be emerged from this one cage, and it can be released into the field. So how to differentiate between male and female cryptolemus? So male, this is one is it is short in size compared to the female, but if you see the forelegs, they are brown in color, they are orange in color, sorry. While in case of female, all the legs are black in color. And if you see the last abdominal segment, it is continuous in case of female, while it is emarginate in case of male. So this is the difference. Now, this list shows the example of successful biocontrol programs of various pests using coccinelles. For example, Cryptolemus montrouseri, the highlighted one belongs to India. It has been evaluated and used against Planococcus in female and grapevine against Meconellicoccus. Though other examples are there which are from the different countries, but from the Indian perspective, the Cryptolemus was found successful against Fericia and tobacco and other crops. Here, similarly, the Coccinella septempunctata against the Lepephis in mustard, and Chiloporus nigitus against the coccids in acid lime and citrus. Now, these are the mode of releases. So different coccinellid species, their doses and how to release. If we see the Cryptolemus in grapes, it can be released at the rate of 1500 to 2000 liters per vineyard. And in case of the citrus, it can be released 20 to 50 adults per tree. Now coming towards bugs. Now a lot of bugs are being used. I have shown you in previous slides also in greenhouses, in protected cultivations, especially anthoporid bugs and predatory millet bugs. So coming to anthoporid bugs, there are the two category of anthoporid bugs. Some lay loose eggs, some lay eggs on the beans. So these are different categories. And if we see the Blactocitus paliscens, it has been evaluated against thrips, Tuta absoluta, and Chylopatylus, Tetranicus artici in different crops, chili, okra, capsicum, rose, brinjal, and mulberry. And the Cardiostethus exiguus, it lays loose eggs. So it was found as a potential predators of eggs and neonates of the BHC, coconut black-headed caterpillar, and it has been also evaluated against the thrips. So these are the Indian amphiporid like Aureus maxidentis tentillus, which were found effective against the insect pest like Helicoverpa officina, Chylopericia, and stored grain pest. This is the list which is showing the highlighted one is the indigenous one. We don't have Macrolophus and others, but we do have, if we see the myriad predators, we do have Nisidocoris and the Daughter's primarius, and they have been found feeding on whiteflies, spider mites, Strepsirrhota absoluta. Similarly, out of the anthocorid bugs, the highlighted one is Bleptoistithus palisson, which is indigenous, and it has been found feeding on thrips, broad mite, and red spider mites, and small caterpillars. Again, this table shows the successful field release of amphiporids across world. So the highlighted one is belongs to India, for example, Blepto, Tetranicus articus, India, and these are the authors who have studied them. Similarly, Cardiacithus exiguus in coconut against the black-headed caterpillar. Then Xylocoris flavips has been evaluated against Corsera cephalonica in the Cytotroga in stored grain trials. Now, coming to the case studies, how these parasitoids and predators were used in managing different insect pests in different region. And this example comes from the greater Mekong sub-region. In that, what happened, the IRI, Philippines, and some biocontrol company in China, CABI in Switzerland, China, and Malaysia, and the Plant Protection Institute in China, they joined hands together, and they have established 11 trichogramma rearing facilities. And because of that, they could tackle the pest problem in paddy. In the similar line, we also have the example in India, particularly in Andhra Pradesh and Kerala. So here, they were the main players. For example, they were the BIPM practices farmers, the farmers who were practicing BIPM strategies. They were supported by NBIR, their state agriculture university in Kerala and Andhra Pradesh respectively, different NGOs, state departments, and they came forward and they have developed some sort of trichogramma facilities and without any special fundings. And now they are practicing full BIPM technologies in rice. For example, here again, the further I am taking example from the Kerala. So large scale validations of VIPM technology, they have been doing in 1500 whole lands hectare area for the last 10 continuous years, in which what they are doing, they are going for the seed treatment with the pseudomonas, they are going for the 5 to 6 releases of the egg parasitoid, especially Trichogramma chilonis and japonicum, and need-based foliar spray of the pseudomonas fluorescence for the foliar diseases and the neem oil formulations. So with that, they are managing borer problems in paddy without any insecticidal spray. And similarly, the Araku Valley also, they have been doing these practices. So by adopting these BIPM practices, the mean grain yield which they have got was 7,595 kg per hectare in BIPM plots, while in insecticide plots, they got 7,430 kg per hectare. So if we computed this, so increased yield which they got was 165 kg per hectare, and the net benefit was approximately rupees 4,584 per hectare from the BIPM practices. Now, the second pest, which is a fall armyworm, once it was introduced in India, it was found in India in 2018, and since then we have been doing several trials on that. So several natural enemies have been recorded on this fall armyworm. This is the list, but the major one is the Telenomus ramus and the Trichogramma chilonis. We got the natural parasitization by these two parasitoids, besides other parasitoids also. So with the help of these parasitoid and other biocontrol compatible strategies, for example, pheromone traps and sprays of BT, metarhizium, and also some trials were conducted in Karnataka as well as Andhra Pradesh. So Karnataka, we got by adopting these strategies like pheromone traps, releasing of egg parasitoids and spray of BT, neem oil, everything. We got 74% reduction in egg mass and 70% here I have mentioned wrongly, 70% egg mass reduction and 74% larval population reduction. Similarly, in Andhra Pradesh, 50 acres area was covered by adopting these strategies. So they found Trichogramma proteusum at the rate of 23 20,000 per acre, plus 2 releases of metarhizium and isoplee. It yielded 39.38 quintal per hectare, which was on par with the insecticidal, but the benefit cost ratio was higher in this BIPM module compared to the insecticidal check. So if we see the area covered by the release of different trichogramma species in different ecosystem, for example, paddy, brinjal, cotton. Cotton trial we have done in collaboration with NCIPM in Jalna, where we have supplied trichogrammadoidea bacteria and they have their own IPM module. So we intervened in the form of the biological control and we have got very good results in that places. So this is the like area, how much area we could cover. So approximately 32,743 hectare area we could cover during these 2 years. So here, this is further the glimpses like trichogramma release in different cropping ecosystem like cabbage, maize, and cotton ecosystem. These examples we are giving that my colleague Dr. Om Prakash and I done, we did one experiment in cabbage. We used multiple insecticide tolerant strain of Trichogramma chelonis, where we made 6 releases of this particular strain of Trichogramma with 2 sprays of the BT and the 4 to 5 sprays of the insecticides. Then we compared with the farmer's insecticidal spray and we have got net profit more than the farmer insecticidal spray. And moreover, in the IPM module, there was only 7% damage in the head. While in case of the farmer's insecticidal spray, after spraying 9 times, there was a 32% head damage at the end of the crop. So that was about the trichogramma. Coming to predatory mites and bugs, as I told that Bleptostithys paliscens and Neosulus indicus, they were used against spider mites in roses. And what we have found is that Neosulus indicus, which is a predatory mite, it provided approximately 82% reduction in mite population, which was on par with the fungus, that is the Hirsutella thompsoni and the miticides. Similarly, the mulberry, it was facing a huge problem of broad mite, especially in Ramnagar district, Mandya district, Chamarajnagar district, and there was a loss of 70% of mulberry production. So they approached NBIR and we did one small trial in which we have evaluated again predatory mite and the Hirsutella thompsoni along with Bleptostithys paliscens, and we got very good results controlling in broad mite in mulberry. Because mulberry, we can't use many pesticides, so we have to depend on the biocontrol agents. Now coming to AICRP biocontrol, it is as I told you, it is an extended arm of the NBAIR. What we are doing in that, whatever the biocontrol agents we have been rearing at our bureau, they are validated. They have been evaluated and validated through multi-location trials under AICRP-BC. Recently, they have also produced some units of these centers. They have started producing Trichogramma, goniges, and other predators. For example, Neosulus longispinosus, Solon Center, that IS Parmar University, they are producing this predatory mite, and the revenue generation was 30 lakhs by producing these biocontrol agents. If we see the AICRP coverage area, so in the last 16 to 20, it was like approximately 32,000 hectare area was covered, while from 90 to 2060, approximately 5,94,060 hectare area was covered, covering 14 crops and weeds. That time weed was a part of the AICRP. So now again, there is a Hugo's spiraling whitefly. It invaded in 2016. Then ICAR-NRIIPM and Dispersa, they were found promising against this particular pest. So initially we recommended the conservation of that, but recently one of our colleague, Dr. Selvaraj, they have developed one very good technology on rearing of ICAR-NRIIPM, which he has been rearing and supplying to the farmers. So, sorry. So what is happening? I am giving you here some case studies where we have used either predators and parasitoids along with some compatible biopesticides and what results we got. So Kerala, Tamil Nadu, Andhra Pradesh and Karnataka state, that trial was done. where the ICAR-NRIIPM here, which is an endopathogenic fungus, Pseudomonada astro, which is a predators, and Encase they were used, and they could observe 80% pest reduction, and total saving was 9,500 per hectare by utilizing this strategy. Now coming towards rice, the rice, sugarcane, maize, we have got tremendous success by deploying this trichogrammatids. So as in Kerala, Odisha, Assam, and Punjab, the trichogramma japonicum plus Pseudomonas and Beauveria as a directing, they could observe 56% pest reduction with 4,000 per hectare saving. In Punjab, particularly in basmati rice, they have made 6-7. This is a success story which they have published also, which I will be showing in my coming slides. 6-7 releases of egg parasitoid, particularly Trichogramma chelonis and Trichogramma japonicum. They, by using these parasitoid, the Cartip hydrochloride application was reduced from 3 to 1. So it could lead to the saving to the farmers for approximately 9,100 per hectare. Similarly, in maize, by intervening with Trichogramma chelonis, because before fall armyworm, the stem borer Chylopatulus was a major problem, particularly in Punjab region. So by deploying this parasitoid Trichogramma chelonis, they have made 4 to 5 releases. So increase in yield was reported 165 kg per hectare, and there was 10,360 per hectare saving was recorded. Now coming to the sugar cane, which is a huge success again by using these biocontrol techniques. So trichogramma technology means the Trichogramma chelonis and japonicum. These two parasitoids were used in approximately 11,730 hectare area in different states, for example, Punjab, Assam, Odisha, and Andhra Pradesh. So they could notice 62% reduction of pest incidence, and there was a net return of 1,72,987 per hectare. Besides this, the high temperature tolerant strain of Trichogramma chelonis were also evaluated and validated, where the temperature is very high during peak summer months, and it was at par. The release of this particular strain was at par with the chemical control in Punjab, Tamil Nadu, Andhra Pradesh, and Telangana area. Now, again, success comes from the Punjab. Here, I would like to mention that the Punjab Agriculture University, they joined hands with the sugar mills, and they developed the trichogramma unit. You know, they are multiplying trichogramma their own, and because of that, they could cover huge area, which is 12,969 hectare area, and regular release of these two species reduce the incidence of sugarcane borers to the tune of 58%, and which could also resulted in reduction of pesticide uses. And it is worth mentioning that it has been included in their package of practices, and there is no chemical recommendation for the sugarcane stalk borer in their packages of practices. So they could ably manage this stalk borer problem in sugarcane by employing this Trichogramma parasitoids. Now the papaya mealybug, as I already told, so due to the release of this Acidophagous papayi, and approximately 2,044.5 crores in Tamil Nadu and 199 crores in Maharashtra could be saved by not using any chemicals against this particular pest. Now, this is the example of BIPM module in apple, particularly against the codling moths. So Trichogramma cacossi at the rate of 2.5 lakh per hectare. So total 4 releases have been recommended, plus trunk binding, plus disposal of the infested fruit, pheromone traps, and the spray of Heterorhabditis pakistanensis, which is an NBI isolate. It was found effective in managing this codling moth in Jammu and Kashmir, and it could lead 52.31% pest reduction. Now coming to the protected cultivations, this Blaptocetus, Nuusulus, Longispinosus, and other predators, and chrysophids also, they have been evaluated against different insect pests in different crops. For example, in Kerala, this Blaptocetus paliscens in cucumber against the spider mite, that is a Tetranicus truncatus, not urticae. It was found effective and it was almost on par with the equicide treated plots. Similarly, it could reduce the Gerbera thrips population and it was also at par with the imidacloprid in Gerbera against the Frankinella species. That study was also done in Kerala. In Maharashtra, the 4 releases of this predator at the rate of 20 nymphs per rose plant was effective in reducing rose mite population. In Solan, they evaluated this Chrysoperla gestrovisalimi at the rate of 4 larvae per plant, and it could reduce the aphid population to the tune of 55.8% in capsicum. And it was on par with the azadirachtin and mecanicillin, but definitely the percent reduction was more in insecticide. But if we see the biocontrol things, that it was found very effective. In Punjab conditions also, the same predator at the same rate, it resulted in 88% reduction in aphid population in capsicum. And this Neosulus longispinosus, which is a very specific obligate predator for the Tetranicus articae, it could reduce 71% population of the spider mite on carnation. So that was some case studies or some experiment which we have done in AICRP and which got some success. Now coming to the commercial production, because if we want to deploy these biocontrol agents, we have to produce them in large number. So how we can go ahead is that commercial production, if we see the worldwide, there are 530 commercial insect trees, particularly European countries and America. And out of 530, the 30 are the large companies, which they are deploying more than 10 people in their companies. And out of 30, 20 are in Europe. Because as I told you earlier, the major biocontrol market is ruled by the greenhouse biocontrol agents. So in European countries, a lot of greenhouses are there and they are using mostly biocontrol agents in the greenhouses. So most of the companies are in Europe. And the 500 are small commercial producers, which are in different countries. For example, in Colombia, especially Latin America, I am giving you the example. In Colombia, they are having 30 commercial insectaries. Mexico, 30 small commercial insectaries. The Peru, they have 82 insectaries for the microbials and 27 for the entomopathogens. And Cuba, they have 280 for microbials and microbials altogether. So if we see in India, the first commercial insectary was developed during 80s, that is the BCRL. So that time they were producing trichogrammatids and other like pheromone things and other things, but I don't know whether it is functional now because I could not hear from them recently. So this I have given little bit NBIR because we are bureau and we are supposed to supply nucleus culture. So we are having huge pressure of giving, biocontrol agents. So I have given just a line that every year approximately 1500 shipment we are sending, which is containing 43 million live insects to various research organizations, students, commercial units and farmers. If we see the biocontrol market, they are the major players in terms of parasitoid and predators. BioV, everybody knows, Copart, Biological System, Bayer, and Syngenta. They are not only producing, I don't know about some predators and parasitoid they are producing. Besides that, they are producing these last four, they are producing microbials and pheromones things also. So they are the major players. But where do we stand? In India, we could find these are the small farms. If we see, they are, because they have been taking some nucleus culture from us and they got training under us. So we are little bit they are producing to sustain, to cater their local need. So in India, the situation is that we are not even in cottage industry. We are just a government bodies. Some government bodies, they are producing natural enemies, and nowadays some progressive farmers, they have started producing natural enemies as per their need. So these are few players in India which are producing natural enemies small scales. Recently, that Cryptox and the Kisan Sahayakta, they have come forward and they are producing some natural enemies like chrysopids, trichogrammatids, cryptolemurs, some reduvids also they are producing. So I would like to give you example of some local setting up of the biocontrol unit. I would not say biocontrol, a small unit like just to produce trichogramma or chrysoperla. For example, in Angru Anakapalli, they have developed they have established one trichogramma production unit in Aruku Valley. Similarly, Raichur, they have established 2 units in Gangavati and Kalburgi district to produce trichocards to meet the local requirement. The third example, again, it comes like the Trichocard Raitu Mitra group was developed with 15 tribal farmers at Chintapalli division in Visakhapatnam district. So they are producing trichocards, and approximately during these 2 years, 2018 to 20, they could produce 520 trichocards and covering 65 acres of paddy crop. Similarly, in Chhattisgarh, there is a KBC, Krashak Biocontrol Lab. They have also started producing trichocards just to meet their local needs, covering paddy and maize crop. So they are also producing trichocards. Another example comes from Kerala. That is the Porathuseri Krishi Bhavan. So this is a group of few farmers. They got training from the KU, Kerala Agriculture University, and they got little fund from the Department of Agriculture just to buy some buy some basic material. And then with the help of that, they are producing approximately 200 cc in the first season. They produced 2017-18 and another 400 cc in the second year, and still they are producing. So what I mean to say is that though in India we don't have any commercial unit, or in India we don't have any cottage industry, but if we can sensitize the farmers about the biocontrol agents, especially the most prominent one like trichogrammatates, chrysopids and others, and depending upon their local need, they can produce their own. We can make some group, we can give training, and at least we can ask them to produce to a little extent to meet their local demands. So these are the few examples where they have come forwarded and catering their needs. Now NBIR, I just want to give here again the picture of the NBIR. We have been supplying nucleus culture across India, and this is during last 5 years we could supply approximately 487 million insects, out of which 243 million natural enemies we have supplied across India. And recently, in the last 5 years, we have these majority students, and then followed by farmers and the government officials. They were exposed to production of the trichogrammatids. We sensitized them. And in the last 5 years, we have supplied these many trichocards into the different stakeholders. And I have shown this earlier also, that the trichocards production, if we can go for that, we can still produce at a time at least 200 trichocards. So now the challenges, we know that the farmers, they know that the biocontrol works and they want to procure these biocontrol agents, but availability is a big question, where they will get it from. So this is a big challenge to meet the demand. And to some extent, I have mentioned earlier also, if we go by local units, local setup, we can definitely meet their demand, like setting up units, and definitely the research on shelf life, because biocontrol agents, the one drawback is their shelf life. So few research has been done, for example, the long-term storage of the trichogramma pilonis. So by inducing diapause, the trichocards can be kept for 3 months with 70% emergence. So this type of technology we should develop more often, not only in trichogramma but other predators and parasitoid as well, so that it will give a boost to industry, because majority industry, the big companies, they do not come forward for the macrobears. They want to produce only microbears. So this is one of the lacuna, the shelf life study, how we can store them. Suppose the season is not there, but we have continuous production, how we can store them for the long term. So this type of studies should be conducted more often. And definitely the attitude of pesticide industry, farmers and policies, there are several things. For example, pesticide industry, they don't come forward because that biocontrol agents are not patentable issue, and their pesticide market is huge, so they don't go by sustainable use. They have captured enough market. So their attitude matters. And farmers, of course, the moment they see one pest or one insect, they will spray the things without giving a second thought what it will harm to the non-target organism, particularly predators and parasitoid, and policies also. So these are the few challenges which should be looked into, and definitely with that, I would thank NCIPM and Manage for organizing this training program. Dr. Mukesh Sehgal sir, who is the course director, Dr. Mahante Shiru, Dr. Rekha Valodi, and Raghavendra for conducting this training very nicely, and Director NBIR for deputing me for this talk, and my colleague Dr. Ankita Gupta and Dr. Om Prakash Nabik for making the lecture notes and helping me in this presentation. And with that, I thank you all. Thank you very much.
Thank you very much, madam, for your wonderful presentation on biological control. Almost you have covered everything A to Z of biological control. And now, dear participants, it's open for discussion. And I think, Madam Richa, you can even interact in Hindi, right?
Yes, very much. I'm very much comfortable in Hindi.
पार्टिसिपेंट्स अगर आपको जो जिसको भी अगर कुछ डाउट्स है तो आप हिंदी में भी पूछ सकते हैं क्योंकि रिचा मैडम हिंदी जानते हैं. ओके, नहीं अनम्यूट कीजिए, नीरज जी अनम्यूट कीजिए.
वेरी गुड आफ्टरनून मैम.
हाँ नीरज जी, वेरी गुड
मैम आपने
आफ्टरनून.
जो इन्फॉर्मेशन दिया बहुत बढ़िया था और पूरा फुल्ली इन्फॉर्मेशन है, लेकिन दिक्कत एक है कि नॉलेज तो पूरी है लेकिन अवेलेबिलिटी नहीं है फार्मर के यहाँ पे जो आपके प्रोडक्ट की है. मैम नॉलेज में नो डाउट सारी चीज बहुत यूजफुल है. मैंने भी अभी तक जितनी नॉलेज जितनी यूनिवर्सिटी विजिट की बहुत बढ़िया आपका लगा लेक्चर, और जो भी आपने पूरा एकदम डीपली बताया. और मुझे भी लगा कि जो आपने बताया इसमें एक एग्रीकल्चर स्टूडेंट होने की जरूरत नहीं है, एक अननोन पर्सन एक फार्मर भी इसको स्टेब्लिश कर सकता है.
जी
क्योंकि
जी
मैं इतनी यूनिवर्सिटी घुमा हूँ तो मेरे अभी तक 42,000 किलोमीटर मैं साइकिल से घूम चुका हूँ यूनिवर्सिटी ICR इंस्टीट्यूट. तो मेरे को एक अभी फील हुआ, जो एक मैंने अपना फ्यूचर देखा है कि मैं बायो कंट्रोल को लेके और खासकर इंसेक्ट को लेके एक आने वाले टाइम पे एक स्टार्टअप की तरफ जाऊंगा.
बिल्कुल बिल्कुल, मैं बिल्कुल एग्री करती हूँ. हाँ, आप पहले अपना क्वेश्चन पूरा कीजिए.
हाँ,
जी.
तो मेरा क्वेश्चन ये था मैम कि अगर मैं इसको स्टार्ट करना चाहूँ तो कितना इन्वेस्टमेंट की जरूरत पड़ेगी, थोड़ा सा उसको बताएं क्योंकि आप एक प्रैक्टिकली बताएं स्टार्टिंग
देखिए
स्टेज में.
देखिए नीरज जी, पहले मैं आपको बताना चाहती हूँ, आपको पहले देखना पड़ेगा कि आप कौन से नेचुरल एनिमल्स के लिए जाना चाहते हैं, अपनी लोकल नीड्स को देखते हुए. जैसे
मैम मेरे को शुगर केन पे जाना है और राइस पे जाना है और ट्राइकोगामा कार्ड्स को लेके जाना है.
ट्राइकोग्रामा, तो मेजर आपको कॉर्सेरा एंड ट्राइकोग्रामा प्रोड्यूस करना है. तो हमने उसका एक कॉस्ट निकाला हुआ है कि डिपेंड करता है आप कितने हेक्टेयर एरिया कवर करना चाहते हैं. कॉस्ट इसमें ज्यादा नहीं आनी है. आपके पास एक इंफ्रास्ट्रक्चर होना चाहिए, एटलीस्ट 2 रूम होना चाहिए. एक आप कॉर्सेरा के लिए रखिए, एक आप ट्राइकोग्रामा के लिए रखिए. ठीक है, 2 रूम इनफ है. और आपको एटलीस्ट 3 मैन पावर चाहिएगा, 2 मैन पावर चाहिएगा आपको कॉर्सेरा के लिए, क्योंकि कॉर्सेरा थोड़ा लेबोरियस वर्क है. ठीक है, तो उसके लिए मेरी एक डिफरेंट प्रेजेंटेशन है जहाँ पे मैं कॉस्ट आपको बताऊंगी कि आपको एक कॉस्ट इवेलुएट, अगर आपको कॉस्ट लगानी है तो कितना खर्चा आएगा, कितने एरिया हेक्टेयर कवर करने के लिए. तो उसके लिए आप मुझे प्लीज कांटेक्ट कीजिएगा.
मैंने आपकी मेल आईडी नोट कर
हाँ
लिया, मैं
जी,
आपको मेल कर
हाँ
दूंगा.
जी, अगर आपको एस्टेब्लिश करनी है तो आप ट्रेनिंग ले सकते हैं. आप हमसे अगर आप चाहते हैं कि हम आपकी हेल्प करें वो भी हम कर सकते हैं. कॉस्ट इतना नहीं आएगा, जितना माइक्रोबियल्स में आता है उतना नहीं आता है. विद इन 1 लाख ऑल्सो, अगर आपके पास इंफ्रास्ट्रक्चर है, 1 लाख विदाउट मैन पावर आप स्टार्ट कर सकते हो.
मैम एक्चुअली क्या है, मैंने SBUT मेरठ यूनिवर्सिटी विजिट किया था. वहाँ पे ट्राइकोगार्मा का प्रोडक्शन था और वहाँ पे बासमती एक्सपोर्ट डेवलपमेंट अथॉरिटी जो उनकी ऑफिस भी है, मैंने उनकी 2-3 दिन रहा तो मैंने वहाँ पे सेल देखा और फार्मर्स को देखा. लेकिन मुद्दा यही आ रहा है कि जैसे अभी आपने प्रेजेंटेशन में प्रेजेंट किया कि जितनी लेबोरेटरीज मैन्युफैक्चरिंग है इंडिया में सारी साउथर्न पार्ट में है.
जी जी
इश्यू
जी
यही आ रहा है कि मैं इंडिया को नॉर्थ इंडिया को अवेयर भी कर दूं तो अवेलेबिलिटी नहीं है. और जहां यूनिवर्सिटी में हम यूनिवर्सिटी को लेके ये नहीं कह सकते कि यूनिवर्सिटी में अगर मैन्युफैक्चरिंग है, क्योंकि सरकारी तंत्र का पता नहीं आज के कि उसका कोई भरोसा नहीं है.
जी
अभी
जी
लैब चल रही है, शाम को पता चला साइंटिस्ट की पोस्टिंग कहीं और चली गई या लॉक लग गया. तो
नहीं नहीं
मेरे को नॉर्थ इंडिया में ये देखने को नहीं मिला अभी तक लेबोरेटरी जो कमर्शियल लेवल पे. तो मेरा भी एक इस चीज को देख के मैं सोच रहा हूँ मैम.
बिल्कुल, मैं आपसे रिक्वेस्ट करूंगी आप बिल्कुल करिए, क्योंकि आई ऑल्सो कम फ्रॉम नॉर्थ इंडिया, मैं यूपी से हूँ. मैं भी चाहती हूँ कि नॉर्थ, एक्चुअली साउथ इंडिया में फार्मर्स अवेयर हैं कि ये बायो कंट्रोल है, काम करता है, प्रोड्यूस करना है. लेकिन ऐसा नॉर्थ इंडिया में मैंने नहीं देखा, क्योंकि कुछ काम मैंने करनाल में किया था ड्यूरिंग मेरी FET के टाइम में. तो वहाँ पे द मोमेंट वो देखते हैं कि इंसेक्ट आ गया, वो इंसेक्टिसाइड डाल देते हैं. तो मैं भी आपसे रिक्वेस्ट करूंगी और आपको देखूंगी कि आप एक डेवलप करें, एटलीस्ट स्मॉल यूनिट स्टार्ट करें, स्लोली स्लोली आप उसको बड़ा करते जाएं, और उसके लिए जो भी आपको हेल्प चाहिए NBIR इज ऑलवेज रेडी टू हेल्प यू.
ओके, थैंक यू.
Yes, yes. Sorry, sir.
Any other parts?
Ma'am, I just have a question regarding glucose spiraling white cell. So, what is the frequency of release? Both, if you want to spray Iceria and also if you want to release Encarsia, what is the frequency? And can it be done together?
No, no, it cannot be done together. There should be always gap. First, if you are spraying ice area, after some time, it can be released. Though in one study, Dr. Selvaraj, he's done, there is no effect on the biocontrol agent, but for the safer side, first, if you are spraying ice area, maintain the gap for 2-3 days, and then you can go for release of Encarsia goodlopi. So, and depending upon the pest incidence, if you are observing that it is reducing, that you need to stop the further application of the Icelia.
How many number of parasitoids has to be released? What is
The number, at least they are recommending around 50 number. I don't remember exactly, but in one presentation I have seen approximately 50 to 100 they are releasing per tree.
per tree? Okay.
Yes.
Okay. If it is Iceria, what is it?
Sorry?
What is the dose for Iceria?
Iceria fumicorosia.
Yes.
I think I am not sure about that. What is the dose? But I will get back to you. I don't know
Thank
about
you.
that.
Thank you.
Thank you so much.
Anybody else? Okay. So if there are no more queries, Shri Krishna, please. पूछिए श्री कृष्णा जी. Shri Krishna ji, you want to ask something? You have raised your hand.
डॉक्टर रिचा,
Yes.
गुड मॉर्निंग.
गुड मॉर्निंग अनूप जी. जी जी
कैसी हैं?
डॉक्टर अनूप बताइए. जी
अच्छा,
ऐसे है
अभी
सब कुछ.
एक क्वेश्चन माइंड में आया. अच्छा,
हाँ.
आपने कभी जैसे ट्राइकोग्रामा रेयरिंग के लिए कोर्सेरा एग स्टरलाइजेशन में कभी ऐसा हुआ कि विदाउट यूवी स्टरलाइजेशन किया हो रेफ्रिजरेटर में? कोई एक्सपीरियंस?
विदाउट यूवी स्टेरिलाइजेशन? नो.
यस. कभी आपने एक्सपीरियंस किया, कभी नहीं किया?
नहीं नहीं नहीं. मीन्स यू आर सेइंग कि हमने कॉर्सेरा लिया है, वी हैव टेकन कॉर्सेरा एंड विदाउट. हाँ.
हाँ हाँ.
Without UV sterilization, if we are exposing to the trichogramma,
नो नो नो.
then
Suppose we want to sterilize the Coursera eggs. We
हाँ.
don't have UV chamber.
Okay.
How do we proceed?
See, it is simply because when and like on-farm production things, we conducted one training when I was in NIPHM. So simply you have to have UV light. UV light you can take. You need not to go for the UV chamber. Just take
No,
the
no,
UV
no,
light.
no, we don't have UV light.
No.
Any other alternative you have tried?
No, no, no. I have not tried that. Because
I
UV
tried.
light usually it is available. So even for on-farm production things, we used to tell you need not to have. You just take one chamber kind of things, sort of things, and just put UV light and then keep you Coursera eggs.
This is the standard protocol.
Mm-hmm.
But मेरा एक्सपीरियंस है, मैंने ट्राई किया.
Okay. Mm-hmm.
मैंने रेफ्रिजरेटर में डीप फ्रीज ऊपर होता है -22 डिग्री सेंटीग्रेड.
Okay.
वहाँ पे अगर 3-4 घंटे अगर रखते हैं eggs को पेटी प्लेट में रख के पहले से, तो उसके बाद वो स्टरलाइज हो जाते हैं.
And what is the percent emergence and percent
इमरजेंसी पे
parasiticity?
कोई इशू नहीं है. हमने चेक किया, उसको देखा था. मैंने
And
ये
percent-
पीएचडी के दौरान भी काम किया था. मैंने कई
Okay.
पब्लिकेशन भी देखे इस पे पहले.
Okay.
क्योंकि मेरे प्रॉब्लम आ रही थी यूवी स्टरलाइजेशन के बाद भी इमरजेंसी हो रहा था.
Okay. That means you are not keeping for the longer time. It
हाँ.
depends like your
हाँ.
height, how much is the height
हाँ,
of the UV.
कहीं ना कहीं वो
and
इशू
what.
आ रहा था. तो फिर जब उसके साथ किया, फिर वो इशू नहीं आया.
Okay. Okay.
तो कुछ फार्मर्स के लिए ये एक ऑप्शन हो सकता है. अगर नहीं है, आप भी एक बार ट्राई करिए अपने यहाँ.
Yeah, but
देखिए,
farmers,
हाँ.
where do we get? They get the -20 or something.
मतलब अगर रेफ्रिजरेटर है उनके पास, अगर
But
घर में फ्रिज
refrigerator
है?
usually that 5 degree or 4 degree
नो नो नो,
something. In deep
ऊपर का जहाँ पे
ऊपर
डीप
का जो
फ्रीजर होता है ना, ऊपर का होता है ना
हम्म,
चेम्बर, जहाँ
हम्म,
पे आइस जमाते हैं,
हाँ.
आइसक्रीम।
Did you do the vacuum packing then? Something?
नो 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 नो,
Okay.
सिंपली,
Simply
हाँ सिंपली उसमें किया था हमने, तो आराम से काम कर रहा है। एक बार ट्राई करिए आप भी, देखिए कुछ रिजल्ट
so
उसमें डिफरेंस
just
क्या है।
one thing. Did you keep those Coursera X in any polythene bag or something, or simply you because
आई
there is
kept
a
in petri plate.
petri plate? Okay.
After
Okay.
sterilization, then I used to make trico card.
Okay. 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 Okay.
Um, Dr. Anup, can I intervene?
Yes, ma'am.
Yeah, Dr. Anup, like we have been doing this not for Coursera per se, but for just taking imaging of the photographs. And it is what is my experience of last 10 years. In many of the cases, the revival happens. So it's not a foolproof way methodology. Otherwise, it would have been in use in commercial insectaries long back. So if you, it can be a chance that you will have some kind of percent sterilization, but then revival will happen because it happens even after 4, 4, 5 hours for us. Because usually I used to do this for various number of species of insects so that they'll become partially immortal so that I
Mobilized.
can take the images. But then revival happens.
Okay.
So like you have to
Then,
be a little cautious
then
if you are recommending this.
ओके ओके, बहुत अच्छा लगा मैडम, पहले आपको देख के, बहुत दिन बाद देखा है आपको। बहुत दिन बाद आए, एनबीआर आए थे।
Okay, okay.
थैंक यू मैडम।
Okay.
क्योंकि मैंने इसमें 4 घंटे के अलावा मैंने 6, 7 घंटे, 8 घंटे भी रखे, तो प्रॉब्लम यही आ रही थी इमरजेंसी की, क्योंकि मैं
Okay.
अपने ट्रायल कर रहा था
Okay,
पीएचडी के
okay.
दौरान।
नहीं हो सकता है कि आप चांस फैक्टर में आपको
नहीं, चांस नहीं, ये मैं चांस नहीं, मैंने कई बार किया।
नहीं नहीं आपने कई बार किया होगा,
हाँ।
लेकिन 100% आपको स्टेलाइजेशन नहीं
हाँ
मिलेगा
हाँ, हो सकता
यूनिफॉर्मली।
है कहीं कहीं, लेकिन
यस, तो
मेरे को
जो
प्रॉब्लम
आप
आई।
पर जो आपके, तो आप जब सपोज यूनिफॉर्मली कार्ड्स बना रहे हैं और फार्मर्स को डिस्ट्रीब्यूट कर रहे हैं और उसमें कुछ एग्स कॉर्सायरा दे दें, तो लाइक यू विल नॉट बी एबल टू कोरिलेट टोटल परसेंट पैरासिटिज्म और रिलीज व्हाटसोएवर। सो फुल प्रूफ हम इसको नहीं बोल सकते हैं। हाँ, छोटे स्केल पे हम इसे कर सकते हैं।
हाँ, वो छोटे स्केल पे, लेकिन प्रॉब्लम
Yes,
ये है
yes.
कि इवन हम यूवी स्टरलाइज करते हैं ना,
हाँ हाँ,
उसके बाद भी इमरजेंस होता है
वो
प्रॉपर।
इसलिए
वो आपने भी एक्सपीरियंस किया वो।
या
वो इसलिए होता है अगर आप हाइट, देयर इज अ
Yes.
स्टैंडर्ड हाइट कि आपको उतनी हाइट पे यूवी लाइट रखना है टू मीट समथिंग।
करेक्ट।
अगर आपको हाइट कम है और आपका एक्सपोजर टाइम भी कम है तो डेफिनेटली इमरजेंस होगा। दैट्स व्हाई आई हैव मेंशन 35 to 40 मिनट्स। अगर आप उससे कम टाइम पे करेंगे तो इमरजेंस होएगा।
मतलब
तो
40
आपको
मिनट्स के बाद चांसेस बिल्कुल कम है,
बिल्कुल कम है
ये
इमरजेंस को।
जीरो।
यस यस यस, ऑलमोस्ट वी आर नॉट एक्सपीरियंसिंग
कुछ पेपर में कंबाइन लिखा था,
Statistically negligible. Statistically negligible.
क्योंकि कुछ फार्मर्स थे उनके पास थी नहीं ये फैसिलिटी, अगर
नहीं,
अपने लिए करना चाहे तो
आजकल
एक
यूवी
ऑप्शन
लाइट
है।
मिल जाता है मार्केट में अब, क्योंकि ये जब मैं व्हेन आई वाज इन NIPHM वी यूज्ड टू गिव ऑन फार्म ट्रेनिंग टू द फार्मर्स फॉर द ट्राइकोग्रामा प्रोडक्शंस, सो दे कैन नॉट हैव लाइक सोफिस्टिकेटेड यूवी चैम्बर काइंड ऑफ थिंग। तो वी जुगाड़ का काम किया था हमने यूवी लाइट देके और एक छोटा सा कैबिनेट बना के, और दे वर डूइंग, though it was also not 100% proof like the commercial insectary they are doing, but to some extent.
वो एक दो
और
बार
हाँ,
जाता है।
और एक तरीका है लाइक व्हेन वी यूज यूवी लाइट फॉर अट्रैक्टिंग इंसेक्ट्स इन द फील्ड्स, हम ब्लैक क्लॉथ लेके जाते हैं और हम यूवी लाइट और उसका होल्डर लेके जाते हैं, हम उसको लगा देते हैं, साउंड हम साइड से ब्लैक क्लॉथ कवर कर देते हैं। यू कैन यूज दैट आल्सो बिकॉज वी यूज अलोंग विद द बैटरी यूवी लाइट इनसाइड द फील्ड, तभी भी काम करता है वो।
अच्छा मैम, ये यूवी लाइट जो है 30 वाट्स का यूज करते हैं उसको?
Yes.
30 वाट का। अच्छा,
Yes.
ये मार्केट में क्या रेट का मिलता है कुछ किसानों को, अगर बताना हो मुझे से
या
कितने का
मार्केट
पड़ेगा?
मार्केट सर्वे करना पड़ेगा, मुझे आइडिया नहीं है तो।
मार्केट का तो पता नहीं, हम तो ऑफिस से परचेज करते हैं।
हाँ, ऑफिस का रेट जो एक बार बता दीजिएगा मैडम।
नहीं, हम तो जेम
बिल्कुल
वाले हैं
बिल्कुल
ना,
बताएंगे।
जेम वाले।
So from the discussion
Thank you, ma'am.
outcome is that to recommend for commercial production of Coursera, so UV chamber is must essential.
Yes, yes, yes.
Just for experimental purpose, we can try with some other means, but for commercial, for recommendation for a commercial trichogramma production, UV is must.
Yes, yes.
Okay, thank you so much. Thank you, Ankita madam.
Thank you, ma'am.
Thank you, Richa madam. And
Thank you
also,
so
I
much.
think Gandhi Grace Madam is also there. Madam, you would like to add something with Madam? Would you like to add something, Madam, on biological control?
Yeah, I think so. Now Dr. Gracie's talk will proceed. After that, I am going to give the talk.
Yes, ma'am. Yes, ma'am.
Okay.
Okay. Okay. Now, thank you, Richa, Madam. Now,
Thank you so
we,
much.
thank you, Madam. Now, we would like to request Gracie, Madam, to deliver her lecture, Madam. Dr. Gracie, please. Madam, your voice is not visible, audible, please. Please unmute yourself. Madam, you are not audible.
She's trying, actually. She's trying to.
Right, right, right.
Can I intervene? Somebody was asking about Iceria fumosoracea. So I have asked her, can I respond to that?
Yes, yes, ma'am. Yes, ma'am.
Yeah, it was 5 gram per liter and 2 sprays they are recommending. We are recommending 2 sprays and 5 gram per liter.
Ma'am, is it available then? What is the formality to get it
Yeah,
from
yeah.
NBIM?
Actually, it is in the form of the technology. Please contact Dr. Selvaraj.
Oh,
Shall I write his email ID in the chat box?
please, ma'am.
Yeah.
Please, ma'am.
Okay, sure.
Thank you so much, ma'am. Thank you so much.
Sure, thank you.
Dr. Gracie, she's changing her microphone because she has to go to hospital also.
Oh,
So yeah, that is why I want her to give her presentation first.
sure, ma'am. Sure, ma'am. We'll wait for her, ma'am.
Yeah, she's changing her microphone.
Yes, ma'am. Yes, ma'am.
I guess she is going to conference hall. So in the meanwhile, I'll finish my presentation because she'll connect from the conference hall.
Right,
So
ma'am.
I'll share my screen. You please allow me.
Saurav, please allow madam to share her screen, please. Saurav?
Done, sir.
Done,
Yeah.
done, ma'am. Done.
So is my screen visible?
Not yet, ma'am.
Okay. I guess so. It should be visible now.
About to share, ma'am.
Yeah.
About to start your screen. Yes.
It's visible?
Yes, ma'am.
So,
Please put
yeah,
into presentation mode, ma'am.
sure. Is it fine?
Yes, ma'am. Please.
So, yeah, so I would just like at this outset, I would like to thank the organizers for giving this opportunity to us. So in continuation with Dr. Richa's talk, her talk has spoken on the practical aspects, the commercialization, the much needed events which are required in Indian agriculture now. I shall be talking more of something like a part which has been neglected and which can be used, may not be for farmers, but then for the laboratory or the case studies where one sort of aspect has been ignored. So I am going to talk about the biological control of recent invasives. These invasives will include the pest as well as the invasive weeds, and vis-a-vis a positive and negative role of native parasitoids. So this is going to be the outline of my presentation. The three recent invasives of the country which were introduced in Asia, I'm going to talk about them and the negative impact of the native parasitoids there. And two of the important invasive weeds which were native to Southeast Asia with known introduced range outside Asia, and what are the steps for the global biological control initiatives which NBIR is taking. So if I start my presentation with per se, like for the pest, it will be for 3: Rugo spiraling whitefly, fall armyworm, and the cassava mealybug. So coming to the case study 1. So I will be correlating the parasitism and the hyperparasitism and the neglected part, what we have neglected in documentation in Indian scenario. So like as you all are aware, Rugo spiraling whitefly entered the country, and then so we actually, the parasitoid Encarsia guadalupe was known as the primary parasitoid of the spiraling whitefly. So then when the Rugos came, it got acclimatized to the new environment. The environment was old, but the pest was new. So though the Rugo spiraling whitefly was expanding its host range and we were getting parasitoids, there was up to 90% we have documented the parasitism by only one species, that is Encarsia guadalupe. But there is yet another species, Encarsia dispersa, which is present in less than 5% in many of the pockets. So this particular species is also partially responsible for parasitism, though it has not been reported from many other areas. Possibly it was not documented, like nobody has noticed this. Now the story what I'm going to talk about is of the predator Apocrisa evanida, which never came into the picture. So this particular predator is one of the most active predators in coconut ecosystem, and this has been an active predator even when papaya mealybug was there in the country, and it was responsible for many of the predation of papaya mealybug. But then we reported Apocrisa evanida during the time of invasion of papaya mealybug. But however, we realized that all of a sudden the population of Apocrisa evanida is now not noticed in many of the pockets. So then we hunted for the reason. Maybe it is some kind of natural control which is happening, and then we got to know that there is one parasitoid, Perilampus, which was responsible for its control. So basically, even for the Rugo spiraling whitefly, the Apocrypha evanida was present in many pockets of Karnataka, because Karnataka was the place where we used to often go for sampling. And there we simultaneously, in the fields where Apocrypha evanida was present, we also got Perilympus. And then our reports say that after 2 or 3 months, there was complete evasion of Apocrypha evanida because of Perilympus. And the Perilympus was never ever documented in the Indian literature, because this is the one. So always my focus of my topic is that we do take into account the primary parasitoids, but somehow we are not aware of many of the secondary, tertiary hyperparasitoids which are there in multiple tritrophic interactions, which are often getting ignored. Then this is another tritrophic interaction which I am going to talk about in context of Eleurodicus rujuperculatus, because when we were rearing it in the laboratory to check for different kind of parasitoids, we realized that we were getting one parasitoid Oincertus. So usually what happens, the moment if you keep some kind of parasitized twigs and all in the container, and you get parasitoids out of it, it is your general assumption that what is going to be is going to be the parasitoid of the pest. But we do forget that it is actually a niche. It has many other species also which are responsible for the total tritrophic interaction. So there we realized that we were having a scavenger which was thriving on the Rugo spiraling whitefly. It was a very strange scavenger, a tiny one belonging to a very rare families in STD. Because of its tiny nature, possibly it was ignored. And whatever parasitoids we were getting were basically the mummified stages of the Xinestia, that is the dipteran, rather than the Rugo spiraling whitefly. Now, if anybody of you have worked on whitefly, you would have understood that if you collect whitefly and keep it in the containers inside the laboratory or in the cages, it is very difficult. After a day or 2 or 3 days, it is very difficult to separate until unless you are quite expert and you can do it under the microscope. You cannot tell that what is the mummified object, whether it is the pest or it is anything, a predator which has been mummified. So what I mean to say is here is many of the reports what you are seeing in Indian literature, majority I'll tell in the Indian literature, are the misidentifications or the misidentifications of the host which are associated with the main host. So here the parasitoid what we were getting in large numbers, anybody would have reported it as a parasitoid of Rugo's spiraling whitefly, but basically it was not a parasitoid of Rugo's, it was a parasitoid of a scavenger which was thriving along with the Rugo's. Now coming to the fall armyworm, I will not go back into the history details because you must be knowing. I'll just tell you that when in the process from 2018 till date, I have documented along with many of the colleagues of mine like Dr. Richa, Om, and many other colleagues, we have come across, and now at present we have altogether some about 45 species of indigenous parasitoids and predators which are directly or indirectly related with fall armyworm. Amongst these are the 20 parasitoids and the 2 predominant predators. So Dr. Richa has talked about the Chilonis and the Remes. I am only going to talk about the role of these parasitoids in negating the predatory potential in the field conditions. So here comes the anomalous role of Telenomus species complex. Till now, it is debatable that whatever Remes we are having in India is basically a true Remes, or it is a species which has deviated its form from Remes, because really nobody knows the types of Remes. Nobody has examined it. We do not have the type series to compare also. So even we do not by confirmation say that this is Remes or not Remes. We definitely go by molecular and some of the NCBI databases, and we say that it is almost 100% near to Remes. So we have now classified into a Telenomus species complex, which is having the species Remes also and species close to Remes. But then if we are recommending remes for fall armyworm, which is very much effective, there is a very potential predator, Eucanthocona furcillata, which is naturally present in maize ecosystem, in fall armyworm, wherever it is present. It thrives very well, and it is a very good predator. So in the initial first year of its invasion, there was not much parasitism. However, in the due course of time, in 2 years from there, now if you take the Eucanthocona furcillata eggs from the field, if you bring and you just put it in the laboratory, you will find up to 100% parasitism by native parasitoids. So you will have Grion, you have Telenomus. I cannot say which species of Telenomus, but definitely one of the species complex which thrives in fall armyworm, and Trisolcus. So here, what is my apprehension is that it is the same remes which parasitizes the fall armyworm eggs, or it is the remes, or whether that same remes is responsible for bringing down the population of predators also. Somebody has to work on it, because whenever I get samples for identification, people usually send me dead samples. So if they send me live samples, I can ask Dr. Richa to check whether they are parasitizing the fall armyworm eggs or not. So there comes the role that some of these parasitoids, which are native primary parasitoids, are basically serving as hyperparasitism in the case of fall armyworm. So there has been a change in the pattern of parasitism in the field, because if you have made field notes, and from the day of receipt till the date, you will realize that the Indian scenario, the Indian agroecosystem for maize also has changed, and the pest and the parasitoids, they have tried first to parasitize some of them, and then now the range is becoming broader and broader. And in the first year of invasion, what we used to get, now we are not getting in third or fourth year of invasion. That is what is my because I collate this data from all the samples which I get all over the country. So in the first year of invasion, there was a lot of Chelonis group, Formosanus and Blackburny. I am talking about natural parasitism, Campylolitis and Potacia. But as you go into 2 to 3 years of parasitism, you have more of ichneumonids coming into the picture. Possibly they took some time to acclimatize to the new invasive pest, and we have more of braconids and ichneumonids of different genera, which were not reported in the first, or possibly they were not noticed, or they were not reported, but then they were not reported. So there is now a range of parasitoids along, which is expanding day by day. So now comes a very interesting phenomenon, what we had observed in the recent cassava mealybug invasion. Not talking about the background and all, because you must be knowing, being entomologists, that it has entered the country, and we were not the first Asians to have received it. Thailand was the one who has received it. Now the problem, or the concern, the topic of concern for Indians is that it does have reproductive ability on other hosts like Alternanthera, Seychelles, and Cinderella, which I have mentioned. So from India, so in other places, it is focused only to cassava, but in India, the point of concern is that it does have many other weeds where it can reproduce and it can proliferate. So the threat is much more higher in India. So, just to start with, the damage scale which started cassava mealybug in the country was on a very, very high scale. So it was like more than 1,000 CMB numbers per shoot tip. So it was the highest of the highest scale, which we have calculated based on the standard protocols which are given. So our issue started that we had the pest with us, and we kept on trying to see whether we do have the parasitoid or not, because there was a lot of confusion to import or not. So you must all be aware that we have to import a parasitoid, we have to first prove that the country does not have its own native parasitoid, because there shouldn't be a competition, and you should not bring something from outside and create unnecessary competition in the Indian agroecosystem. So you have to be doubly sure. So we got to know one particular video which surfaced, and there was one parasitoid, and the gentleman was rapidly showing the video and distributing the parasitoids in Tamil Nadu to the neighboring farmers, telling this is the parasitoid of cassava mealybug. So we got the clip, and with the clip we could tell that it is not even that genus. And Dr. Late Rabindra was the key person who brought us into the notice that this clip is getting circulated and farmers are trying to procure this particular parasitoid. Then when we got the sample, we confirmed it that this is not Anagyrus lopezi, which we were looking for it. And then based on our report from NBIR, as a team, we had published a cautionary note that there is no primary parasitoid present in the country. And then based on that report, that was the basis for us to go for the importation. And with the help of IITA Benin, we could, on gratis, we could get the Anagyrus lopezi. And 7th March, we had gone for the first release of Anagyrus lopezi in the field. Now, what I'm going to tell here is that the gentleman who was circulating the video was basically the parasitoid of a predator. So he had the predator, the most predominant predator in cassava mealybug is the Hyperaspis menderoni, which is one of the coccinellid. And in Hyperaspis menderoni, up to 80 to 90% parasitism you will get from Homalotilus turkmanicus. So the gentleman was basically supplying Homalotilus to the farmers, which actually created more harm to the niche rather than basically he was killing the predators, the natural predators. So here I would tell, then we thought of studying the niche, and when we came across, we thought that it is a very complicated niche with many predators which themselves have inter and intra-specific competition. Along with that, now at present, when the invasion started, it was only papaya mealybug to some extent in cassava fields, and it was added by cassava mealybug. So papaya mealybug was never crossing ETL level in cassava, but then with the addition of cassava mealybug, there was economic injury. Then came on addition. Now at present, there are 7 mealybug consortia which is present in cassava mealybug. Now, if you are going to report any parasitoid, please make sure that you have 7 complex consortia of species of mealybugs, plus you have n number of predators, which I am mentioning here in the red marks are the one which are the predominant predators and their successive parasitoids, as well as there are many scavengers and many lepidopterans, which still we do not are unable to say what is the specific role of these lepidopterans in that particular ecosystem. They can be just a niche sharer without any prominent role, or they can play any prominent role, but we haven't proved it yet. So basically, what was the pathways? What we were getting? We were getting the predominant predator. So I'll just start with coccinellid Hyperaspis. Then you had the parasitoids of Hyperaspis, and then you had the hyperparasitoids of Hyperaspis. Finally, you had multiple neuropterans. I could not get hold of Chrysoperla zestrovi salami in cassava, but then it's not strange that if we get in days to come. But then we do have Malada, Pseudomalada, and Apertochrisa, and then the parasitoids of Pseudomalada and all. Then definitely no parasitoids of geocorids and spiders I have documented as of now. Huge chunk of lepidopterans we had, and then the secondary parasitoids and the hyperparasitoids. So then, the moral of the story here it is, when we with little bit of experience and whatever knowledge, we know that this has come from this. If we are not able to ascertain that this is particular the species of this, then it is very difficult for anybody to just report any species out of, because you really do not until you have read it. This we have read it. So what we did, we brought the cassava, we cut it into twigs. Each and every predator and the mealybug was separated physically under the microscope, and then we have come out with this. So if you are just going to keep them in one container in a cage with n number of these species, then definitely what is going to come out of it, you will not know what is coming out of what. So basically we had, now if I just tell you, we have these 4th trophic level hyperparasitoids, which are all responsible for bringing down the population of predators in the native predators in the field, so thereby negating the overall biological control potential of CMB. Then we had third trophic level predators, which were very prone to parasitism, plus they had high interspecific and intraspecific competition amongst themselves. And then the scavengers, which I've told that many of the scavengers or the unspecified lepidopterans which were present in the ecosystem. So here, if you see the two predominant predators amongst the coccinellids was the Hyperespis and Malada amongst the neuropterans. Both were competing amongst each other, plus both were competing with their parasitoids to win the race. So that was the scenario in CMB. So now coming to this, these were the three case studies which I had dealt with the pest, the recent pest. I'll just talk about something that where biological control we also can make use of and people can make use of it. It's not always that we import the parasitoids from other countries and get benefited. We also supply parasitoids to others so that other countries get benefited. So since 2014, NBIR has been under the leadership of Dr. Balal. We have been in association with a global platform where the invasion of the weed has happened from the Himalayas into the Europe, and it is creating a problem there. So basically there are two important weeds, Hydicium and rubus. The native host is Himalayas. And so now what we are doing is that in Himalayas, we are hunting for the biocontrol agents so that we can pick those biocontrol agents and we can multiply them and supply them to the foreign foreign bodies, wherever places where these particular weeds are. Because what has happened is that the weed has traveled, maybe because of seed or by manual, whatever it is, but the biocontrol agents have not traveled. So here what I'm going to tell is the major natural enemy which we came across was the stem borer or the stem fly. What you talk, we have a Mirochlorops species complex which we are targeting, and this is the biocontrol agent which is responsible for bringing down the reproductive potential of that particular weed. Now there comes the role of yet another native parasitoid bank, which is responsible for bringing down the population of Mirochlorops. So time and again, what we were supplying was Mirochlorops. We were working on Mirochlorops, but nobody thought of hyperparasitoids, which are basically bringing down. So in some pockets, the Mirochlorops was not working. So then when we did massive sampling, we realized that there are parasitoids which are basically the hyperparasitoids affecting the Mirochlorops. So the Mirochlorops is not able to develop. So the briconids in general, which we consider as the good parasitoids, are basically giving the negative impact. And from 2014, when I started my survey, I used to get some species of pteromalids and like Calidula and Chlorocystis. And 2018 in large numbers, when the consignment was cancelled and it was kept in India only, when we studied that consignment, we realized we were getting two different species based on the stem. So you can see how the speciation happens based on the thickness of the stem. Because if you see Hydicium, it will be with a small girth or with a huge girth. So the first Mirochlorops evolved because it has to go inside the stem. Then this parasitoid evolved. You can see a difference in the ovipositor length. So the species has evolved. There are two different species at present, which I have documented, which are responsible for bringing down the population of Mirochlorops. So this, we have published that it is not that only Mirochlorops is going to solve the problem. If in the places where they are releasing Mirochlorops and they have hyperparasitoids which have the potential to bring down the population of Mirochlorops will not work in those areas. That was the essence of our studies. Secondly, in 2019, for the rubus, yet another weed in the Himalayan range, when we surveyed, we brought back many samples, and we realized, so our target is whatever kills rubus. So our target was the pseudococcus, which was responsible to kill, this millibug was responsible to kill the, so we were recommending pseudococcus for the next release program. Then when when Dr. Joshi kept pseudococcus for identification and giving us the species name, we realized we are getting parasitoids of pseudococcus. So we were getting Anagyrus out of it. So this particular publication of ours is still in press. It is yet to be published. So we here again putting one kind of caution there that suppose if you're going for pseudococcus, then you have to be very careful because you have anagyrus as a species which is responsible to bring down the population of pseudococcus. So this particular thing. So now coming back, coming away from the invasives, there are n number of parasitoids which are responsible to bring down the population of beneficials also. And time and again, people have ignored it, but as and when we have come across, we have published them. So like for the silkworm, we have got one, this is the Assam silkworm, Cotacia in gregarious form, which was bringing down the population of silkworm there in natural conditions, that we have reported. Then for many number of bees and all also. So what I mean to say is that the crux of my presentation is that the general perception is that all species of parasitoids are beneficial. So not all species of indigenous parasitoids are beneficial. Some are really playing a negative and a very distinct negative role. So many of the hasty reports and discoveries are based on misidentifications and misinterpretations. So we as entomologists have to be very careful in documenting anything as a first report. So because it has many repercussions. So this is an interesting research area with many prospects of discovery and discoveries of many unknown ecological interactions. So these need attention. So with this, I would just like to wind up my presentation, and I look forward for questions. I thank the organizers, especially ICAR-NRIIPM and Manage, for giving us this opportunity, and definitely Director for choosing us to share our views. This is all.
Thank you, madam. Thank you so much. And to add to your presentation, unfortunately, in the nature, we don't have, I feel that we don't have any control over the natural predation upon the biocontrol agents, which has been augmented for management of insect pests. But we, as an entomologist, should think over to what extent we can do some researchable things in these lines. Okay, thank you, ma'am. Thank you. Now it's open for discussion quickly because Madam Gracie is waiting. She needs to go to a hospital, please. Then we'll do one thing, ma'am. Ma'am Gracie, ma'am, please you start your presentation. At the end, we can have a discussion.
Sure,
is it okay,
sure.
ma'am? Is it okay?
Yes, fine. Perfect.
Please.
Very good afternoon. I mean, in another 5 minutes, Sadi. Okay. Good morning, everyone. I'm sorry that there was some problem with my
Saurav?
host. Should enable.
Yes, ma'am. I'm this. Saurav? Saurav, please allow ma'am to share the screen, please. Ma'am?
Yeah. ICAR-NBR I joined.
Ma'am, just give me a. I'll go there, please.
So before I start my slide, I would like to say a brief what I'm going to talk about you. I mean, I don't know how this will fit for you, this thing, because what I'm going to talk about is DNA barcodes. Okay, recent days, many people you would have heard and some people would not have heard. I don't know how many of you heard about DNA barcode. This is nothing but like how my colleague Dr. Angita spoke about. Actually, she's working on taxonomy. She explained about all the parasitoids. How
You share your screen, ma'am. You can share your screen.
yeah. Thank you.
Yes, ma'am. Yes, ma'am. Visible. Please put to presentation mode, please.
Yeah, it's not coming. 1 second. It's taking time. Yeah.
Perfectly.
Very good morning, everyone. I'm Dr. Gandhi Gracie from ICAR-NBR. Let me go into my slide. Okay. Today I'm going to talk about actually DNA barcodes. What is DNA barcodes? Why we need DNA barcodes? And what is the advantage of DNA barcodes? And what is the challenge? And what is the precautions when we do DNA barcodes we have to take care? So as we know that this is nothing but as evolutionary studies we have made, like how to identify the species, everything. So how they have the identity of the species is nothing but the relativeness of the species. How in nature the organism are interacting each other, and the same species how it is interbreeding, and in ecology how the gene flow is happening. So at the time when the gene flow stops, then there will be a variation. Either it can be seen as a morphological like eye color or other characters, or it can be visible in the genome level also. So definitely in the genome level there will be changes. So what people have done, taking that as a consideration, they have identified some of the marker gene, because whole genome we cannot go. So marker gene which can reflect actually the what is happening in the ecology, nature. If there is a gene flow, so diversity will be there. So based on the diversity, they are delineating the species. So this is what in a nutshell, this is what actually DNA barcode or molecular characterization. Okay, it is nothing but DNA barcode, nothing but the part of the genome. It can be any gene, anything, no, coding, non-coding, or any organelle or extracellular mitochondrial, whatever you say, it is the part of the genome to determine the position of the individual in taxonomical hierarchy. Okay, you should be very careful to determine the position of the individual in the hierarchy, either genera, species, subspecies, whatever. We can use this, the characteristics of the gene when it is used, that is called DNA barcode. So we know what is DNA. It is deoxyribonucleic acid. I'll skip all these things. So, and central dogma of life, why we are seeing DNA is DNA will be converted into mRNA and it will be translated into protein. This protein is only doing all the functions. So any changes in the DNA will be reflected in the protein, that ultimately reflected in the characters. Okay, that's why we are seeing the DNA. We are using DNA as, see how in morphology we are using characters, morphological characters. Same way we are using the changes in the DNA as a for the identity of the species. Okay, so what is DNA barcoding? What here we are saying, obtaining DNA sequence from one small reference genes or whatever genes, it can be multiple, assign sequence from unknown individual to species. So unknown individual specimen, we are assigning the species name. So that is the DNA barcoding. Okay, this will enhance the discovery of new species, because there are a lot of cryptic and a lot of disadvantages are there. I will discuss it later. This was first, the idea was long before it was existing, but Paul Hubbard made it in a progress in 2003. Okay, this is one of the gene the Paul Hubbard finding is mitochondrial CO1 or CO2 or entire gene. Nowadays, it's not only sticking with mitochondrial, it can be anything which will give you the data to delineate your species, information to delineate. So I will skip these things. So what we are going to do in DNA barcode, we are going to collect the sample, then take the tissue and isolate the DNA. Then once the DNA is isolated, we are going to see the particular gene. We have to target a particular gene which can give you the information to identify the species. So for that, we will go for PCR. So PCR, we need the particular genes primer and all other components. Primer is very important. Then you perform the PCR. Then once the PCR is done, your product is ready, your gene is amplified, then go for DNA sequencing. Either you can outsource this DNA sequencing from outside company, mostly Sanger sequencing. This particular PCR product we will call Sanger sequencing chemistry. So that chemistry will be using. There are other things are there. If you are not going PCR, directly after isolating DNA, you can go for meta barcodes, that is NGS platform also you can use. Therein some other bioinformatics analysis are involved. Here it is very simple. Once the DNA sequencing is done, you are going to compare the DNA sequence in the databases. If suppose already some databases, the sequence is already submitted, then you can go ahead. Or if it is not there, yours is new, always you have to identify your specimen with the morphological characters. Am I clear? Here I want to make a clear. If suppose your particular species sequence is not available in the databases, please identify your sample from the taxonomy. So taxonomies are very important. Okay, because there are a lot of things that I will discuss you later. So basic protocol is 3 steps: DNA isolation, and then okay, this is DNA extraction protocol. I will skip this because everybody knows this. A simple steps are involved. One is we have to lysis the cell wall, because in animal we don't have cell membrane. So I mean cell wall, only cell membrane. So with the small lysis, any changes in the osmotic pressure, the cell wall cell membrane will rupture. So easily the DNA will come out, because DNA is stored in the chromosomes. We know that in nucleus, inside the nucleus, and some of them are in the cytoplasm also, like mitochondria, other plasmids. So this all will come out. Then once that usually we will go for sodium acetate or sodium chloride, those things, and SDS, that is soap solution, also will be used to bring the osmotic pressure to rupture the cell membrane. Then once the DNA comes out, we have to precipitate the DNA, that is phenol chloroform or isopropanol ethanol. These are all the things, solvents, which will precipitate the DNA. Once the precipitate DNA, then we will purify the DNA by any absorption method or silica membrane method or only ethanol method. So this is actually conventional, the simple steps. Either you go for conventional method or you go for kit method. This is the simple thing. Kit method, they will not tell what is what. They will say step 1 you have to do, step 2 you have to do. So most of the kit method use the silica membrane, wherein the DS double stranded DNA will be bind to that. Then you can elute with any, even water also you can use, or any solvent like TE buffer. Those things you can elute and you will get the DNA. Okay, this is the simple extraction method. Then once you get the DNA, you have to know it is the entire genome, entire that chromosome content, the entire genome content is there, and with mitochondria and other plasmids, whatever it is, it will be there. So in that you have to target only a particular region which you wanted to sequence. Like in insects, so many characters are there, but only few characters will give you the actual identity, right? The divergence which you are looking for. Same way here. So few genes we are using as marker gene. That particular gene you do primer is very essential. Then you go for the primer and you do a PCR reaction. And this is actually the sample. This is all the amplified. See, like that the product you will get. Then once the product is ready, you send it for sequencing. Once sequence comes, this is the NCBI database, National Center for Biological Information. And this covers EMBL also, that is European Molecular Lab. And another one more is Japan also have a separate database, which is all interlinked in NCBI. So you can use only NCBI. There is some bioinformatics analysis called BLAST. So which will give you the result, your sequence. Like simple Google, you are searching in Google, right? How you will search, you will give the keyword and it will go and match with other thing and retrieve the data and give you. Suppose some Maruka Vittata, like that you are giving, then all the information Google will retrieve and give. Same way here, the BLAST, this is basic local alignment search tool. So what it is giving is whatever the sequence you got, you are giving there and searching. It will go and search against the databases and it will bring you and give you the information, so that you know if somebody already identified the specimen and submitted, then your specimen will match exactly up to 99%. It will match. Then you can say it is same species. Even less than 99, you don't think that it is the same species. So please make identity in any other manners. With taxonomy or any other thing. So there are another thing, important thing is why I am telling right now, it is in the concept, it is in the progress. The database is not complete. Even many of the insects are not barcoded. So this barcode of data lipase, that is the bold databases, it was the initiative of many countries, and mainly it is located in Ontario Institute in Canada. That is Dr. Paul Hubbard. He has started his work. So these also, apart from national NCBI databases, in bold databases, you will get the barcode information. This is exclusively for all animals and plants. It is there. Okay. So you can search barcode of life databases are bold. So this will give you, here also you can you can match your sequence and you can find out. This database also have some of the insects identified and the sequence barcoded sequence. So we are actually in NBIR, we are doing barcodes from since 2011. These are all some of the information we have done so far, 1500 more than. This is how the barcode. So in GenBank, you will get only GenBank accession number. When you submit to board, you will get the barcode picture and also barcode ID you will get. That is from the bold databases. Okay, this picture, what is the future is, you no need to like shops, right? They have the barcodes like that. It is nothing but ATGC, this color difference. Okay, the same thing. They will, they are to develop the barcode picture. It is already developed in test stage. These are all the barcodes like I have done for a PCD example. So like that only, this is fruit fly. Okay. Before going to barcode, what is the okay, this is actually barcoding must validate the current taxonomy, because whatever like right now what kind of taxonomy we are systematics we are following is CLAD 6. It is very well fit. This I will skip because now the trend is integrative taxonomy, both morphology as well as molecular. You have to give the data and it should complement each other. And right now we are doing that. Even our taxonomists are collaborating with us, and we have come out with many publications using integrative taxonomy. This is, as I said, it is a barcode of life databases. These are all the partners in there. And from NBIR, we have some 16 projects registered in this barcode of life database. So you can see how many countries, more than 20 projects. These are all the places here in NBIR. We have more than 20 projects. Okay, these are all the information when you submit to the. So what is coming to the advantage of DNA barcoding? The major advantage is faster and accurate identification. We can't like, as you know, taxonomy, even one person to excel in one particular group, it will take many years. So, but in case of barcode, if you know the procedure and if you know biotechnology, anybody can do this. So this is the major advantage. And even different life stages from egg to adult stage, pupa, even pupa, larva, any stages it can be used because the DNA is going to be the same. And cryptic species also can be identified. That is the advantage which cannot be identified through the morphological character. And some of them sexual dimorphic. We know many species are sexually dimorphism. If you see male and female differently, and you might end up in different identification. So, and food contamination, these two things quickly I am going to tell few example. And also it is useful in biodiversity quantification and conservation. Some of the IPR related issues, like some of them are protected animals. So those things easily we can use with DNA barcode. And biosecurity also, like invasive species, we have done when Spodoptera furciperda and tapioca, that mealybug when it came. So we used quickly identified using molecular work. And also, of course, taxonomy is the first, and also this is the further, because it is very easy and simple. So all the things we could identify easily. And protection of endangered animals like olive turtle. So if any dispute is there, we can use DNA barcode and we can identify food contamination and solving some of the legal cases like cow or buffalo, those things and all we can easily identify. This I will skip. This is the actually different. It is very well fit into different what to say, whether evolutionary systematic, biodiversity assessment for ecology or diagnostic. This will always the DNA barcode has the advantage. So various needs, various faculty, it will cater the various needs of the different faculties. So that's what I wanted to tell. There are some limitations here. So only Cohen genes has some limitation. Nowadays we are coming with other markers also. So many markers comes, then how to go about, there are, okay. I have said that all stages you can use. And once I said even a small part can be used for identifying the species, wherein the small part cannot be used for the you cannot identify using morphology. So this is the one example in our NBR we did one drug that is actually medicine which was contaminated with the wings. So only wings had come and nobody could identify, and from the wings we isolated the DNA and did DNA barcode and we could identify. Not only wings, then in some other drugs, actually it is a cancer drug, some cockroach leg was there. So that also we have identified. Then pharmaceutical company is sending the sample whenever contamination comes. So in that way, this DNA barcodes have many advantages. And I said sexual dimorphism. This is one of the example. I worked in speciday. You can see male and female entirely look different. So if you are collecting this fellow from somewhere and this female from somewhere, then you will think that they are both are different species. So in this case, actually, you can you see the in general it has 12 segment in antenna. This has 3 segment female. Like that, so many changes will be there. Here you can see, for example, I worked on Carinostricmus castratus. This is the new record to India. So in order to confirm, then male, female, to make it, I used, apart from molecular, morphology, I used molecular characters also, DNA barcodes also I did. And another example is, this is female, similar looking females. This is different species and this is different species. This is Griffus and Congurus. They look exactly similar. So these kind of things we can use DNA barcode. And when we did the diversity studies, we could identify which is the female of that particular species. Then another thing is that Carinostricmus attarimus, it was first described in 1971. After the, based on the only female, nobody know how male looks, because this is actually sexual dimorphism is there. So that person Turner, he described from India in Nilgiri. Okay, he described this particular species. After that, nobody, based on only female, he did not get male. So how to identify now male? So this kind of things, DNA barcode will give you the advantage. So I have done and I have identified the male, and it has been published also. After 100 years almost, you see here the character male and female. So it looks entirely different, but when we do barcode, it is exactly matching. So this has been submitted. And what is the future of barcoding? The future of barcoding is just if you develop any databases, then that barcode database it is doing. So you go about mobile van, you go anywhere and collect the insect, and there is this all DNA extraction, PCR, and the sequencing and bioinformatics analysis, everything will be done with a small machine. So you just put your insect, collect the insect and put, it will isolate DNA and it will do PCR and it will go and match with the database and it will retrieve and it will give you the information. This is the insect which you are collected. So that is the idea. So they are actually come up with the barcode reader. So many of you have seen the video also. Like last year in WhatsApp, the video was circulating from the Ontario Institute. They have developed that barcode reader. Another important thing is even there are something called NGS meta barcode, like pool of sample, like next generation sequencing. It is high throughput sequencing of like whole genome, those things we are doing, no, like that it will be. So there it will be useful how they will identify when the database are ready, based on the how to explain you. Okay, there are something called each organism will have the based on the DNA, it will have the melt curve. When we analyze after NGS, it will give you. So it is quantification, diversity quantification can be done using the meta barcode. It is not only, you no need to go for individual insect. And if you have the database of already identified all the insects, then that sequence can go and match the database and it retrieve and give. So this is what it is going to be in future. So this concept will help in every way. Suppose in one ecosystem, what is the diversity and what are the insects? So how much time and energy need if you go with a conventional method? So this kind of advantage will give you the very robust data, as well as it will solve the problem in no time. This is the barcode reader. I was talking, right? When you put one, then it will give you the barcode and it will give you the picture, everything. Because this all should be stored, it should be stored in the databases. So right now, the bold is in the progress mode. They are making the databases. It's not completed. You know what is the diversity of animal and insects? It's huge, but it will happen soon. Maybe not in 10 years, maybe 50 years or down the line, it is going to happen. And I want to thank you very much for giving me opportunity, especially my director, NBR director, and also NCIPM director, and also the course coordinators and course directors. Thank you very much, everyone, and all the participants. Thank you very much.
Thank you very much, madam. Please unshare your screen. And participants, quickly, if you have one or two questions.
Amit,
Participants.
thank you, sir. Clear करना है क्या?
Okay. If there are no questions, actually the lecture delivered by Madam Ankita Gupta and Gracie Madam is much relevant to the academics. I know, I can understand the farmers or the other than academics may not have much aware of these things, but still, if there is anything to be clarified, please do write your queries in chat box. We'll get back to the speakers, and we are always there to answer. Our speakers are always there to answer your queries. Thank you very much, ma'am. On behalf of NCIPM and Manage, we wholeheartedly thank, first of all, Director NBIR to nominate the eminent scientist who has extensively covered the information on biocontrol, including DNA barcoding, molecular aspects of biological control. Thank you very much, ma'am. And we sincerely thank Madam Richa Vasni, Madam Ankita Gupta, and Madam Gracie for sparing your valuable time to enhance the knowledge in the field of biocontrol to our participants. Thank you very much, ma'am.
Thank you. Thank you.
Now, I request Dr. Rekha Madam to please introduce our next speaker. And ma'am, over to you, please.
Thank you, Dr. Raghavin. And from my side also, I place a very heartfelt thank you from to all the scientists from VIR for giving the very valuable time and very exhaustive lectures on predators and parasitoids in biological control. Next is a very important lecture by on integrated pest management in vegetable crops by a very eminent scientist from NCIPM, Dr. M.N. Bhatt sir. Sir is working he is leading the vegetable team of NCIPM, and he has a very vast experience of working in the vegetables, specifically in the potato. While he was working in CPRI, he worked a lot on late blight of potato, and in NCIPM, he has taken up the task further, and he has worked on many crops, including brinjal, okra, and he is working in the ICT area of ICT also related to vegetable crops for sending the advisories and all. So I'll not take much time not take much time, and I'll request Dr. Amin Bhatt sir to please deliver his lecture, sir.
Oh, thank you very much, Rekha ji. Had a lot of talk on basic aspects on parasitoids, and it is a good orientation. Now I can move to applied aspects of same along with the other practices in integrated pest management. It's a very wide topic. It's a very large topic. So I will try to be brief, just tell as much as abstract type possible. The title is Integrated Pest Management in Vegetable Crops. Am I audible rather? Am I audible, sir?
Yes,
Hello?
yes, sir. Yes, sir. Yes.
Acha, good afternoon to everyone, all participants from across the country. So now turn is on IPM and vegetables. So vegetable, as I told, it's a large number of crops. India has tropical, subtropical, temperate. See, broadly under solanaceous, I'll be telling about the major crops only, not all the crops. See, if you see that chili, tomato, potato, and brinjal are major crops across the country under solanaceous crops. Under cold crops, we have got cabbage, cauliflower, mostly hilly areas, temperate region, but it is domesticated now. Throughout India, even including Kerala, you can see cabbage, cauliflower quite happily growing. Nolcol and broccoli. Cucurbits, all sorts of melons. We have got many types of gourds. Under leguminous, we have got peas, French bean and cowpea. Bulbous, we have great demand and continuous supply is required of onion and garlic. Among the leafy vegetables, we have got quite a large genotypes like Basala, Amaranthus, Chinopodium, Palak, Pudina, Sag, Drumstick. These come from various parts of the country. I think everyone will agree with me that India is virtually a vegetable herbarium of the world. So if you go to Northeast, if you go to western side of the country, that is Rajasthan, see the northwestern India, that including Indus Plain, central region, that is mostly Madhya Pradesh and surrounding states, western and eastern peninsula regions, that is southern country, southern part of country. In all these regions, we have continuous cultivation of various genotypes of crops. If you see from Rajasthan, we have got very unique, some of the crops like Matira, we have got snap melon, we have something like kachri, we have got long melon, and cluster bean is also there. So we have got quite a large number of vegetables grown across the country, suiting the climate requirement for that particular crop. Why vegetable is so important in our day-to-day life? See, cereals and pulses can at the maximum supply carbohydrates and protein requirements, but where to go for vitamin C and vitamin series, various minerals and dietary fibers. These things can be supplied only by vegetables, and they are very good sources of antioxidants. So they give good nutrition security to the country also. If you come to the yield, overall yield of the crop, cereals and pulses can maximum give 5 to 6 tons per hectare on an average, I'm telling. But various vegetable crops like a tomato and all can go even up to 25, 30 tons per acre. On an average, you will get at least 20 tons per acre, tomato and other vegetable crops. So about 4 to 5 times more, even up to 10 times more, you will get yield through vegetables. Short duration crop, 90 to 100 days, you can easily accommodate 3 to 4 crops in a year. Labor intensive, right from sowing till harvest, you need continuous labors for various activities, and they also earn good foreign exchange. Coming to the area and production for the last 30 years, if you go 30 years back, the area under vegetables was hardly 6 or 5.5 million hectares with production of about 58 to 60 million tons. But of late, 30 years later, we have production of about 3 to 3.5 times of production what we had during 30 years back. Area also almost doubled during this period. Productivity also has gone up much high from 10.5 to 18.4 tons per hectare. UP and West Bengal are the major clients. They are the major stakeholders in vegetable production, and followed by Madhya Pradesh, Bihar, Gujarat are the major vegetable growing states in India. And the figures here given, they represent the major crops grown across the country. Why? Why increase in productivity and why there is a production during last few years? One thing is very clear. Now all the research institutes, both central and state governments and private agencies are seriously engaged in release of high producing varieties in various crops, and now of late they also look into entertaining or ingressing genes resistant to, say, about a DBM, late beta potato, shoot and fruit borer of brinjal. So policy has now creeping in that they should look into producing resistant varieties. Even leaf curl also they are taking very seriously. And when released, all those crops are bred for resistance against the local or national level serious pests, insect pests and diseases. We have got quite improvement in nutrient and water management. Digital system has come, so we can check or regulate the water and nutrient movement. We have got quite advanced vermiculture and biofertilizers. Organic farming is booming up. Protected cultivation is giving encouragement to go for seasonal crops or unseasonal crops rather. Integrated pest management is also adding to the higher productivity. Biocontrol is taking over so that residues problems is reduced to great extent. Molecular diagnostic techniques, faster diagnostic techniques have come up, especially for viral diseases. Then we have got many locally formed vegetable growers cooperative, which gives time to time information on market price and various technologies. Then added to this, now ICT, information and communication technology, is booming up, and thanks to ICT and advancement in this digital per se, the whole India, the various institutes are seriously engaged in developing mobile application apps, and NCIPM also having a lot of apps, maybe through a development of IFC, that is insecticide and fungicide calculators. With a few buttons, you will get a lot of details. Even in IPM, we have got a lot of mobile apps. We have got mobile based pest management information system also we have developed. So it's a continuous process, and we download it in Google Store, and many farmers and stakeholders are taking advantages from time to time. Having said this positive aspects or good looking aspects, we have got many issues still unaddressed or request serious address. That is healthy seedlings, for example. As healthy seedlings getting is a serious concern. Even now also 50 to 60% of the nursery beds are having its own problems. Grading is another problem, sorting out and grading, and marketing is a big issue. Packaging is a major problem. Marketing, I'm telling that there's heavy glut in one part of the country, but there is a scarcity at the same time at other regions of the country for the same this one. So marketing and transporting is a major problem. Still has to be addressed on serious note. Value addition, people are putting a lot of efforts, but still more effort is required so that we'll get more market and foreign exchange. Pesticide residue is a serious problem. Still we are laced with so many chemical pesticides. Farmers go for so many cocktail of pesticides, which is causing residue problem in many of the vegetable harvest. Perishability of vegetables is another aspect. Unlike cereal, pulses, and fruit, this one we cannot keep vegetables for more number of days. This is an issue. Lack of cold storage. So we can have cold storage and preserve these vegetables, but unfortunately we are shortage of cold storage. Though government is seriously looking into this policy. Poor economic condition of farmers also makes him not to take a lot of risks in his venture. And quality of seeds, I'm already telling that good quality seeds is a major issue. only 20% seeds are of good quality seeds. And this is a continuous problem for many years. So we have to look seriously into this availability of quality certified seeds. And most of the times, these private partners like Syngenta, Nunhans, so many private companies, they are having greater share in market and seed distribution. They almost dominant the entire market with more than 80% of market taken over by those people. So they mint lot of profits, leaving behind the farmers at the very first instance of sowing, feeling the pinch of high cost, especially okra, tomato, and all. They are very costly items. 1 kg seed costs about 10,000 to 20,000. Tomato, even few grams, is costing around 2 to 3,000. So cost of cultivation increases. So the seed has to, good quality seed has to available at lower rates. Now, slowly we are venturing into the insects and diseases per se, but we will try to, let's see what happened recently in recent years. The pest scenario, maybe the case of too many release of varieties and hybrids, maybe climate change, maybe too much of addition of nutrients in the form of NPK. So many factors are running simultaneously. So pest scenario is also changing from time to time. And these are some of the very, very few examples. Take, for example, outbreak of late blight in tomato. Tomato late blight was not as serious in India, especially in UP and Karnataka prior to 2015. But in tomato, it has started in 2016, and now it has most of the places, especially this November, December crops, are severely infested with late blight and have become endemic. Bottle gourd stemmy blight is very notorious, especially in Maharashtra and parts of Punjab and Haryana over the last 5-6 years. It is now slowly spreading to entire country. Tomato leaf curl, as you know, is a Begama virus, is very serious in many parts of the country. Ridgey yellow mosaic disease, again a Begama virus, is becoming a major constraint in gourds everywhere. Virus that is a very serious. It creates a lot of ring-like structure on leaves, and it reduces the plant size. It creates a stunting of the plants, and production is reduced and quite rampant in states of Maharashtra, Karnataka, and Andhra Pradesh, with sometimes 100% incidence. Bacterial wilt, wherever sub-mountain region like Solan or your coastal areas in Odisha, Kerala, Karnataka, and a whole lot of eastern and northeastern states of India are plagued by bacterial wilt, especially in solanaceous crops. Mealy bug is an issue. Everybody knows that it is an alarm in most parts of the country, especially during last 5 to 6 years. Even some other example like in West Bengal, it is very serious in brinjal and okra. And we have got a B biotype, a recent genotype in white fly. It is causing too much havoc in Kolar and other parts, and now it is seen in Andhra Pradesh also. And it is a very good efficient transmitter of leaf curl virus in tomato. Everyone knows about brinjal shoot borer and how serious it is in entire country. It causes 20 to 90% loss in first as a shoot borer and slowly as a fruit borer. And farmers go for very large number of sprays in okra against shoot and fruit borer and later problem of resistance. It is not just in okra, it is same case in brinjal and other crops. Fruit fly is a major havoc, especially during the Kharif and when there's high temperature and high humidity. This is a very great cause of concern, especially in melons and gourds. 30 to 100% loss, even in mango, it causes very serious, it's a very serious pest in those crops. Diamond back moth, especially in North India and South India, of course, depending upon the season and temperature availability, especially in North India during March and April, it is a havoc. No field is left without the incidence of diamond back moth. If you go right now to the entire North India, especially parts of Uttar Pradesh, Haryana, and all, wherever cauliflower is there, right now, as off-season crop in summer, you can see lots of incidents of diamondback moth and a lot of cocktail sprays sprayed, either with a good intention or sometimes unnecessary spray has led to severe outbreak of resistance. Tomato leaf miner tuta absoluta is now embarked upon the Karnataka and Maharashtra since 2014, and now lure is available and standard technology available for this management of this deadly pest. Aerophid and Tetranychidae has become a very serious in many vegetable crops. Hadda beetle is very serious cause of concern in Uttar Pradesh and Bihar. And recently, I was told that some of the farmers have come NCAPM, and Dr. Anup also told me that recently this is Product is a major cause of concern in the entire northern part of Uttar Pradesh and other states, adjoining states, especially in this summer crop like cowpea and other things. Trips, parvi, spinosus, one of my colleague, Dr. K.V. Raghavendra, he knows much about it. He went almost all parts of Andhra Pradesh, especially during November and December of last year. And he knows better than everyone how havoc it caused almost most of the fields in South Indian states, especially Andhra Pradesh. They were plowed in because it is no pesticides are working this particular pest. It was initially noticed somewhere in Kolkata during 2015, and now it has become a very serious pest in not only chili, in some other crops also. And hopefully this year we are well prepared, and they have already come up with some modules, and accordingly we'll be able to, hopefully we'll be able to manage this destructive insect pest in chili. Outbreak of flea beetle is another cause of concern, especially in radish and other things. Dr. K.V. Raghavendra has come up with its report, especially in cruciferous crops like radish and other things. And recently we have seen cauliflower also in Padana and nearby areas in Haryana. Now, added to this, farmers, there are some issues from farmers' side also. Farmers have very poor scientific knowledge on handling of and spraying of pesticides. How to go for the exact quantity of pesticide required. They just go for very rough calculation, and they go for either underdose or overdose of the pesticides. Safety measures like are seldomly practiced by farmers, and you can see they indulge in smoking, chewing tobacco at the time of spraying. Soil testing is a must. Many of the, thanks to government policy to go for the soil testing, the health cards, Mruda Parikshak. This is very important because farmers go for andadunda application of nutrients, urea and all. They go for even 300 kg per sometimes per hectare, even sometimes per acre. They go for a huge quantity of fertilizers, and that also we have to go for carefully. We have to apply because that also decides the flora and fauna of the soil system and the aerial pest problems. We have to discourage unreliable products. Plenty of markets, plenty of products are available in the name of biostimulant, seaweed extracts and humic acid. So kindly all the participants see very seriously look into this aspect. We have to discourage the unregistered firms to get into these products and selling. And overdose of plant growth regulator like your gibberellic acid and all, you have to be very careful. If you go for overdose, then these are the symptoms we can see. So that is applied only at PPA level. So we should be very careful by applying these things. Now, these are some orientation I gave before getting into the IPM per se. Integrated pest management says that we have to use all the available pest control techniques, be it cultural, be it mechanical, be it biological, be it need-based application of pesticides. All should be carefully moduled, and then only we will go for ETL-based pesticide application, so that we are protecting our environment very judiciously and very friendly way. Now, before going to the IPM aspect, let me go to the general approach. What are the concepts? What are the various approaches we can go for in IPM, which are commonly applicable to all the crops, irrespective of pest and diseases? In a way, these will help in prevention of the pests. In a way, they help in prevention of the pests at the very first go. What are those things? We will go one by one. Follow fertilizer-based soil test, weed management, intercropping, sowing time. Sowing time means you have to manipulate. If some disease like downy mildew or some sucking pests are very serious at a particular sowing day, you can either postpone or you can prepone the sowing time. Seed rate, planting density, etc. as per recommendation for the region. Chemical pesticides always keep that are only the last weapon. We have to think that chemical pesticides are only the last weapon in our IPM. Grow locally recommended resistant cultivars for major pests of local importance. For example, Kufri, Giriraj, Kufri Himalini, for example, for hill regions of the country for against late blight of potato. So we can carefully select the region-wise what are the major insect pests, and accordingly we can select resistant cultivars against those pathogens available through Agriculture University or central government institutes. Use only clean certified healthy seeds is very important. And in case of potato, always go for 3 to 4 years of, you have to replace the seeds. You cannot use the same one because there will be degeneration of seeds. So you have to go for replace the seeds. So these are the fundamental techniques in IPM approach. Then do not collect seeds from virus infected plants for seed transmitted viruses like portivirus, cucumovirus, tobamovirus, tobera and virus. In this aspect, the scientists, local scientists can help farmers. How can they do not select the seeds from the these virus infected plants? When you come to prepare nursery with the polythene bags or protest under protected nursery, this is very important. Protect from sucking pests by covering beds with the insect proof. See, these are the simple techniques, but these will have a long benefits in vegetable cultivation. Then you can go for soil solarization for nursery raising. Then you can go for deep summer plowing. These are very historically farmers were doing, but we have forgotten for timing. So we have to again restart this deep summer plowing, deep flowing after the harvest of the crop, so that all the pupa and the pathogen propagules will be exposed for harsh weather. Here you can see some of the birds. They just pick up the pupae and eat and reduce the number of propagules. Always avoid planting in low-lying waterlogged areas. This especially helps in avoiding the bacterial wilt and fungal wilts. Otherwise, what will happen? The bacterial wilt or fungal wilts, these low-lying areas with the water stagnating in one area will predispose those pathogens and will help in initiation of the disease, and they will easily spread to the entire field within overnight. Flow soil tilt to a fine tilt and form basin for approved sites, bed ridges. These are very important aspects. Thinning gap filling to maintain optimum plant population. Rogue of disease plants showing wilting, severe melting, mosaic, crinkle, necrosis. Especially this is very religiously followed in case of potato for seed production. Keep weed free at least for 30 to 45 days by regular hoeing and manual weeding. This will help in avoiding the especially for sucking pests. We can collateral host, what we say, those plants can be we can remove and we can make pest free. And always go for install drip irrigation wherever feasible, because this flood irrigation will help in spread out many of the soil-borne diseases. So this drip irrigation will help in avoiding the weeds, avoiding the soil propagules to spread and multiply and spread the diseases. So it has got numerous advantages. You can save the water also. And repeated application of same chemical insecticide or inappropriate ones, one leads to resurgence of whiteflies, other sucking pests, especially pyrethroids like deltamethrin and other things, cypermethrin. Those things we have to avoid, especially in whitefly and for against whitefly and all. In a way that repeated application of same insect should be avoided. Then we will go for IPM pest-wise across vegetables, because most of the pests, sucking pests, lepidopteran insects, fruit flies, majority of the foliar diseases, majority of the soil-borne diseases, or 50 to 60%, even up to 70% are similar or akin to most of the crops So it is better to talk in terms of pest wise or disease wise rather than going by crop wise. That's why I always go for IPM pest wise across vegetables. For example, see, we can start with the from diseases. We can start with always damping off. How to manage damping off in cruciferous crops, in pulse vegetables, or in your solanaceous crops. So the concept and pathogen is mostly same. Like Pythium F1 is across the crops. It is common whether it is brinjal, whether it is tomato, or in many of the cruciferous crops. So here, what this is a common pathogen across crops, especially in Kharif season. It causes both pre-emergence and post-emergence damping of. It increases either rotting of the seeds before germination, or it will topple the seedlings at 10 to 15 days age. It topples this one and entirely kills the seedlings in the nursery. So this is one of the causal agent of damping off. There are other causal agents like okra root rot, Fusarium. You can see the either in the nursery or just transplanted okra. You can see this Fusarium problem. This is all symbolic only. Like other crops also, we have got many alternate blight in case of cabbage is a major problem. We have got Rhizoctone virestem in case of cruciferous crops like cruciferous crops rather. Tomato, we have got our own problems. So all these crops under different genotypes will have damping off caused by either Pythium, Rhizoctonia, Phytophthora, Fusarium, Macrophomina, Rhizoctonia, all those soil-borne pathogens. Now, how can we deal with these pathogens across crops? One, for example, Pythium, always and mostly it appears during the Kharif season because it ideally requires 20 to 25 degrees centigrade and 95 to 100% and heavy moisture, standing moisture will always encourage a damping of caused by Pythium and Phytophthora and ill-drained soils. So always you go for raised bed nursery. Rhizoctonia is between warm to hot temperature and moderate moisture levels. So these are the broadly two pathogens which are common in case of damping off. So we can look into cultural, biological, and chemical methods of this one. So in IPM, what we insist is that always go for 2 to 3 common per se. Like you go for always soil solarization wherever feasible. This you cannot tell in high altitude places like Shimla or some temperate region because you have to harness or you have to use the high temperature available during June, July in subtropical plains or southern states where you can go for small area, you can roll out the mulches and you can trap the solar radiation, increase the soil temperature by few 5 to 10 degrees centigrade, which will help in killing the propagules and insect pest, pupa and other things. So this way you can, this soil solarization also helps in scientifically proved that they help in release of some of the nutrients and minerals and some soil fixation will be activated by this process. So these things you can take advantage of soil solarization and always you have Trichoderma bacillus or Pseudomonas fluorescens for nursery application or seed treatment. And you can apply through farmyard manure or neem cake. All those things you can even. And always there are some need-based application of fungicide like Captain or Thyram based on the label claim. You can use some of the fungicides if there is any still if there is any problem of damping off. These are the few techniques: soil solarization, seed treatment like this. Healthy seedlings always go for as much as possible go for pro tray. Here you can use perlite, you can go for peat soil, you can go for vermiculite, even you can go for coir pit. All those things mixed in particular ratio, you can make it as a nursery this base, and you can raise pro tray, which has got very advantage of retention of moisture, and they will allow only the seedlings of healthy nature to grow fast and available for transplanting. This is a very beautiful technology, pro tray technique. This can be harnessed And here, because of the already sterilized material in the as a base, you cannot expect any kind of damping of in pro tray. It's not that you will not get damping of, but in that case, you can go for a need-based application of this one. You can go for need-based application of fungicides. Seedling dip in Pseudomonas fluorescens will help in initial colonization of root by this bioagent. It will have its own advantage at later stages. Always go for nylon netting in nursery because there's a sucking pest like whitefly, which already instead of in place of sucking the plants and degenerating the plants also allows the leaf curl virus to spread faster when you transplant it. So keep in mind that you always go for insect proof net houses invariably so that you can protect your seedlings for the first 30 to 40 days. Then I leave alone those damping of already we have talked at a concept level. Now I will go for only major foliar diseases. We will not discuss several minor diseases which has been quoted in so many scientific academic interest. We will be talking only the major problems. Like next we will go for downy mildew. It's an obligate pathogen. This one is a various cause of concern, especially in cucumber. Right away, right now, if you go through and make a survey in northern plains of Haryana, Uttar Pradesh, and Punjab, right now during the fourth week of April, even third week of April, and unseasonal rains cause lots of incidents and severity of this down mildew in cucumber, the summer crops, especially in summer crop, you can see this severe incidence of down mildew. A whole lot of North India, you can see this. Especially we had severe rainfall about 1 week back. It has caused severe incidence of down mildew. So you have to be very cautious about this particular disease because it spreads like wildfire within no time. This is the upper side symptom. This is the lower side symptom. This is the microscopic organism. You can see these are the symptoms and these are the ideal conditions which I have given in a note I already supplied to you. You can go through it when you get time. And these are the various factors which readily spreads the pathogen. These are some of the interventions you can go for managing the down mildew diseases. Always go for local suitable tolerant varieties for down mildew, powdery mildew, late blight potato, and such severe foliar diseases. These are the other physical, chemical, these are the cultural activities you can add on. There are many label claim fungicides and the latest fungicides are there in our baggage, like azoxystrobin is there, meteram is there, pyraclostrobin is there, we have got tebuconazole, we have got pyraclostrobin, all those and we are having label claim for this deadly disease, and you can use very carefully on need based only one or two times in a season, not more than that. This is the powdery mildew. Always powdery mildew is always synonym with the chili powdery mildew because chili is the crop which is severely affected, especially in places like Andhra Pradesh and parts of Karnataka. In the dry season, especially March, April, and it is a very serious cause of concern in those belt. In the when the temperature is moderately high, and even without humid or this mist, it will cause severe this defoliation in crops like chili. So here also what we can do in the initial stage itself, you always prefer some resistant varieties if there is a very serious problem with this powder mildew. If not, you go for only label claim, judicious use of label claim pesticides or fungicides giving proper waiting period after harvest. So ideally you can go for protective 1 or 2 sprays. If there is no control, you can go for need-based systemic fungicides only 1 or maximum 2 times in a season, and that will take care of your chili powder mildew. Then anthracnose is again a very serious cause of concern, especially in chili. You have got a very serious cause of concern in case of bottle gourd, and that distinctly cause severe dieback in case of chili. And within overnight, within say about not overnight, at least 5 to 10 days, entire field under the conditions of high humidity and a few drops of rainfall and highly fertilized nitrogen fertilized crops, you will get this anthracnose in a very deadly fashion. So here also you can just seeing the endemicity of this pathogen, you can go for seed treatment and you can go for a need-based judicious application of label claimed fungicides. Lots of label claimed fungicides are available for chili and one or two fungicides in case of cucurbitaceous crops, particularly bottle gourd. So these are the conditions. I have given all this in detail my notes. Here we will talk some more only deadly diseases like you must have heard about a little bit of tomato, you must have heard about a little bit of potato. Little bit of potato is a recurring problem in hilly areas, but not to be in plain areas where it causes disastrous once in 3 to 4 years. Little bit of potato, but last few years in parts of Mirzapur and parts of Uttar Pradesh, you can see this little bit of tomato coming almost every year during October, November. Thanks to many of the institutes, they are working day and night to develop a wet-based decision support system, especially for diseases like late blight of potato. We have got many support system from Punjab. We have got from Meerut, that is Modipuram Potato Research Center. We have from Shimla. We have got from West Bengal. So all those research institutes are having their own this prophylactic and this warning systems well placed in their jurisdiction, and they give time to time the warning system when the disease will come and what to do and what not to do. Next disease is early blight. It is almost synonymous with the early tomato crop. Wherever tomato crop is there, there is early blight is there. Be it in Kanyakumari, be it in Kashmir, be it in Arunachal Pradesh, be it in Rajasthan, wherever you go, tomato early blight will be there. This with this particular typical symptom, it affects both leaves, it affects the stems, it affects the fruits, especially in the blossom region. And this is the one here also nothing to worry, because lots of private firms have released or marketed label claim fungicides are available, but see that it will not be used injudiciously, and see that lot of waiting period is given appropriately. Another deadly disease coming to my mind immediately is Phomopsis blight in case of brinjal and Sclerotium rot in case of brinjal. This is the typical symptom of Phomopsis blight in case of brinjal. You can see the pycnidia-like structure at very first instance in the stem, in the shoots, and you can see in the fruits also it will cause rotting. You can see a lot of pycnidia-like structures. Here also you have got lots of, not lots, at least we have got a few label claim fungicides. Use it very judiciously only when it is required and when it is above ETL, and don't go for unnecessary spray when the crop is at the fag end of its crop season. Here this is the best example of how if you give timely spray of chemical fungicides, this is an unsprayed plot of potato. Here it is completely knocked down, and if you give timely the spray, how the plot can be saved by spray of fungicides against late blight of potato. There are other diseases, bacterial black rot. You heard this right since 1950s or long back. This is a common disease in case of cauliflower and cabbage. You have got alternate blight in case of cole crops. You have got stem phylum blight in case of onion. It is a very serious disease, especially in North India. You have got purple blotch, especially in South India during Kharif crop. You have got Sarcospora leaf spot in chili. We have got okra yellow vein mosaic in case of okra. We have got lots of leaf curl viruses, Begamo nature in the chili, tomato, and many cucurbitaceous crop. I already told about Tospa virus in case of tomato. We have got extensive damage vectored by so many sucking pest problems in melons, gourds, solanaceous crops. These are the things. These things I will deal as and when it comes in the further slides. Then next one is still I am in fungal diseases only. Rather I am in diseases only. I have to move to insect pests. Fungal diseases. The first and foremost disease is the wilt caused by many Physarum species It has got broad hosting like watermelon, cucumber, muskmelon, that is karbooj. We have got watermelon, that is tarbooj. We have got bottle kallangadi in Kannada, bottle gourd. We have got bitter gourd, bell pepper, chili. All this is a chili. When I visited parts of Karnataka in Ballari area, this is the condition, is an endemic area. Like this, you can see in Kanpur also. You can see entire country. This is feasibility of chili is a major cause of concern wherever chili is grown. Then feasibility of bell pepper is also serious cause of concern. So sclerotia built in case of this is in the case of a bell pepper. You have got a major problem of gummy stem blight. Already I told you in Maharashtra and parts of Haryana, Punjab, and other northern states. You have got a gummy stem blight last few years. It's a very serious cause of concern. Please take it very seriously and go for appropriate management practices against this disease. Brinjal feasibility is a major problem along with bacterial built of brinjal. Now, all the participants, you also have got very lots of field experience working with very lots of crops. But I just try to take your attention. Look at this vast field of tomato, which I recently visited in Bhiwani in Haryana. At the first instance, if somebody sends it through WhatsApp group, you will say first think of maybe early blight because there is no any death of these plants. Bolke, you may give recommendation for early blight, which I also did, frankly speaking, in the initial time, because when we look into the individual plants also, the symptom is not clear about the sclerotia wilt. Friends, whole field is seriously and severely affected by sclerotia wilt, something of the order of this kind. You could see the lot of mycelial mat with the sclerotia in almost all the crops. But since they are at the various stages of infection, some plants are looking healthy, although they have got infection. So friends, be sure about the always, if at all you got the doubt, always look till the roots. So that you knock down the plants and pluck it and look carefully at the root region also. Then only you will realize what the kind of disease it is. By the time I went to Bhiwani, after 10-15 days, whole field was completely knocked down. Even after so many years of experience, still we are learning. So all friends, be sure about the wilt diseases. Sometimes wilt diseases are fooling us. They do not give the obvious symptom at the overnight, unlike in case of late blight of potato or fissure wilt of chili. There are some diseases which do not give symptoms immediately. These are the various approaches. Always go for trichoderma application either through seeds or soil application, and have patience at least 3 in the endemic areas. You minimum require 3 to 4 years to get good result of trichoderma. So avoid, for example, if at all you get lots of incident of feasibility this year, avoid such fields next year and plunge in lots of trichoderma into those fields, and then make those fields resistant or free from propagules of fusarium or other pathogens. So this is a very important steps in managing the fusarium wilt. And I have to always say that you have to go for summer plowing for obvious reason. Avoid low-lying areas. Always go for good quality trichoderma. Many times you are not getting good quality trichoderma, so go always depend on the local agriculture research institutes or local agriculture research centers who are producing trichoderma, and make it available for farmers. This is the kind of mixing, and you go for see for sometimes growth of trichoderma also. Always go for drip irrigation. Still, if you get this one, I am telling that always go for leveling of the plants, the areas. If you have avoid low-lying areas, if at all you have got low-lying areas, avoid this. So these small things will make you free from wilt diseases. And if there's a serious 1 or 2 plants seen as a 1 or 2 plants, go for drenching with the tebuconazole or carbendazim, or you can go for trifloxystrobin at a local level and on emergence level, and not to the entire field, only at a few spots only you can go, so that you can reduce the spread of the wilt diseases. Bacterial wilt, as the name indicates, is a very serious disease. Because of its seriousness, it is an overnight disease, especially in ginger and solanaceous crops. It is why it is overnight means Today evening you go to the field and see, you are happy that whole field is healthy and whole field is very free from diseases. But very next day when you go, all the plants are just shying down, and they, in spite of the irrigation, they will not come up again. So this is the kind of seriousness bacteriability is. And always go for many of the KU, Kerala Agricultural University, IHR Bangalore, IAVR Varanasi, and OUT, this Bhubaneswar, and your West Bengal universities all have got many resistant varieties against its bacterial wilt, depending upon their major crops in their region. So go for always resistant or tolerant varieties available for bacterial wilt, and always go for this Pseudomonas fluorescens. You can apply 2 kg per acre, and treat the seeds with the talcum formation Pseudomonas fluorescens. Dip seedlings, already I have shown you how best it will control the diseases. These are the things by which you can manage the wilt diseases. Apply bleaching water. In spite of this, if at all there is a serious problem of wilt, you can go for bleaching powder, but don't give in writing because it does not have any label claim. So you have to be very cautious about all this. Then coming to fruit rots, you need especially those plants which do not have any mulching or something like, or which do not have staking, especially this tomato and all. You have got this kind of rotting, buckeye rot. In case of tomato, it's very serious. Always go for staking in such cases, or some paper mulching or some other cheap mulching, and always go for staking. That will take care of everything. Yeah, you go for 1 or 2 protective sprays of zinab, yeah, mancozeb, or copper based fungicides that will take care of your fruit rot. But don't give a frequent, because since it's directly eaten, don't go for frequent this one. There are so many other diseases which I feel that not to deal with, because either they are not very important or local important. Looking at that, you can take your own decision and you can focus on those diseases. But I have got the major diseases are foliar diseases and salmon diseases. Now we'll move to swiftly, because time is very short. We'll go for sucking pests. These are the, because time is already 2 o'clock. So we will try to run off these slides. This is whitefly. First one comes to mind. Here it is a two-pronged problem as a sucking pest. When it crosses ETL, it is really damaging. It causes honeydew because of the excreta and dews, this saliva. So then this sweet material, this honeydew secretes, and this is even transmits deadly viral diseases like begoviruses. And these are the some of the classical examples where they cause too much of problem in vegetable crops. And here these are the managing practices. Always go for seed treatment with the label claim insecticides. Always go for protect seedlings in nursery bed. Already we have talked. Rogue leaf kill infect plant before transplanting. Plant fast growing crop like maize, sorghum, perlite millet as a border crop. Avoid excess nitrogen, very important. Use nylon sticky trap. This we have seen practically in Pradhana and in Haryana. If you go for early yellow sticky traps, you will not get leaf curl diseases, because you will get about 100 to 200 white flies within no time caught up in the yellow sticky trap. So you can reduce the population, remove the weed plants, control natural enemies like Encarsia. Encarsia is very specific for female eggs are laid internally in white fly nymphs. So Encarsia is very good parasitoid against white fly. Just you can inundative release, you can make 2 to 3 sprays of neem kernel extract, and avoid synthetic pyrethroids. Already I have told deltamethrin you avoid only 1 spray per season. You can go for diaphantheron, that Pegasus, and Santalopron. This have got label claim, and these are ETL already given in the notes. Then this is a thrips. This is slender, very fast multiplying insect, and onion, cucumber, bell pepper, and chili are severe infested. It causes transmission of. This is a very clear symptom of transpovirus. Round ring-shaped spots it causes in the growing tips and other young leaves, and in this fruits also it causes round spot. These are the various symptoms in fruits. And chili, you know, it causes bloating in case of chili, and you know how common, how friendly it is with the onion. More than 25 to 30 is a common. That's why this ETL is also very high in case of onion. It's about 25 trips per leaf in case of onion. because it's so common in case of this one. So all these things, so many things have, there's a lot of basic studies has been done, why it is difficult to control, why, and it is a lot of generation they have done. Biology they have studied in detail, all those things. They have got many label claim insecticides which you can do. They have done lots of forecasting and this epidemiology aspects. These are the, some of the triggered. Then, as I told you, some of the cultural practices are very common for sucking pests, and these I have given in my note. So you can go through, friends, all those things, and you can go for label claim insecticides for white fly, and you can go for onion, even sprinkler irrigation, which will run off the thrips, and you can reduce the infestation. Now we'll come to 1 second. I will take for thrips. That is black thrips. Recently we had in Andhra Pradesh. It causes heavy flower damage. First it is appeared in the leaves, and then it slowly, and it is preferred over these flowers, and causes complete flower drops in case of chili. It's a huge loss, 90 to 100% loss in many of the districts in Andhra Pradesh and Karnataka. These are some of the modules they have developed, and I have given it in our and that you can go through. These are some of the label claim insecticides which will work if used judiciously. You can spray Beauveria bassiana at the initial stages. You can go for Pseudomonas fluorescens also, or you can go for Bacillus albus NBA strain. So these things will take care of your black thrips, but still it is a very challenging pest. So you have to give a model approach to manage this particular. Then aphids, when you talk about aphids, immediately comes to the cruciferous crop like cauliflower, cabbage, and also potato. In these two crops, it is very serious at various seasons in the entire country. So these are the cloudy and humid weather favors rapid multiplication. The moment you reach February or March, first week in North India, you will get lots of infestation aphids in coal crops. So in irrespective of the crops, you will get this aphid infestation in North India. And these are the some of the modules specifically are dealt against these aphids. This I will not read it. You can read of these are the ETLs worked out against in various crops. Then you have got hopper. Whenever you get hopper, first thing comes to mind is okra. In okra, it is very serious, especially if you go right now to the entire North Indian belt, okra, wherever crop is there, you will not get crop without okra hoppers and fruit borers. This is very, very important pest in case of okra, and it's also very serious in case of brinjal. So, thanks to DPPQ, they have got many label claim insects like thimethoxam. We have got imidacloprid, and we have got santonprole. So many, there are many label claim insecticides for this jacids. And there are many tolerant cultivars. If you go to literature, and depending upon the locality, you have got many tolerant varieties bred for this particular pest, right from Kerala to Kashmir. So you can use it depending upon your local variety demand. Then these are the some of the modules. You can intend to release of chrysoperla. You can go for this parasitoid, and you can these are the ETLs, and this is the mites, another deadly pest. Through saliva, through injection of saliva, it causes the particular type of symptom called inverse boating. You can see the leaves just curling downwards, especially in case of potato, and it is also very serious in case of chili. But in chili, it is Polyphago tarsonemus latus. But in case of okra, it is mostly red spider mite. If this is a very deadly disease, I can tell you, it causes webbing. In case of okra, I have seen, especially now May, June, July, in entire North India, it is very serious in case of okra, red spider mite. Chili mite is also very serious, especially during February and March in case of South India. So these are the things to note down. Lot of label claim insecticides are available like spiromesifen, Spiromesifen is there, Vermitech is there, then Abamectin is there. Quite a lot label claim insecticides are available for mites, but depending upon stage of the egg means you have got some other insecticide, then larva you have got some different insecticide, that you should be careful. So these are the symptoms and these are the this ones, and ETL also worked out for various crops. Then I have told you there are many insecticides, level claim insecticide you can use for this one. This is a very serious problem. Next one is mealybug. This is a very crop, especially in case of okra, tomato, brinjal, potato, chili, bean, pointed gourd, cucumber, melons and gourds, and papaya mealybug is a famous story, and how Tamil Nadu Agriculture University, with the assistance of their entire gambit of research institutes, they could multiply and supply Asterophagus papaye, a parasitoid wasp, within no time. They worked in a very integrated approach and a team spirit, and they could control this mealybug within 1 or 2 seasons completely. And they have got very well established labs for supplying this particular parasitoid, which madams from NBR, they were also talking about this particular parasitoids. Now work is going on because the major problem in case of mealybug is the problem is it has got a wax and a floor-like substance around its body. That's why most of the insecticides, but there are some like imidacloprid and thiamethoxam, they have been reported to give some protection, but in general, they are very less effective against this particular insect because of its floor-like and wax-like substances coating in the outer covering. Anyway, we are parasitoid wasp. If you work hand in hand in a team spirit, you can really control this particular menace. Leaf miner is a major problem of late. This is another dipteran insect inundated to release of parasitoid, larval endoparasitoid and parasitoid wasp. We have to work very day night. We have to put a lot of efforts in multiplying these parasitoids, and then we can control this one. And you can use even for tuta, this one you can go for pheromone traps also. These are the various approaches because it has got a pupal stage in the soil, so you can go for neem cake and other things. And friends, always I forgot to tell you about nematode problem. Nematode is a major cause of concern in many of the solanaceous and especially solanaceous crop. And those nematodes also you have to take seriously. Bacillomyces, lot of biocontrol agents include Pseudomonas, Trichodon and Bacillomyces. Many laboratories are working, and you can use those nematicides for managing. And thanks to some private firms, now 2 nematicides have come up, and they are very effective. And label claim work is going on about those nematicides, and you can use those nematicides for nematodes. So always don't, nematodes has to be taken very seriously because they also give predisposition for many of the soil-borne diseases. So coming to leaf miner, as I have told, this is also very serious. So you have to use all these modules appropriately, and 1 or 2 label claim insecticides are also there that you can use. Then fruit fly, because fruit fly I have seen from closely, very closely fruit fly when I work with the fruit fly project in case of this melons. So how dangerous, and it causes almost 50 to 70% loss in case of bitter gourds and musk melon. That is kharbooj, what we say in North India. So those 2 are severely affected, along with the snake gourd in South India. Especially snake gourd is severely affected in South India by this fruit fly. Then we have got watermelon, tinda and pumpkin are also severely affected, and the species Bactrocera dorsalis and Bactrocera cucurbitae. These are ones. It gives a puncture and releases the egg. Then the larva is very it is very active and eats the pulp, and it reduces the marketability and the quality of the produce. So you can see these are the various approaches. This because of the plowing and other things, because pupa is not to go for plowing, neem cake again to take care of your pupa and other things. Then remove infected fruits and destroy. These are the cultural practices. Earthing of soil around vine to expose pupa for desiccation. Wide area management. There are two techniques under wide area management. As the name indicates, you have to go for large area management of this fruit fly. One or two farmers taking the practice will not help. One is male annihilation technique. Another one is bat. That is bat technique is also there. So these two techniques. In male annihilation techniques, what you do is you have got Q-Lear. This is commercially available. Q-Lear methyl eugenol at particular ratio and proportion of insecticide and ethanol. You just mix it and just soak the wooden block for about 6 to 7 hours and just hang it through a trap. Then you will get lots of fruit flies are attracted to this cellular Q-Lear ethanol. This is a male annihilation technique. Then you have got bat technique also. Here you will use jaggery and some insecticides and banana pulp. So you can mix it and just give a using a broomstick. You can just spread this solution in about 200 spots in an acre. They will attract a lot of flies and they will just die. So in this way you can reduce the population. Coming to lepidopteran insects, here it's a big chapter. You have got many fruit borers like Helicoverpa armigera in case of tomato, cabbage, cauliflower, okra, cowpea, pea, bee, whatever the crops you talk, fruit borer is there. That is Helicoverpa armigera. Okra shoot and fruit borer, ERS vitella is very serious. Okra fruit borer, Cosmos heliotis armigera is very serious. Brinjal shoot and fruit borer, wherever this brinjal crop is there, Lucinatus urbanalis is found. Diamond back moth, already I have told you. Cabbage butterfly is a serious problem. I have seen my own eyes how it produces lots of eggs and then the larva comes out. This is the first instar larva. How polyphagous it is, especially on cauliflower and cabbage. It skeletonizes the entire tissues within no time and causes huge damage. Friends, I want to tell that the initial notice of these lepidopterans is the presence of few. Here you can see carefully, if you see, you can see white butterfly. This is nothing but a cabbage butterfly. When you see one or two, this cabbage butterfly just roaming around the cabbage field, that means there is incidence of already the larva of this one. So that is also, you can use it as a monitoring tool for this cabbage butterfly and other lepidopteran insects. Cucumber moth, it's a very, very serious problem in case of bitter gourd and sponge gourd. I have seen my own eyes how serious it can be. Daphne indica. This is the cause of market. This one. Tobacco caterpillar Spodoptera is another giant, huge problem, especially in kharif vegetable crops. It is a very voracious feeder of many of the vegetables. Whichever crop it is there, it is a voracious feeder and lots of larvae, especially in the kharif season, it kills. Head borer is a problem in case of coal crops, and there are many other lepidopterans, but I have symbolically I have given this, and these are the management tools. You can go for locally available tolerant varieties or hybrids. These are some of the listed varieties you can go for depending upon locality and demand. Then these are some of the culture practices. You can go for all these cultural practices, and these are some of the tactics. You can go for marigold seedlings, you just sow here and there. Then you can spray some insecticides because these are the preferred hosts. Erect bird perches that will eat happily the larval and bring down the larval population. Install light trap that will take care of moths, and keep borer damage shoots in the early stage itself so that you can reduce the population. Rake soil for accepting fruit borer pupae, neem seed kernel actors spray, and then some of the baiting techniques I have given in our notes, so I will not go for this one. Then you can go for many poison baits depending upon the species. Bacillus thuringiensis commercially available. Kustiki, this you can spray so that you can avoid chemical insecticides. Then you can go for inundative release of trichogramma about 20,000 per acre. So like that you can go for 2 to 3 times in a season and install pheromone traps. These are the individual very effective components of IPM. Don't rely on chemical insecticides. Go for BT, go for trichogramma, go for pheromone traps like helilure and spodolure. Then you have got all lures for specifically for Lepidoptera insects. Even NPV is also available, and that also you can spray. So spray only label claim insecticide like amamectin benzoate, chlorantraniliprole, novaluron, thiodicarb, so many the lambda cyhalothrin, so many label claim insecticides are available that you can use against Lepidoptera insects. Friends, if you go on talking, it will be endless game. So I stop here. If anyone has got some small clarification, kindly do it. Otherwise, this is I complete and thank all organizers for giving an opportunity.
Thank you, sir. Can I ask something, sir? If you allow.
Yes, sir. Yes, please, please.
Sir, isn't it that carbaryl is banned in 2018?
Ajit?
Carbaryl was banned in 2018, if my knowledge is there.
It is banned now. Yes, carbaryl is going to be banned shortly.
No, sir. Okay. What we were told that it is already banned. So I was just.
Yeah, it is banned. Yeah.
So because you have mentioned it, so I was just curious.
Yeah. So I might have written by mistake. Don't rely on those. You can update also. Not an issue. Okay. Any other question?
Hello.
Oh.
Hello.
बताइए सर बताइए.
Hi sir, I am Sachin Adi from Water.
From
Water, WOTR.
Full institute name?
Watershed Organization Trust, Pune.
Okay.
Sir, my question is in melon crop, the huge symptoms of leaf miner.
Yeah.
If we go for chemical control, there is a problem of pollination in cucurbita crop. Then what to be alternative instead of insecticide to reduce the infestation of leaf miner and as well as to increase the pollination in a cucurbita crop.
So you can go for insecticide which is not against any pollinators. So avoid using the thiamethoxam or imidacloprid against this thing because they will affect the pollinators also. And you have got the lures also because it is a dipteran. You can go for some lures that will take care of the these things. And you can go for some biocontrol agents, parasitoids that also will take care of this leaf miner. Yeah.
Okay, sir. Okay.
Thank you. Any other question? There is no more question. I thank all organizers for giving an opportunity.
I have one question, sir. I am Aruneshwar from Tamil Nadu. Sir, yes, sir, how to calculate the formulation concentration for based on ETL, sir? Based on the field observance of disease threshold level and pest threshold level, how to calculate?
Generally, this is ETL for insects. It has been worked out for most of the crops, like aphid. You have got this yellow mite. In case of chili, it is 1 per this one. Like that, mostly for all lepidopterans, it's about 4.8% infested fruits. Like that, or 1 larva per plant. Like that, it has been standardized in case of insect pest. No, so you go for field this one, and whenever there is a low level, you go for as much as possible, like lures, pheromone trap. You go for this. You go for BT. You go for yellow sticky trap, or you go for blue sticky trap. Such kind of gadgets you can go for in the initial stages, or neem seed kernel extract and neem oil. Now, when the problem has reached the ETL, then you can go for your label claim insecticides. For example, all the label claim insecticides already it has been given, whether it is A formulation or formulation based dose, all the insecticides they have calculated, isn't it? So ETL for diseases, it is not there at all, but it is almost 1. The scale of 1 is considered to be a warning bell for go for prophylactic spray. For example, scale number 1, 1 to 5 scale is there for diseases. For most of the foliar diseases, 1 to 5. 1 means about 5 to 10% of the area affected of the plant. At that time, you can go for prophylactic spray. And if the incidence of severity increases, then you go for the higher the systemic fungicides, but having label claim in fungicides. Am I right? Any clear? Any other doubt?
Yes, sir. Yes, sir. Clear, sir. Thanks.
Okay.
Sir, and we are working on watermelon crops, sir.
Yeah.
The major problem is that wilt, sir. So almost all crops are affected by wilt. We are not able to control.
As a wilt, especially the fusarium wilt is a major cause of concern in case
Yes.
of melons, especially for your watermelon, yeah, this musk melon, and that's a major problem, isn't it?
It's a very major concern here.
Major problem. What you can do is avoid planting in the same field next year. One of the approaches. Second approach is always go for seed treatment, always go for soil treatment of trichoderma, and make it healthy and free from pathogens. You require it 2 to 3 years. First year you will not get the good result. Always full of trichoderma in your field, that especially the sick plots or endemic areas, you start today itself. Complete, irrespective of the crop, you always go for pre-planting of trichoderma application in those sickly and endemic areas with the pseudomonas, with the trichoderma. Those will take care of your fusarium pathogens. And go for deep summer plowing and avoid as much as possible flood irrigation. Always go for
Drip irrigation, micro.
drip irrigation. See, drip irrigation major advantage is 2 to 3 pronged. One is you are saving the water, and especially for soil-borne diseases across the table, you are not allowing the pathogen to go from one point to another plant. That is the beauty of drip irrigation. And the government is also supporting through subsidy to go for drip irrigation. And the technology has come to install drip irrigation in any crop. Even in cereal crops, they have started now, the drip irrigation. So are you clear now? So avoid the sickly or endemic areas again next year for the same crop, especially watermelon or your muskmelon. And in the meantime, you flood with the trichoderma or pseudomonas fluorescens, entire soil. You should have patience. You have to keep out of the soil for that crop at least for 2 to 3 years. And always use either neem cake or FM for enriching trichoderma. Then after 2 to 3 years, you can go for those crops. Till that time, you
I think there is some connectivity issue. I hope your question is answered, sir.
Yes, ma'am. It was answered.
If there, Sri sir is also disconnected, I think some internet issue is there. If anybody has any other question, then they can write those questions in the chat box. We will get back to the Bhatt and give you the answers. And for now, we'll close for lunch. And at 2:15, we'll join back for the next session, which is an ICT in relation to integrated pest management. By 2:15, we'll join. So kindly join by 2:15, and now we'll break for lunch. Thank you.
Sir has talked about his notes, ma'am. So shall we get that notes?
Sorry?
Sir have talked about his notes. He have mentioned in detail.
For all the lectures, we have collected the lecture notes, and they will be, they will be compiled in a form of a book that we released on the inaugural day also, and that book will be sent along with the certificates. So everybody will get all the resource material. There is no issue related to that. And If
Can
at all you have any problem or you want further clarification, then also you can get back to us and we will try to clarify it from our resource person.
okay, thank you, ma'am.
By 2:15 we'll join back, sir.
We'll be listening to Dr. A. Prabhuraj. Sir will be dealing with the role of ESAP, an ICT application in smart agriculture. To introduce Dr. Prabhuraj sir, I think most of you people already know about Prabhuraj sir. Prabhuraj sir is presently working as a professor, Department of, and also is a head of Department of Entomology, College of Agriculture, US, Raichuru. So his interest is in the field of insect ecology with special reference to migration and reproductive biology. And he has very much worked on noctuid moths, which is very much important in agriculture pest management. So he has developed ICT tools to enhance the extension service in crop health management. Himself and his team has been involved in developing a novel ICT application called ESAP, that is Electronic Solutions Against Agricultural Pests, which empowers the extension personnel to diagnose and quantify the problem related to insects, disease, weeds, and nutritional disorders in crops and provide solutions on real-time basis. So, to his honor, he has received various prestigious awards from international and national organizations. To quote few, he is a recipient of World Summit on Information Society prestigious award, that is World Summit on Information Society Championship Award in the year 2018 for designing and developing ESAP solutions and ICT application for crop health management. The award is instituted by International Telecom Union in association with United Nations Organization and conferred in WSIS 2018 conference held in Geneva on 20th March 2018. It is a very prestigious honor, prestigious award to himself and to our whole plant protection fraternity. He is a recipient of ICAR Best Teacher Award, and as I said, he is an excellent teacher, and most of us, including myself, we have referred Dr. Prabhuraj sir's lecture notes, though we have not listened directly as students to him, but then we have referred in many occasions his notes and his guidance to clear agriculture research service examination, and he is of that honor. And to quote a research accomplished, he has designed and developed ESAP. He has developed web-based GIS application which pools analyze and survey reports sent through the handheld device and provide decision support system on real time. He has also developed an eco-friendly economical IPM strategies, especially for L-Corpa, Armichara, and chickpea by integrating L-Corpa indica with other biopesticides. Successful field control is achieved in brinjal root weevil management, grape flea beetle, and chickpea pod borer management. So, without wasting much time, we are very much eager to listen to you, sir. As we all know, the ESAP, which has become a very massive success case study in the field of application of ICT in integrated pest management or plant protection as per se. Thank you very much, sir, for accepting our invitation and sparing your valuable time with us. Now over to you, sir. Please put your slide into presentation mode, sir.
I think I have shared it. Is it visible?
Yes, sir. Shared, it is visible, but please put into presentation mode, sir.
Yeah. So now?
Yeah, perfect. Please go
Yeah.
ahead.
Thank you, Dr. Raghavendra. At the outset, I am very much thankful to the ICAR-NRIIPM and Manage Hyderabad and Dr. Mukesh Saigalji for giving me an opportunity to share my experience in designing, developing, and deploying an ICT application called ESAP, which at this context very well fits into the smart agriculture. So before starting this, so if you just look at the background of the development or the evolution of agriculture, so we can see that from the zooming cultivation or shifting cultivation to an one settlement cultivation with a very old traditional agriculture to the changing into the green revolution agriculture with a high input, the varieties and the other the agricultural inputs. And from there onwards, because of the very high cost of intensive inputs and the harm to the environment, so we slowly again shifted back to IPM. And with the IPM, so we started moving towards the sustainable agriculture. And now we are at this junction where we are now embarking on the high-tech agriculture and the smart agriculture. So as the human civilization is moving towards the smart the living, then those smart applications which are developed for the ease of the human life, and how best that actually can be adopted in the field of agriculture. So several the works, the research, and the technologies have been developed, and in this context, I would like to share one such ICT application which US Raichur has developed called ESAP, which is an electronic solutions against agricultural pest, and how best that it fits into the smart agriculture. So with this background, so I will just move through my next slide. So giving a little bit background about this, as at this junction, this ESAP application is now adopted by the government of Karnataka, and it is made available to all the farming communities of the Karnataka under two main projects operated under the State Agriculture Department and State Horticulture Department. And it is not US Raichur alone which is taking it forward, though it actually was in the forefront in developing, designing, and doing the pilot scale studies. But at this moment, all the agriculture universities of Karnataka state, including the ICR IAHR Institute, is involved in driving this ESAP application right into the farmer's field in terms of providing the on-field solution to the biotic stress experienced by the various crops, so in a real-time basis. So what exactly is this ESAP? So as I told you, as an electronic solution is against the agriculture pest. Here the pest means not only the insect, so it also includes the microbial pathogens and to some extent that the nutritional disorders and as well as the weeds. Okay, how exactly it works and what it does? It basically, this is designed to link all the stakeholders in agriculture on a single platform on a real-time basis in the field of crop health management. So we know that the pests and diseases are so dynamic. Okay, so they need to be addressed and case by case, individual by individual. Some farmer who grows a paddy might be experiencing a different biotic stress compared to another farmer who is just growing a paddy besides his field only. So they are so the dynamic, just like the human health, where every individual have their own the problem. Each field experiences a different problem, and addressing it on a real time is a very challenging. And we as a team actually has made some efforts in the last 10 years, and we have evolved in fitting into the present smart agriculture, where the department officers, extension personnel, and the scientists, and as well as the policy holders or makers can all be made use of this platform in assessing the situation and also making the decision. So basically, it helps or empowers the extension worker to independently assess the crop health, not with the help or support of an expert always. So majority of cases it happens by independent assessment. And not only assessing the problem, but also estimating the extent of the problem, that is through the surveillance mode, and providing a solution there in the farmer's field itself based on the intensity of the problem as he has actually assessed through his application. And of course, we don't leave it there only. And after some days, so we'll take a follow-up. We'll take the feedback of the farmer to assess the strength and weakness of the technology also. So these are all the activities that a ESAP does once in the field. And also on the other hand, it also gives all the information which is being collected at the time of field visit by an extension person into a ready-made reports. And those reports can be viewed by administrators, researchers, policymakers, or any other stakeholders to assess the situation and take an appropriate decision. Let us see that how it is going to work. So crop health, as I told you again, is an insect, disease, nutrition, and the weeds. These are the 4 important components which have been integrated in the ESAP application. This is a very simple workflow of ESAP, how exactly it works. So as you can see it at the bottom in the field, there's a farmer and there's a field user. Field user is the field extension personnel who actually has the ESAP application in his or her the mobile or a tab, whatever it might be, and wherein when this field extension person visits the farmer, he registers, which is of course one time, and then he diagnoses the problem and then quantifies the problem looking at the intensity and then gives the recommendation. Then after some days, without visiting the field again, he takes the feedback. So everything happens in the field itself and it's completely offline. But for the data which he has collected, so when once he is in online or whenever he is in onsite, online will go to the server and this data will generate into the reports, and these reports in a multimodal reports, maybe in a graphical or the map, GIS map or the table. So whatever the form that we want, we can take it. And these reports will be viewed by the researchers, departments, then the policy holders or the makers for the decision making. In certain cases, if the field user fails to identify the problem, maybe in an extreme situation, which actually based on our experience happens about 5 to 10%, then there is an option for the field user to transfer that undiagnosed scenario to an expert. And this expert will have another app called as an expert application in their mobile, and who immediately after looking at it will be able to resolve the situation, which once again through the server comes back to the field user and then gets transferred to the farmer's mobile in terms of the advisories. So this is a one part of it. The second part of it is the knowledge. The knowledge. So the ESAP is strengthened with the knowledge about 52 crops, about 20 field crops and 32 horticulture crops, over 1,000 problems related to insects, diseases, nutritional disorders. All these are being validated and certified by the team of experts which are drawn from the various agriculture and horticulture universities of the state, including IHR. So they actually decide that what is a major pest occurring in the state, at what stages of the crop it comes, at what parts of the crop it comes, then how you are supposed to identify it based on the symptoms, and then so how you are supposed to survey it or quantify it based on the very scientific survey protocols, and according to the intensity, how an advisory should come and get into the farmer's mobile or maybe in a printed form. All this actually is first decided, and after the certification gets into the server, into the ESAP application, which will be made available to the field user. The third dimension of the ESAP application is, of course, as it is a smart agriculture. So this is always like to be supported by the IT group, and we have a very strong IT support with us for the last 10 years, who actually so modify, then changes, and then upgrade it. All these activities will be going. So the some special features are it is a real time and it is an offline process. Even if there is no connectivity, so this process that is at the on field of the registration, diagnosis, quantification, and recommendation happens offline. So now let us look at the what exactly are the components that are involved in it. So basically, I can say that ESAP application involves two important applications into it. And one is an mobile-based application, as you can see it here. It's an Android application for the extension worker, which is also works offline. And there is another called as an expert application, which is also an Android application. The second component is a web application, so which actually being used by the scientist administrators in viewing the reports which are generated automatically once the data is been transferred from the Android application, that is a field application, to the server. So this helps in the viewing the reports, assessing the situation, and the taking the appropriate decision. Now, let us try to look at the identification process. How exactly it happens? The architecture which is being involved or built in this ESAP application is a very unique architecture, which is exclusively an image and voice assisted architecture. Okay. So what happens here is that as soon as the application is opened and there is an option for the farmer registration, and the farmer is going to register it, and once it is registered, then the images of the crops will open up. As I told you, it is completely an image and voice assisted architecture. So the farmer needs, I mean, the field user needs to simply keep selecting the images, and say, for example, a sugar cane, if you select it, then the next the window which opens is the option for selecting the stage of the crop, whether it is like 3 to 6 months or in mid stage or in, so whatever that stage which is appropriate to that. Okay. So there you can see there is an option here to confirm it. So once you confirm at a particular stage, then it opens up into the part which is being affected, because the field user is in the farmer's field in the in the middle of the field crop, and he keeps selecting it as that situation is existing. So whether the problem the notice on the leaf or on the stem or the whole plant, so that needs to be selected. So once the particular stage or part is selected, then the symptoms which are related to that particular part of that particular crop stage will open up. There are multiple images which have been put into this database, and the field user only has to do is to compare the problem or the symptom that he's seeing in the field with the images that are being actually built in the ESAP application. It is a manual comparison and selection. And at the bottom, you can see there is a blue triangle, which is a voice assisted architecture. Even if there is any problem in looking at the image and understanding it, they can take the help of the voice, which clearly tells about what exact symptom that a field officer or a field user has to look at. There are multiple images into it, so they can make a lot of comparisons with it. And whichever the system are the symptoms which are actually being going to be matched, then so they are going to confirm it. Then there is a further confirmation is being done here. I have taken one example and then showing it to you. So say for example, in case of insect, there might be a presence of insects to be confirmed, or if the problem is at the ground level or below the ground, then there might be an instruction to pull out the plant and then look at the root system or so on and so forth. For all this, there is a different levels of identification happens, not in a single level, like you show an image and then try to identify it. In some cases, it will be very easy, but majority of cases, it is very difficult. So after the confirmations further, then ultimately the identity of the pest will happen. So with one example of a root grab, I am just showing this the sequence of the identification process. Here the big advantage is that the extension worker can independently identify the problem in the field by following a simple sequence of steps. So with a little bit of field knowledge, with an experience which almost all the department officers or the officials, extension personnel will be having, so it is very, very easy and everything happens on field itself. Now, once the identification happens, the most important is not the immediately jumping to the advisory, it is to go for the assessing the extent of the problem. So here the estimation, the ESAP has got an inbuilt estimation, the architecture. So before going to the extra assessment, so the ESAP asks the field user to take few photographs, which has two important advantages. One is to build our own database with the field images. And the second one is that we can verify it whether the identification made by the field user is right or wrong. So that also is a monitoring it is going to help. So after that, then when we click it, then it takes the field user to the extension on the survey protocol. Survey protocols appropriately, as I told you, is a scientific based survey protocols have been designed and approved, and they have to simply follow through these the survey protocols. So and as instructed, so they should move around the field and then they have to simply select the drop down menus, whatever which is most appropriate. So there is no typings and there is no readings, so there is no writing, nothing. Okay. So once they actually moves around and then enters an appropriate number into it, then it will be that the job is done. Okay. It might be a point sampling, block sampling, row sampling, or in some cases, it might be even a visual observation also. Say in case of a nutritional disorder, so there might not many of the cases who don't have the very specific protocols. The visual observation will also is going to hold good. So both the things are actually what is called as available. Now the field data which are compared with these fold, which the figures which the field user has entered, so they will actually get analyzed at the back end and they are compared with the threshold values in case of the pest and the disease scoring values in case of the diseases or visual observations within terms of percentage in case of the nutrition. Whatever that it actually happens immediately. And after that, the advisory will pop up. And this advisory is not a general advisory. It is exclusively based on the survey which has been conducted at that time. So with whatever the data which actually is entered. So here the advisory pops up immediately. So showing the intensity of the problem and also the what is called as the advisory related to that intensity. So I have given here the two important things. Say for example, for the same pest in one field, if the intensity is low, then the management that the farmer gets is very different when compared to maybe another field where the intensity is very high, the management will be very different. It is exactly like how a doctor prescribed to a patient looking at the intensity of the problem that a patient is actually facing. So that is being actually involved here. And the biggest advantage in doing it so is that we can actually push the IPM components very effectively into the farmers' the fields by doing so. Otherwise, simply depending on the pesticide application for every identification is not going to hold good. The second thing is that so it also saves a lot of money from the farmer point of view in terms of unnecessarily dumping the the inputs or the pesticides, and also saves the environment. There is a multiple the advantages that we have by following this. Another very special or unique feature that we have added into the solution which has been our advisory which is generated can be pushed to the farmer's mobile through SMS. So it's available in right now in Kannada and English language, whichever the language we select. So that will be pushed, so that will be saved in the farmer's mobile, and the farmer can actually so make use of it. Another important thing that we have done is it also we have connected this advisory to the printer. So there is a small thermal printers are available. In fact, Bluetooth connected. So when you connect it and then give a printing option to it, then it prints out the prescription. So which includes or which will have all the details about the farmer, the farm, then the crop, and then the age of the crop. Okay, and then the intensity of the problem which is there, and the recommendation according to that. Okay, so this is a big advantage to the farmer, as they can have this prescription and take it to any pesticide shop and then do it. Okay. Right. So this is an advantage with this the thing. Then another important thing is that the follow-up. So again, so we are not leaving it alone. Like after the providing a prescription to the farmer, then after few days in this ESAP application, we give a minimum of 7 days. Then after that, a pop-up message will come to the field user in ESAP application to connect to the farmer and then take a feedback from the farmer about the result of the advisory which has been given. So that will actually runs through in very definite the questionnaire, and so looking at it, so various the responses given by the farmer will be selected there and submitted. So by this we are going to complete one complete circle by connecting to the farmer to taking in complete feedback about the service that has been given to the farmer. The simple sets of these questions related to adoption and satisfaction will appear in a very in a specific sequences. So this is the functionality of the application for the field user. In fact, now let us come to the another application which I told you is an expert application. In case that field user fails to diagnose it, which might happen in the two situations. One is that if the field user is new to this application, then there might be a certain confusion that he might faces. In that cases, so it is always advisable to connect to an expert in the beginning for a few cases. The second situation is like if there is altogether a new problem, maybe a new pest or a new disease, or maybe something like the new symptoms which has been expressed or which is not in our the database. In such cases, so very conveniently the field user can shift this the challenge to an expert application. So in our experience, I told you that 90 to 95% cases the field user will resolve the problem in the farmer's field itself. Only in 5 to 10% cases, so it actually gets shifted to the expert. How it actually works. So there is always the option for the field user in the ESAP application called as an UDM. UDM means an undiagnosed malady. Okay. So if there is any confusion in identifying, the field user can go and then select this. Once he selects it, there is a series of some questions which come, such as just to assess how much is the severity of that undiagnosed problem. It is a visual observation, whether it is a high, moderate, or low, and then take few photographs, maximum 5 photographs of an affected part, affected plant, and affected field like this. And also there is an option to record the voice that a field user can himself record it for about few seconds, 8 to 10 seconds, 15 seconds or so. So which helps the field, expert in diagnosing it. And of course, it automatically captures the GPS location. Just so take care of these three issues and then submitted. Once it is submitted successfully, and then so now it will get into the expert application like a pop-up, so as an alert, and which because it's a mobile application, the expert wherever he is can actually open it and then look at it. Now, how exactly it is going to work in the expert mobile? So he gets a series of undiagnosed scenarios which is raised and sent by the field user on different crops or the crops for which that expert is specified. And say for example, here I have shown you that banana and cotton, and different colors indicates that based on the visual observation, how they have actually entered it as in low, medium or high. Then once they select any one of these, and then so it opens up the details about the farmer, farm, crop, the images that he has taken, okay, and also the voice that he has recorded and sent. Along with that, everything will actually come here. So for many cases, it's very easy job for the expert to look at the images and also listen to the voice of the field user and assess it. Then they directly can go to the resolve. But there also we have given an option of calling the field user or even the farmer. There is a call option at the bottom. So they can actually, the expert, if he needs a little more information, so either the expert can call to a field user or he can directly call to the farmer, because once you press that call, then both the phone number will be available and they can directly connect to it. Or if there is still some complications related to a nutrition disease or something like that, there is a chat box here where the expert can chat with another expert, because this will have a group of experts, an entomologist, pathologist, so soil scientist, crop physiologist, who are that. And they can actually chat before resolving it. So once they confirm it, then they can go and then click the resolve. And if the problem is already mentioned in the ESAP application, then it will come as a drop-down menu and they can simply select. So in those, any of the problem that they have selected it and then send it back. So once they send it back, then what happens? Because the intensity is already been recorded. So with that problem, what is a management will directly or automatically go to the farmer. Go to the farmer's mobile as an advisory. Suppose the problem is a new one. And if it is a new one, then there is an option to select a new pest, and there an expert can actually type the new pest and also give an ad hoc recommendation. So this option is also there. And once they actually, if it is a new pest, they write it and give an ad hoc recommendation, immediately an alert will come to the mail of an admin telling that a new problem has raised or identified. So this gives an what is called as a quick and a real-time message to the higher authority that what is that new problem. Because the details are available, they can immediately go to the field and then check. All this is possible with the expert, the application. Now coming to the web application. The web application here, as I told you, will actually exclusively for two important purposes. One is that the report viewing. Okay, all these things, the data which is generated, as soon as they come to the server, they develop into a multimodal the reports, like as you can see the pie charts, graphs, the tables or the maps. There is several filters which are available. So the report user can actually view it up to a village level. Okay, it might be up to a village level to an entire country. You can view the individual farmer reports. They can view the crop-wise reports. They can view the pest-wise report. So any way, whatever the way that they can, because on the top you can see these are the filters, and also whatever the mode that they need, they can actually view it and so taken very appropriate the decision. Okay, so the instant reports for quick decision support to administrators and policy. Then another important thing is that, see, because it provides the spatial maps, so this is very easy to take them out and then integrate with the weather parameters. over a period of time, and they can actually come out with the pest distribution and the risk assessment maps. The second important use of this web application is the device management. So through which we actually add a particular device to the ESAP application in the sense. So we actually provide an access to the content of the ESAP application, completely manage the content into it, addition, deletion, okay, change of management practices. So all those things can be done into this. The UDM management can also be done through the web application. That is, apart from the expert mobile app, they can also do it through the web applications. So all this is actually possible with the web application. So then how exactly, like when it started, how it has actually they traveled and all, as I told you in the beginning. So ESAP application actually has a genesis in 2011-12. So at that time, so we designed, we developed and tested within 1 year with that concept of it has to be on the field on a real-time basis. Then for the next 3 years, so we did a pilot study in our US Raichur jurisdiction only in among the 5 the districts there. And within a very short period of time, we could actually able to reach around 100,000 farmers and looked at the technological robustness and also the content, the robustness and everything. With this success story, in fact, there is also a third-party evaluation by IIM Bangalore, so who involved with us in using the ESAP application in 2 important projects headed by them, in fact. And simultaneously, it was adopted by the other state agriculture universities of the state. So from 2014 to 16. And after that, looking into its potential, then government of Karnataka suggested and then took a decision to adopt it for the entire state. And from 2017 onwards, now it has been adopted by the Karnataka State Department, and from 2019 onwards by the Karnataka Horticulture Department. So under both the schemes, now it is being running continuously. And some of the outcome of the data which we actually have generated from the ESAP application, I told you 100,000 farmers and so much of data we did, and actually try to develop a GIS map, so spatial distribution map of say leaf hopper in one of the districts across different years. We did it, looked at how the pest is behaving over the years in different places, and we also integrated that with the weather parameters, as you can see it, and looking at the which weather factors are more influencing, and so that we can come out with the prediction models, and then from the current curative methods to the prophylactic or from alert system we can actually develop. So we are in the process of it. And so how many number of crops? I told you 52, and these are the important 20 the field crops, and these are the number of problems that we are actually incorporated or integrated. And these are the horticulture crops, so 20 plus 13, so 53 crops content is now sitting in the ESAP application and being used by the officers of the agriculture. How many of them are using it? In fact, you can look at the ESAP field deployment. So in state agriculture over 1300, and from the horticulture over 1000, and state farming faculty, so 400 users. NGOs, we are actually giving it to NGOs also. It's completely free services, and also students. So our the final year students who actually go to the farmers field as a RAVE program. So we are giving this ESAP application in the plant health clinics. They go and then actually support the farmers in identifying and giving the advisories. Or now more than 3,500 users are there in fact. And this is one glimpse of the number of the fields which have been visited by the horticulture as well as agriculture. You can see that whole space is actually riddled with this, indicating the extent of coverage that ESAP application is having. Different color points indicates the intensity of the problem. If you press any one of these points, then it gives a whole details about the farmer, their address, the crop, the stage, the problem, the intensity and the advisory, along with the farmer's photograph also. Every detail is going to come into this. The individual case by case we can actually follow it. And of course, I am also very happy to share this also. This is the field visits made by the RAVE students. So in a couple of years back, when the entire country was under lockdown condition, and when all these students were in their homes, at that time we could very successfully complete the RAVE program just because of this application, where they could able to go to the nearby fields of their place where they are staying and then cover up the entire the area. So this is what is a smart agriculture. So without any problem, they could be able to support the farmers so much. So within 3 months, over 300 students have actually given over 9,000 advisories, so spread around the Karnataka state. And so some of the important highlights of the ESAP application is that so we could able to first report the FA incidents during 2018 in May in our northern part of Karnataka through ESAP application. And during 2019, so RAVE students actually identified there was an outbreak of Padka grasshopper on maize in some pockets of the Davangere district, so which they could actually send it as the undiagnosed scenario, and where the experts identified it, and then we also gave the alert to the state. In fact, fortunately, this did not spread. This was also at that moment also, if we can remember, there was a huge locust swarms which are coming to the Indian subcontinent, and everybody were actually like suspecting that whether it is an locust or so. But so this we could actually resolve it. In 2020, in fact, in our Tungabhadra project area, in our US Raichur jurisdiction, so there was a first time report of the paddy gallmitch, so which used to be a coastal pest now, so it suddenly appeared, and through ESAP application only government of Karnataka took the report, and accordingly they actually acted. So these are some of the important highlights that I just wanted to share. So for the last couple of years, so we have actually given around 30,000 advisories covering most of the all the districts and around 2,000, 5,200 villages, and over 13,000 farmers have been benefited with the advisories. This is only last couple of years back, and of which 1,800 women farmers are there. Over 47 crops were actually surveyed, and the expert interventions was around 4,000, like UDMs that was generated and resolved. So I'm also very happy to share with you that, like, by looking at the success of this ESAP application, the government of Karnataka launched a mobile plant health clinic vans, which are called as a Krishi Sanjeevinis, and where the ESAP application is the integral part of this van. Of course, along with the plant health management, there's also a soil testing kit and the PT testing kit and all there. But the main major focus is on identifying the pests and diseases in the farmer's field. So it is provided with a tab and a printer, and so which we have integrated, and there is an exclusive a person in the attached to this van who actually takes the van to a different field visits and then diagnose it and then reports it. So this was last year, 2021, was launched by Honorable Chief Minister and Agriculture Minister. Over 75 vans were released, and in fact, last Sunday, that is on 12th, so another 100 vans were actually been launched at GKVK, US Bangalore. So where I was also. So just to summarize up, what are the unique features of ESAP application? The system ensures that the extension worker will have only those crops which are relevant to that location. So need not have to download all the 53 crops and then keep selecting over that. If that particular place where only few crops are there, they can actually access only those crops. They also have an access to have the location specific management plans. This is also very interesting. So paddy crop grown in North Karnataka and South Karnataka, the farmer will have a different management practice in North Karnataka for the same pest, so over the South Karnataka farm. So it's a very location specific management. available in local language, and there is a local experts are available so that it is very easy and quick for diagnosing the undiagnosed. Estimation of pest intensity is the important thing because it adheres to the principles of IPM and it discourages the generalized recommendation. We don't give any general recommendations. And we have already moved towards the prescription-based farming. Everywhere now we are insisting that an expert has to give like a doctor who gives a prescription to the patient. So we have already adopted several years back along with a Bluetooth printer. So which actually given a very good authentication to the prescription base. And true empowerment to the extension worker. All these years we were actually struggling. In fact, extension workers never used to, they used to very much hesitate to go to the field simply because that majority of cases they were afraid, unable to resolve it. So they used to send it through WhatsApp to the expert, wait for them for days together. Just by looking at the image, they used to get the recommendation. But now, no problem. So they can do it their own and independently, and only in certain cases they can actually take it. And most important is a big data. So you get a huge data about the pest, about the crop situation, about the farmers' details. That this is a very much useful in developing the early warning and the forewarning system, which are already we are into it, and then shortly will be coming out because the data accumulation has to be made continuously. So with this, so I thank you all for your patient listening, and I'm ready to take any questions. Thank you very much.
Sir, can anybody outside Karnataka can access this app?
No, at this moment, as I told you, it is available only in the Karnataka, but outside Karnataka, in fact, the Maharashtra government sometime like back, like that connected, but Andhra and the Telangana are showing interest in adopting the ESAP application. Whichever the state which are showing interest, we are ready to extend it to that state. Not an issue.
So
We
what is
in
the
fact want
modality?
to extend it.
Say if our department is interested in doing that, so what are the modalities?
You are from which state, sir?
Sir, I'm calling from Jammu, sir.
Oh, Jammu, wonderful. Yeah. So I think like 7, 8 years back, we had been to Jammu and as well as Kashmir explaining about ESAP application.
Yeah.
So actually, with the official communication to me, so then we can actually come over there or discuss online and then look at the modalities of adoption.
Okay, sir. Okay. Thank you.
Because the content, everything is already there. So it's little bit of the modification from your end and also to converting into your language. Fine, you can immediately start using it.
Actually, we are already working with one of the international agency. Maybe you may be knowing CAB International. They are sharing the knowledge with us, and even we are creating local content. But your content is very much indigenous, so we thought it's better to contact you people.
Thank you, sir. We are always ready.
Thank you.
Anybody else?
Good afternoon, sir. I'm Aruneshwar from Grower Farms Private Limited. Sir, you have explained a really nice, really interesting app. So my question is, how this ESAP application ensure the location-specific advisory is delivered to the farmers?
Yeah.
Is there any?
Yeah.
Yes.
Sir. Yes, actually, in our the software, we have made a provision like that. Say, I told you, take an example of a paddy crop grown in northern part and grown in the southern part of the same state. Okay. So the advisories which are very much relevant to each of this part, so that particular location can be selected. And as they actually, the identification of that problem and the quantification has come for the advisories, whichever the advisories that they have selected will pop up. So this is all a software, the arrangement that has been made.
Okay. So, being you talked about on the knowledge partners, so for each crops, the package and practices are already been inputted.
So basically, you see, I told you that we have drawn the experts from the different universities like entomologists, pathologists, horticulturists, soil scientists, and crop physiologists. And so basically, we compile not only the content of the package of practice, that is start with, but we have compiled the management practices which have been given by the crop specific institutions of ICAR, then CIB and RC, okay, and the any other international organizations which is relevant to it. All that compilation, we first make it, share it to the expert, and once we get an feedback from them, call for a workshop. They will come over here, debate it, and then they will decide that which has to be done. It is something like that. And sometimes even with the farmer, their own field experience, we have added the advisories into it. It's not simply taking from package of practice and then putting it there.
Yes, it's an inclusive you have developed. Really nice. Thank you.
Pardon me, I didn't able to hear you.
Yes, sir. The package of practices involved even the farmer, so it's really interesting, sir. Inclusively, it was developed. So nice.
Yes, sir.
So, anybody else would like to have any interaction with the speaker, please? Sir, kindly unshare your screen, sir.
Yeah.
Okay.
Shall we get your contact number or mail ID, sir?
Yeah. I will just. Where is the
chat box? Yes.
Yeah. I am just writing my email ID. And the mobile number. Yeah, you can contact me over phone or through this email ID.
Thank you very much, sir. It was a wonderful session, and always it is pleasure to listen to you, sir. And we also look forward for such a wonderful lectures from your end in the coming days, in which we organize from our NCIPM, sir. Thank you.
Thank you, Mukesh ji. Thank you.
Thank you so much, sir. Now we would call upon the next
Dr. Bagraj.
speaker.
Thank you very much.
Next,
Sir,
we would.
thank you. Nice seeing you, sir. Nice to see you.
Thank you, sir.
Dr. Raghunder, continue, please.
Yes. Okay. So next we will move upon. Now we would like to call upon our training coordinator, Dr. Mahentesh sir, Deputy Director Agriculture Extension from Manage, to give a lecture on new initiatives and approaches in agriculture extension. To the large extent, we request Mahentesh sir to give a lecture on the Manage and how it works and how it will be useful for the youth or upcoming generations to take part in agriculture and agriculture extension, sir. Over to you, sir.
Thank you, Dr. Raghavendra. And can you just enable the share content for me?
Yeah, just a minute. Saurav, please enable the Mantri sir to share his screen.
Well, in the meantime,
just wait.
Sir Mukesh, sir, you wanted to tell something, sir?
No. Actually, he's just gone out for a. He'll come in a minute. I'll tell him.
No issues, sir. No issues, sir. I'll engage the participants meantime. Dear participants and all my colleagues from National Center for Integrated Pest Management, very happy to share my thoughts on some of the important extension initiatives and approaches of Manage. And this presentation will be basically focusing on the introduction to Manage and its activities and some of the important programs and schemes implemented by Manage, and also the new initiatives of Manage which are probably of some interest to some of the people who are attending this training program on integrated pest management. I'll try to contextualize and emphasize more wherever I feel that these type of things are relevant for this group of participants. And in case I feel that these may not be relevant, I'll try to skip. But in the end, we will always have some scope for maybe discussion in case some of the things are of interest to you. A passenger has convenience fees. And just introduction to manage. Basically, this institution was established as a response to the challenges of agricultural extension. And till this date, this is very unique of its kind in the entire world with respect to agriculture extension management. The institution was established in 1987, and basically institution has an organization consisting of General Council and an Executive Council. General Council is headed by the Honorable Minister for Agriculture and Farmers Welfare, and the Secretary Agriculture will be the chairman of the Executive Council. And Director General will be the chairman for academic committee, and he acts as overall in charge of taking care of the managed activities. Starting from the inception, Manage has set a vision to be counted among the most pioneering, innovative, user-friendly, and self-supporting agriculture management institutions in the world. And now also Manage takes care of 40% of the expenditure to be met on all its accounts. And we also have the mission of facilitating acquisition of managerial and technical skills by extension officers and all the other stakeholders. Basically, these activities are implemented through 10 different academic centers at Manage. Each center is headed by either a director or a deputy director. And each center is having some of the activities focused on these themes. But besides these activities, each center is also doing taking care of some of the important schemes of the Government of India. For example, I am heading the Center for Agriculture Extension Policy, PPP, in Extension and International Center of Excellence. So from this center, we are taking care of the DESI program, which is a very important Government of India scheme for agri input dealers. Similarly, another center is taking care of the agri clinics and agri business centers. Another center is taking care of the skill training for rural youth scheme, RKVY Raftar scheme, the agri startup and incubation. So on these lines, there are different schemes. And we also have School of Agri Business Management, which is a very unique program and first of its kind in India. And since its inception, it has 100% placements. And this year, the average CTC on the company, for all the, on an average of all the 66 candidates, it is more than 12 lakh rupees. And from the last 25 years, 26th year, including the recent placement, it has achieved 100% placement. So besides these things, Manage takes care of Government of India schemes, consultancy, agri startup, outreach, international training, and many other things. So thanks to COVID, the number of candidates trained has jumped from 4,761 to 26,730 during this year. And the number of training programs, because of the collaborative nature of training programs and the online nature of training programs, including the short duration workshops and training programs, we could organize close to around 431 training programs. And basically, Manage was an Indian response to the Indian agriculture challenges. Because of its competency and because of its willingness and its positioning, Manage could organize several international training programs. And two of the important training programs are the Feed the Future India Triangular Training Program funded by the USAID India and the ITEC training programs supported by the Ministry of External Affairs. So these training programs were important both for the trilateral agreement between the Indian government and the US government and many of the African nations. Manage also conducts certain action research spreading in different states, and these include some of the research areas and the consultancy activities, which are basically involving the assessment and evaluation of certain schemes and projects and all these things. Now coming to the important schemes of the Government of India, the first scheme, maybe many of you are aware, this is an Agri-Clinics and Agri-Business Center scheme implemented in all over the country since 2002 with an objective of giving training and support to unemployed agriculture graduates to take up the entrepreneurship opportunities in the agriculture sector. This is a 45-day residential free training conducted through more than 100 nodal training institutions across the country. There will be a loan provision of up to 20 lakh rupees, and 1 year hand-holding support will be there, and NABARD is mainly supporting the scheme. Initially, this was a 60 days program. Now it has been reduced to 45 days training program. So far, 75,000 unemployed agriculture graduates fresh from their graduation have been trained, and 31,000 have started some form of agri-venture covering 32 different areas. And another important scheme is the Diploma in Agri Extension Service for Input Dealers. This is the scheme I'm implementing as a principal coordinator for 22 different samitis in 21 different states. And the objective of this training program is basically to train the agriculture input dealers who do not have a formal education in agriculture discipline or any horticulture discipline, to make them competent to guide and to advise farmers on agri extension services, and also to make them as a para extension worker so that they can join hands with the State Department of Agriculture or any of the private institutions to give the extension service support to the farmers. So this scheme is basically implemented in two different streams. One is the self-finance scheme, where the candidates who do not have a valid license to sell the fertilizers, pesticides, and agri-chemicals are enrolled by paying the full fee of rupees 20,000 as of now. And if the candidates are having a license to sell any of these agri inputs, Government of India will give 50% subsidy. And this is a 1-year diploma program wherein classes will be conducted once in a week physically. And these classes are basically conducted for the entire day's duration, before lunch and after lunch, coupled with some practical exposure and hands-on training program. The 40 theory classes are also, 80 theory classes conducted on 40 days, plus 8 field exposure visits, industrial visits we call. And after this, there will be a final examination. But before that, there are periodical assessments, just following the pattern of what we do usually in BS Agriculture or in Diploma in Agriculture. So that kind of approach has been brought deliberately into the program. So far, more than 60,000 input dealers have enrolled, and around 40,000 input dealers have already been certified from 21 states. Skill training for rural youth is 1 week short duration training program conducted by different samitis and nodal training institutions across the country. And this is a new program of the Government of India, newest among all the three. And around 20,000 rural youth have been trained in the country so far. Then Manage is a knowledge partner for RKVY, and Manage promotes the agri startups, and 200 agri startups have been incubated, and 700 innovators enrolled as members with the Manage Incubation Center. And every year, Manage takes up the evaluation of these schemes in giving the grants under the Agribusiness Incubation Program up to rupees 25 lakh rupees. And besides this, there are other agri startup ideas from Aqua One Center and many other schemes of other disciplines and departments also. This I already made a mention, postgraduate diploma in management. And this is basically a student-driven program wherein the candidates will have a very good opportunity to get the placement. And this is rated as the third best B school in agri-business sector, only after IIM Ahmedabad and IIM Lucknow. Another important program for in-service candidates is the PGDAEM program, and this is just created as an opportunity for the people who are not able to pursue higher education in agriculture extension discipline, who are working in the agriculture extension field, maybe in the state governments or in the central governments. There are different MOOC scores. The major ones are the PGDM in agriculture extension management and in agri warehousing management. And these programs are running continuously on the MOOC course every year. This is one interesting program, the Certified Farm Advisor and the Certified Livestock Advisor program, wherein the experts in the respective disciplines will be given a refresher training program where they can become certified advisors in different major crops, commodities. For example, I am an entomologist, and maybe I will have my expertise in controlling the pests in grapes. So in that case, concentrating on one crop, the competency of such resource persons will be built through the collaboration of different ICR institutes or state government institutions. Now I'll shift to some of the new initiatives of Manage. This is one is the Seva Manage, where the service extension through voluntary association. Basically, this platform is created for retired agri professionals with an idea that they still have the energy and motivation to contribute for the agriculture sector or the development of agriculture sector in the country. And all the retired professionals can join hands with Manage in implementing the scheme, in doing some activities of Manage, for example, conducting the examinations of PGDAEM, DESI, ACABC, and other things. Because these schemes of the Government of India demands frequent evaluation, frequent assessment, then selection criteria, all these things. So the Seva Manage members can be used, or they will be given a token honorarium, and their cost will be met from the Manage grants. And this is the Seva Manage. And then a national network of agri journalists has been promoted, because the idea is there are exclusive journalist experts. Cinema journalists are there, sports journalists are there. But why can't we have journalists looking after the agriculture sector, wherein the burning problems of agriculture are brought to the limelight through the agri journalism? So for that, we conduct regular webinar series and training programs for agri journalists, where the issues will be brought to the notice of these journalists, and subsequently they can either publish based on their secondary research and working further on those things and compiling data on those things. Another initiative is the Manage Krishi Gyandeep Knowledge Lecture Series, wherein renowned experts from the agriculture discipline will be called to Manage, and their talk will be recorded on their experience of handling the agriculture extension, which can be of immense help to the field extension functionaries who otherwise may not have the opportunity to hear them again and again. So these talks are converted into a small documentary or a movie, and are posted on a Manage YouTube channel, where you can see some of the important, so far around 14 Krishi Gyandeep knowledge lectures have been recorded and put on the Manage YouTube channel. So the illustrious speakers include Shri Goyal sir, Dr. Shobhana Patnaik, Dr. S. Ayappan, Dr. Ashok Dalwai, Dr. Mohan Kanda, many other illustrious people who have rendered very unique service in the agriculture extension field are invited, and these talks are recorded. And one new intervention which is created as a complementary idea to the promotion of 10,000 FPOs by the Government of India, that is the Manage FPO Academy, wherein the training and capacity building and the consultancy support is extended to the newly formed farmer producer organizations or the CEOs of these farmer producer organizations. So there are different courses also, and the important recent course that we have launched is a 3-month course for CEOs and board members of FPOs in order to strengthen their understanding about the FPO, the rationale and the idea behind FPO, and how they can be made successful, or rather profitable, rather sustainable in the present scenario. This is another interesting program which probably many people will be interested. This is the Manage National Facilitators. Basically, Manage conducts so many programs, so many activities, schemes, all these things. And it's inconceivable to implement all these schemes with thin faculty number of 18. But for this, we have started networking with some facilitators drawn from different ICR institutes, state agriculture universities, Department of Agriculture, Sametis, KVKs, and all these things. Basically, Manage will identify and train these committed and competent officers from these offices or the institutions. And we sensitize them with respect to the managed activities, and they save the resources and the time of managed faculty by attending to some of the important events and activities in their nearest locality possible. For example, to conduct an event in Uttar Pradesh, it is illogical and irrational to send one person for 1 hour event from Manage. So in order to do such things, this facilitators network has been created. So far, 100 national facilitators have been trained, and we often use them for conducting examinations, interviews in different ACABC, training them. And they also act as resource persons, as master trainers for some of the important schemes of the government of India and of Manage. Manage CSR Forum, I think now we know that there is a stipulation to spend at least 2% of the average net profit in the corporate social responsibility among the companies. So we have started this activity, and there is a different center and the cell looking after that. One more new initiative is the Jai Jawan Kisan, Soldiers for Agriculture. Here the idea is to use the physical and the financial powers of the retired soldiers who are willing to take up agriculture in some form or the other. So maybe, for example, protected cultivation, they want to start up in their location. And what kind of trainings can be started? So since this is in the very inception, we are collaborating with the Directorate of Settlement, Ministry of Defense in implementing this scheme. And this is in different phases. So Manage has launched a few new awards to encourage the startups, to encourage the best research in agricultural extension. These are the Samunnati Award for Best Agri Startups and Best Book and Best PhD and MSc Thesis in Agricultural Extension. And the Agri Film Festival is a new idea wherein this year on 11th March, Honorable Minister for Agriculture, Government of Karnataka, who is Agriculture Minister also and a cinema actor also previously. So he was invited to inaugurate this Agri Film Festival, wherein 280 plus films on agriculture and agri innovations were screened, and the best films were given with the Best Agri Film Award. And the Agri Startup Idea Bank is basically provides a platform to share innovative ideas of agri startups in the country. And this is in brief. I tried to make it contextualize to the needs of the participants. But in case you feel that I can share some more information on any of these things, please do let me know so that I can share some of the information if required. Raghavendra, please, please unmute. You are on mute mode.
Yes, sir. Sir, thank you. Thank you very much, sir, for summarizing Manage as a whole. And now I request the participants to please have an interaction with the speaker.
Anyone have any doubt?
Can I ask?
Yeah, please, carry on.
सर मेरा एक क्वेश्चन ये है कि अभी आपने एक जो जैसे ऑर्गेनिक सर्टिफाइड ट्रेनर या वैसे करके आपने एक चीज बताई, उसका ऑलरेडी मेरे को 2-3 बार मेल भी आ चुका है आपके मैनेज के थ्रू। मैंने उसमें अपनी मेल आईडी सब्सक्राइब करके जो डाली। सर मैं ये पूछना चाहता हूँ, क्या उसमें ग्रेजुएट होना जरूरी है या ग्रेजुएशन के बिना भी कोई कर सकता है अगर इंटरेस्टेड है, या उसमें कुछ है तो?
देखिए, सर्टिफाइड फार्म एडवाइजर या सर्टिफाइड लाइफ स्टॉक एडवाइजर इज बेसिकली फॉर द प्रोफेशनल्स। ऑलरेडी जो लोग उस सब्जेक्ट में अपना एक कॉम्पिटेंसी डेवलप किया है, उन लोगों के लिए किया हुआ कोर्स है।
ओके।
और उसके ऊपर जो मतलब जो ऑलरेडी उनका कॉम्पिटेंसी, उसके ऊपर जो भी चाहिए स्पेशलाइजेशन के लिए ये सर्टिफाइड फार्म एडवाइजर या लाइफ स्टॉक एडवाइजर किया गया है। आप अपना जो भी प्रश्न है आप लिख लीजिए, लेकिन अभी तक ऐसे कोई सुविधा नहीं है उसमें, बट देखते हैं आगे अगर।
सर क्योंकि एक्चुअली क्वेश्चन ये है कि जिसके पास ऑर्गेनिक का प्रैक्टिकल नॉलेज है और जिसके पास जो किसान एक थोड़ा हाई फाई हो चुका है या थोड़ा सा जिसके पास प्रॉपर नॉलेज है, एक वो कर रहा है, या किसी फार्मर का बच्चा है लेकिन वो एग्रीकल्चर से नहीं है, किसी और से उसने ग्रेजुएशन कर लिया लेकिन उसको एक्सपीरियंस हो गया है, वो इंटरेस्टेड है, लेकिन फिर उसकी वही कहीं ना कहीं कारण पड़ रहा है कि वो एक बीएससी एग्रीकल्चर नहीं है। इसीलिए मैं पूछा, तो ये प्रॉब्लम मैं भी फेस कर रहा हूँ।
नहीं, मैं आपसे सहमत हूँ, मैं समझ गया आप जो बोलना चाह रहे थे। लेकिन देखिए, कोई भी स्कीम की जब शुरुआत होती है उसका कुछ रूपरेखा होती है, उसका कुछ ऑब्जेक्टिव्स होती है, और उसके लिए बेसिकली जो भी क्राइटेरिया तय किया जाता है उसके लिए चलता रहता है, लेकिन टाइम के हिसाब से बदल जाता है। ऐसा नहीं है कि जैसा है वैसा ही चलता रहेगा। ये यूजुअली प्रोफेशनल्स के लिए है अभी तक। जैसे मान लीजिए कि एनसीआईपीएम में कोई साइंटिस्ट है और वो साइंटिस्ट अगर कोई पर्टिकुलर क्रॉप में ही स्पेशलाइज करना चाहते हैं, तो ऐसे लोगों के लिए एक डिपार्टमेंट में है, डिपार्टमेंट ऑफ एग्रीकल्चर, ये यूजुअली जो प्रोफेशनल्स ऑलरेडी है उनके लिए अभी है ये।
मीन्स नए के लिए नहीं है कि कोई नया इंटरेस्टेड, उसके लिए नहीं। जो आप मतलब आपका कहने का मतलब जो ऑलरेडी बीएससी एग्रीकल्चर कर रहे हैं, जो खुद परेशान हो के भाई अब करे तो क्या करे, या जिस करना चाहता है, तो उनको एक हम रास्ता दिखा सकते हैं कि आप एक फार्म मैनेजर का करके आप उसमें अपना प्रोफेशनली लेके आगे काम कर सकते हो।
नहीं, सुनिए, आप नाम को लेके मत बैठिए, क्योंकि इसके लिए बहुत विकल्प है। एज एन एग्रीप्रेन्योर, एज एन एग्री एंटरप्रेन्योर, आप एसी एबीसी जैसे स्कीम है, या कोई और अग्री स्टार्टअप है, या कोई आरकेवीवाई रफ्तार के स्कीम के अंडर आप अप्लाई कर सकते हैं। जरूरी नहीं है कि सर्टिफाइड फार्म एडवाइजर कोर्स में ही करना है। जहां आपके लिए एडमिशन नहीं है उसके पीछे आप दिमाग क्यों खराब करना चाहते हैं? जहाँ प्रोविजन है आपके पास, जहाँ इतना सारे विकल्प है आपके पास, कोई भी प्रोग्राम में आप एनरोल करके जो आप हासिल करना चाहते हैं, आपका ऑब्जेक्टिव क्या है, और अगर एक रास्ता बंद है तो दूसरा रास्ता कैसे ढूंढ के निकालना है वो सोचिए।
ठीक है सर, जस्ट मैं क्वेश्चन पूछा है, जो डाउट
सही है आपका प्रश्न से मेरे कोई आपत्ति नहीं है। इट वाज वेरी मच राइट।
Anybody else?
Hello.
Yes, sir. Yes, please.
Sir, who is eligible for the farm advisor, certified farm advisor? मीन्स उसके लिए क्राइटेरिया क्या है, और फॉर्म कैसे भरना, फिलअप करना है मैनेज का?
फॉर्म नहीं, देखिए ये प्रोग्राम यूजुअली हमारे पार्टनर इंस्टीट्यूशन से अनाउंस किया जाता है। सर्टिफाइड फार्म एडवाइजर कोर्स, फॉर एग्जांपल अभी जो कोर्स चल रहा है इंडियन इंस्टीट्यूट ऑफ मिलेट्स रिसर्च के साथ एक कोर्स चल रहा है, तो वो लोग कैंडिडेट्स को स्क्रीन करते हैं। मैनेज बेसिकली ये सर्टिफाइड फार्म एडवाइजर कोर्स पार्टनर इंस्टीट्यूट, क्योंकि जहां अभी इंटीग्रेटेड पेस्ट मैनेजमेंट में आप सर्टिफाइड फार्म एडवाइजर बनना चाहते हैं, आपका ट्रेनिंग मैनेज में तो नहीं हो सकता है ना, आपका ट्रेनिंग एनसीआईपीएम से ही किया जा सकता है। अगर एनसीआईपीएम हमें प्रपोजल भेजता है कि हम सर्टिफाइड फार्म एडवाइजर कोर्स कंडक्ट करना चाहते हैं, ये 15 डेज शॉर्ट ड्यूरेशन कोर्स है, ये 15 डेज के लिए कैंडिडेट्स को मोबिलाइज करेंगे, और उसके लिए जो भी है स्टाइपेंड वगैरह है, और उनके लिए जो प्रोविजन है, अंडर दोज प्रोविजंस दिस प्रोग्राम्स विल बी कंडक्टेड बाय द स्पेशलाइज्ड इंस्टीट्यूट्स जिस इंस्टीट्यूशन में वो स्पेशलाइजेशन है उसी इंस्टीट्यूशन में ये ट्रेनिंग किया जाता है। और आप पहले ये डिसाइड करिए कि आप कौन सा इंस्टीट्यूशन से ट्रेन होना चाहते हैं और किस सब्जेक्ट में ट्रेन होना चाहते हैं।
ओके, मुझे ये और कहना है कि अगर वो जो कैंडिडेट है वो ऑलरेडी एंटोमोलॉजिस्ट है या फिर उसको सब नॉलेज है फील्ड की प्रैक्टिकल, तो उसके लिए ये जरूरी है कि वो अप्लाई कर सकते हैं क्या किसी भी इंस्टीट्यूट में?
नहीं, वही बताया मैं आपको कि जब भी आप सर्टिफाइड फार्म एडवाइजर कोर्स का नाम लेके बैठ गए तो ये फिर दिक्कत होता है, क्योंकि उनमें से जो एलिजिबल क्राइटेरिया होता है वो डिसाइड ऑलरेडी किया हुआ होता है। ऑलरेडी पर्सन्स प्रोफेशनल्स वर्किंग इन द फील्ड, उनको एक और दर्जा देने के लिए, यानी हायर कॉम्पिटेंसी एक्वायर किया, एक स्पेशलाइज्ड ट्रेनिंग लिया करके बताने के लिए वो ट्रेनिंग का सुविधा किया गया है। वही मैं बोल रहा था, वो 15 दिन का शॉर्ट ड्यूरेशन ट्रेनिंग प्रोग्राम है, सर्टिफाइड लाइफ स्टॉक एडवाइजर या सर्टिफाइड फार्म एडवाइजर। काइंड ऑफ देखिए, एमबीबीएस के बाद हम जो वो हायर डिग्री के लिए जाते हैं वो एमएससी में जाते हैं। ऐसे अगर कोई प्रोफेशनल विथ ए शॉर्ट सर्टिफिकेट कोर्स जैसे कोर्स करना चाहते हैं तो उस कारण के लिए वो कोर्स किया गया है।
ओके सर, ओके, थैंक यू वेरी मच।
हेलो, गुड आफ्टरनून सर।
जी गुड आफ्टरनून
सर, मैं ये पूछ रहा था, अगर
सर
एग्री बिजनेस मैनेजमेंट में डिप्लोमा करने के लिए क्या प्रोसेस है और कैसे अप्लाई कर सकते हैं?
सर
हाँ, मैं इस प्रश्न का पहले जवाब दे देता हूँ। देखिए, एग्रीकल्चर डिप्लोमा यानी आप see there is a misnomer in what we are offering, like PG diploma in agri business management is actually not a diploma program, it is a PG program. So it has been from the commencement it has been floated as PG diploma in agri extension management. The diploma word is there, but it is the proper PG program. It is a 2-year residential program, and उसके लिए BSc एग्री या अभी कुछ और डिग्री भी चलता है, कुछ कैंडिडेट्स के लिए वो सुविधा रखा गया है, लेकिन कैट का स्कोर होना जरूरी है। इसका मल्टी लेयर में सिलेक्शन स्क्रीनिंग किया जाता है, कैट्स का स्कोर, आपका ग्रेजुएशन का स्कोर, और यहाँ जब आप आएंगे पर्सनालिटी असेसमेंट यानी इंटरव्यू या ग्रुप डिस्कशन, वो ऐसे जो भी असाइनमेंट्स होते हैं, इन तीनों का क्राइटेरिया के बेस पे सिलेक्शन लिस्ट प्रिपेयर किया जाता है और 66 बेस्ट कैंडिडेट्स विल बी सिलेक्टेड फॉर द पीजी प्रोग्राम।
ओके, थैंक यू सर।
डॉक्टर अभिजीत सर, आप कुछ पूछ रहे थे, प्लीज अनम्यूट।
एक्चुअली सर हाँ, actually sir, I am asking about is any KVK scientist willing to join this farm advisory certificate course? Can they join?
नहीं, मैं वही बताया आपको, ज्वाइन कर सकते हैं, बट what is your area of specialization that you are looking for and what is the institute that you are looking for? These things are important, but you may write, in case you are interested in particular subject and particular institute, you please write to us. We will see whether a batch can be mobilized. Individual interest से कई बार दिक्कत हो जाती है, अगर एक बैच मोबिलाइज हो पाएगा तो ये ट्रेनिंग ऑर्गेनाइज किया जा सकता है।
अच्छा सर, ये ट्रेनिंग क्या डायरेक्टली मैनेज ऑर्गेनाइज करता है या कोई भी इंस्टीट्यूशन, आईसीआर इंस्टीट्यूशन ऑर्गेनाइज कर सकता है?
कोई भी आईसीआर इंस्टीट्यूशन इस ट्रेनिंग को ऑर्गेनाइज कर सकते हैं।
तो ये अगर कोई केवीके,
देखिए ये मैनेजमेंट
केवीके भी ये कोर्स इम्पार्ट कर सकता है सर?
नहीं, केवीके में आप कैसे कोई स्पेशलाइजेशन लेंगे? यू नीड सफिशिएंट रिसोर्स पर्सन्स ना उस सब्जेक्ट को डील करने के लिए। ये बहुत ही स्पेशलाइज्ड है। मान लीजिए कि ओनली मैंगो कल्टीवेशन में एक सर्टिफाइड फार्म एडवाइजर कोर्स बन सकता है, ओनली ग्रेप में बन सकता है, ओनली आईपीएम में बन सकता है, ओनली सॉइल हेल्थ मैनेजमेंट में एक बन सकता है, प्रोसेसिंग ऑफ मिलेट्स में एक सर्टिफाइड फार्म एडवाइजर कोर्स बन सकता है। तो इस तरह का एक स्पेशलाइजेशन है, स्पेशलाइजेशन कोर्स, इसीलिए मैं बोल रहा हूँ शायद I could not communicate it very appropriately, but what I am trying to say is these are very specialized course organized by specialized institutes. KVK is basically, देखिए, it is a multidisciplinary team of multidisciplinary scientists, and 15 days talking on very specialized subject by one or two faculty may not do justice to the entire course. So maybe with this idea, I don't know whether any KVK has proposed to organize, come forward to organize these training programs. But usually these training programs are conducted so far by ICR institutes, specialized ICR institutes.
Sir, presently all the KVKs are conducting DESI course and certificate course for pesticide science, pesticide course also from conducted by the NIPHM. So why not this course could be implemented by KVKs? We have some faculties and faculties with us, and it is not a big deal to conduct a 15 days course like on this particular specialization as you have about. So it is not just a big deal to conduct such training program. We have so many resource person in our KVK as well as in our sister organization also, sir. Sir, is there any boundary to restrict this KVK for regarding conducting this course?
Dr. Abhijit, I think you did not fully receive what I was trying to communicate. I did not say that it is a big deal to conduct these training programs. What I'm trying to tell is these are very specialized subjects, and 15 days intensive program, making them competent with a hands-on training. All these things are imparted. Coming to your argument that KVKs are implementing DESI, AC, ABC, and all these things. DESI and AC, ABC are basically conducted over a curriculum. There is a fixed curriculum. There is always a chance to invite the specialized resource persons from the nearby agri institutions, and you can always conduct. And if you really feel that you are also competent and it is convenient for you to conduct certified farm advisor course, you can apply. You can very well apply and justify your case. There is no reason that we will simply deny it for the sake of denying it, or simply for the sake of saying that KVKs can't organize these training programs. But if you sufficiently justify with your resourcefulness and your subject matter expertise and all these things, certainly you can organize, but the onus of mobilizing the candidates to take up these training programs also rests with the KVKs along with the manage.
I am not arguing, sir. I am just clearing my question.
Fine, I fully understand.
There's some difficulty regarding conversation. Please don't mind, sir.
No.
Thank you.
No, I'm trying to just put it very, very objectively so that there should not be any confusion. You can very well apply, and
Yes,
you can take the certified farm advisor course.
Thank you very much.
Sir, myself Kartik Devnath from Assam. मेरा एक क्वेश्चन था, इग्नो में रिसेंटली पीजी इन एग्री बिजनेस और डिप्लोमा इन हॉर्टिकल्चर कोर्स चालू किया है. वो कोर्स कितना यूजफुल है हमारे लिए?
Sir, no, I'll safely avoid to answer this question. It all depends on how these courses are subjected, and also courses are taken up by the students. But let us not go into those things, because I can speak on behalf of Manage as what Manage does.
लेकिन नया आदमी के लिए, अगर कोई नया आदमी जो कुछ भी नहीं जानता एग्री के बारे में, वो अगर डिप्लोमा इन हॉर्टिकल्चर कोर्स एडमिशन ले ले, उसके लिए कितना कॉन्फिडेंट होगा वो उस कोर्स के लिए, कोर्स करने के बाद? यही मेरा क्वेश्चन था एक्चुअली.
नहीं, सही है, लेकिन देखिए मैं भी वही बोल रहा था कि कोई भी कोर्स BSc एग्री तो सब करते हैं, बट फाइनली जो 100 कैंडिडेट्स निकल के आते हैं सभी एक जैसे नहीं होते। कोर्स तो एक ही है ना सर, कोर्स तो एक ही है, फैकल्टी भी वही है, लेकिन स्टूडेंट्स की जो नॉलेज लेवल अलग अलग होती है। Now there were 380 candidates. 90 are presently, visible uh, on the online training program. So it all depends on how individually we take responsibility for what we do and how we take the things forward. So that is why I did not want to comment on the usefulness of the course. Courses are basically designed to make them useful and relevant as much as possible. But it is the consumer finally decides as how you want to use it. That's why I was trying to just leave it, but
Avoid.
Okay, okay, sir. Thank you, thank you. Okay.
Okay. So if there is no more queries, I would like to thank our honorable speaker, Dr. Mantes sir, who have given insight upon the manage and the programs activities which is being taken up by their end towards the nation building. And thank you very much, sir, for sparing your precious time. And it was a wonderful lecture. We too, even in fact, we too got a very good insight into the manage activities. And certainly it also will help many of us who are present here to come ahead to take some courses or some capacity building activities under the leadership of manage. Thank you, sir.
thank you, Dr. Thank you so
Thank you very much, sir. So, over to ma'am, Rekha ma'am.
Thank you, Dr. Raghavan. After this session, we will have a session on IPM and serials by Dr. Mukesh Sehgal, sir. Dr. Sehgal?
Yeah, yeah. Yeah, ma'am. Thank you. Yeah. हाँ मैडम बताइए.
Sir, we have just finished the session from Mahantesh, sir. We will continue with the IPM and serials, sir. Just the rice part will continue today.
Okay. Okay, what I'll do today we'll discuss the rice only, right? The time is also very short.
Yes, sir.
And then tomorrow we'll discuss about the wheat and maize, right? After sugarcane and oilseed crops in the last lecture we'll give. So anyhow, let's start with today's one. So can we start?
Yes, sir.
Yes, sir. Sure.
Saurav, Saurav, who come now presenter?
Dear presenter, sir.
Okay. Can you see my slide?
Yes, sir.
Okay, fine. Good afternoon, friends. Let's talk about today's third session. Because of the manage guidelines, the coordinator has to give 3 lectures. So I'm proposing the third lecture, and this is very, very important and close to my heart also. So I welcome you all in this session, a part session, I can say, the integrated pest management of important cereal crops of India. Friends, when we talk about now, let's talk about your system. When I say your system, means the agriculture system. I consider plant is also a kind of a system we have. So then, in that case, we say what is normal and what is diseased. That is very, very important to know to us, because when we define disease, it means morphological or physiological disorder of the man or plant. If we talk about plant system, then we talk about plant is known as a deceased. And if there is no morphological or physiological changes and there is no loss, there will be a normal plant. So most of the plant health problem is caused by biotic factors such as virus, nematodes, insects, diseases, weeds, rodents, and many other you have heard about post-harvest also. And majority of the health problems are also due to abiotic factor, just like you have a deficiency diseases. You can have the irrigation, you can have sun scare, like that we have many other abiotic diseases also there. So the important point to handle or to tackle this is the identification, and identification is really very critical. So when I say identification, you need of the plant as well as of the, now sometimes we find certain
एवढं बंपर हिट लागलं तुमच्या
symptoms
डोसाचं
just like
RRC
nematode
मध्ये अरे
symptoms,
कोण डेपो मॅडम आहेत का
or
म्हणे तिथे
certain time we are
बापरे
even
त्यांना
your wilt,
नाही त्यांना इकडे घ्या म्हणे
you will
अकोल्याला
find out these
ब्रीडर सीड
symptoms
प्रोडक्शन मध्ये
resembles the abiotic
अच्छा
factor
बाय द वे तू फोटो कुठला होता
differences also. So
तो
therefore
त्या
we are talking
प्रेझेंटेशनचा
about today is about the cereals. In India, we talk about cereals. We have wheat, barley, rice, maize, oats, and millets. No doubt we have a millet. The millet year is going on, but we don't have much of experience in millets. So therefore, we don't talk about millets. We in our rice team in NCIPM, we are working mainly for rice. We go for a cropping system, wheat also, and certain time we have started with the maize, and still other crop we have not touched because of our manpower constraints. So if you look at the food grain production in million tons, if you talk about, as I told you, that is also here 295 is increasing, decreasing, fluctuating, but majority of time is keep on increasing. We have shown data for last 15 years. This is by KBK data is there, and 2021 22 data also, last time I showed you 103 million tons is the data. So we can say we wanted to match with our you can say population of 1.36 billion, or in coming days we may be 1.40 billion in 2030. So we need 350 million tons. So we are not much of scared, rather we are comfortably increasing almost in the level we have. So now the food grain output, if you look at the targets, majority of time India is well sufficient of the food grain production. Our estimate, no doubt, I told you 2021 we have 293 million tons, but we have produced 303 million tons. So we have always produced, except 1 year, we are, except in 1819, we are able to produce much more the food production in our country. So therefore, if you look at this is the data I wanted to show you little here. If you look at the area, production, and yield per hectare, you can find out is almost increasing. And rice, wheat. So always you find out your targeted will meet down, and we have the sufficient amount of the, you can say about the land is also holding. But we are very surprised that we are producing much high, but there is a big loss is taking because we don't have much of proper storage facilities. So therefore, we have a storage loss of the pest. So surplus stocks exceed storage capacity. We don't have much of capacity available to store our food grains. Likewise, use of improper storage grain method. We don't use the proper storage method also. We don't use polythene sheet. We don't use the tin. We don't have much of idea where to store this all. So we can say constraint in the liquidation of excess stock. There's one bigger constraint we have, lack of interest by the private sector. Private sector don't want to store much of things. They want to earn, except few vegetables they wanted to store. Lot of food you are producing, lot of your target you are meeting, but still our many million population strives, or the children are looking for food. There's still the situation. If I look at here, if I tell you situation, if you go to Chhattisgarh, Andhra, Madhya Pradesh, Arunachal Pradesh, here you can say even Karnataka, we find the people are, or in almost all state, even what to talk about Bihar, what to talk about even sometime in Punjab also. The Because of the policy, we don't have much of good policy available, and we are lacking of the distributing we don't have much of this proper distribution system, or we could not streamline till after even after 70 years of our independence. So there is a need to have a strong policy. This is a slide I already shown to you to tell you that lot of grain food export we make. And today we are going to talk about rice, rice only today, and then tomorrow we talk about wheat and maize crop. We will discuss what are the different problems we are facing and what efforts we are made or what efforts can be made. If you look at the wheat loss, you can see here pest loss, or pest means the insect loss, is 8%, disease is 13%, weed is 8%, and remaining is other loss. This is a big question. We are looking for the other losses, rodents. We have a loss of the, you can say, storage. Likewise, we can find major part is not because maybe due to all farmer, because of the policy. What I want to stress upon, the policy is such that we have a loss. So yield losses caused by pest and disease and weeds you look at here. So you can find out, this is the data we are showing to you, that rice, wheat, barley, maize, potato, soybean, cotton. So we find out there will be economic loss. And in dollars, if you go ahead, $1,000,000, $4,420 million is a big amount to sustain our, you can say, economic power. So this is the one you can say demand. If our demand is 107 million tons in 2019-20, according to the Ministry of Agriculture, but we are producing 108 million tons. So we are producing much high. In wheat, also high. In total seed is also high, but pulse is no doubt is less. But we can say our, if we have a good policy, none of our either our demands are pseudo demands, or our demands are such that we cannot meet them demands. So but we are producing more, so therefore there will be a chance of, you can say, Niti Ayog, according to Niti Ayog, we have a different policy by which we do not give it the much of things to the our generation. So let's talk about rice today first. Rice is now, if you look at the ecology in the rice is grown in irrigated, upland, deep water, hilly, flooded, and nowadays I have started with we have also started with the direct seeded rice. New initiative, we are we have started at ICAR-NRIIPM in the, and we are done from, no doubt it's not a new means, last year we have started with 2013 onwards, and then we're making a good achievement in this. So friends, the integrated pest management, I will come one by one to you. First of all, what is integrated pest management? That you have heard many times, but again to repeat you, to make you remind that IPM is a decision support system for the selection and use of pest control tactics, either singly or combined into a management strategy, not a control strategy. We want every individual to stay back in the environment, but may not cause much of loss. So that is their level. Their level should be such. Their level should be that they are not going to be harmful to us. Now, our IPM must be based on our cost benefit analysis. There must be, you must have the cost benefit analysis must be there to take into the account the interest and inputs on the producer and society and the environment. Meaning here, it should be socially and economically viable. That's very, very, and environmentally safe. Our thing is to maximize the use of biological control. In the morning, you heard from three ladies. They are the pioneer. They are doing good work, and you have heard about how the role of parasite and predators play an important role in the management of this. So first of all, try to see. Sometimes we use the word integrated control. Don't use the word control. Always try to use integrated pest management. Now, when we are modulating, when we are basically what IPM module is that we look for the our our thing is based on the pest population. So let's say our IPM synthesis and validation should be on for major pest. We cannot go for every minor pest because you look for the cost benefit ratio. So your cost benefit, keeping your cost benefit ratio alive, you need to see that you are talking about key pest. And what is the IPM strategy? We must know to manage the key pest. And now, once you synthesize the module, you must have the form validation. What we are trying earlier, we are looking towards the sick plot at the KBK or at the state government department. Seed, you can say, we did in chickpea, we did in Suratgarh, where a lot of seed production is there. And we try to inculcate various IPM strategies and validate it themselves. Then we must develop success stories, and that success story must be properly, you can say, go ahead. So if you look at friends about the insect pest, look at there, this is very, very important to know what is the status, because you are looking for key pests, and key pests, those are economically viable. So let's say our thing is leaf folder, moderate to serious, green leaf hopper, it is minor to moderate, brown plant hopper, moderate to serious, white blood, WBPH, minor to moderate. And now, what I will going to tell you in some time that we have a carryover pest also. There are certain pests are carryover. Just like if I give you one example is root knot nematode, Melodogenic graminicola will be is multiplying in the rice field, but if you keep your stubbles over there, and the you can say in the wheat crop, even maybe next day you can find they will become active, and they may cause a loss, or they may destroy the whole crop. So therefore you need to know what are the key pests and what are their status we have. Now, stem borer. Now if you look at the stem borer, you find out stem borer, they are the key groups of insect pests. This is one of the key pests or major pests we have in our basmati rice, or in our rice, or in cereals. Just like I was giving nematode example, we have also legume pod borer is also that feeds on the spike of the, you can say, of the inflorescence of, or spike of the, you can say, ear head, and may cause the loss. So now 5 species of stem borer, yellow stem borer is available, but sorry, 5 is known to us, but yellow stem borer is the most widely spread and dominant and destructive. Now, you can find out if you look at the affected plants, if you go to the field, you find dead hearts or ear hurts. White ear hurts you can hurt, but you can have a dead heart. Infestation results in the yield of approximately 3 to 90. This much yield will be brought about by here. Now, the affected plant show dead hearts or white hair, and infestation result in the yield of 3 to 96% loss. So their dead hearts as well as the white hair heads will be there. Plant hopper, you can find brown plant hopper is one of the notorious pests. Is also known as BPH, which may not be managed easily by the chemicals. By the use of chemicals, it cannot be managed, but other way we have to apply to manage this pest. Now, why WBPH is another one. They survive best in the stagnant water. Likewise, we have major disease of feed, These are the major diseases of the field: blast, bacterial leaf blight. You can have then sheath blight, brown spot, fell spot, urbata. Then they are serious or moderate serious or minor serious or sporadically available. Now, rice blast are the typical symptom on the leaves: leaf blast, neck blast, and panicle blast we have. Now, sheath blight, you can find sheath blight. The disease occur in the field at the hard, tough to mature of the rice. Try to see that I am sharing my experience, keeping in a view of the whole country. I'm not talking about one of the pests is common in one area, and these are the majority of them. They are normally present every time. Next, if you look at the secondary disease, we can have brown spot, pulse smut, bacterial leaf blight, and if you look at the weeds, we can find grass weeds, broadleaf weeds. You can have sedges like that we have. So these are our weed problem we have. They are also considered pest. IPM strategy. The IPM strategies start from prevention, monitoring, and intervention. That's very, very important to us. We have to prevent our crop, we have to monitor properly our crop, and if we feel that some intervention is needed, we try to make the effort so that they will be losing. They will be you can say getting good ground for stating that there will be a chance of intervention. Now, what do you do in the intervention? Summer dry deep plowing. What do you do? Paleva, right? So what do you do here? Deep summer plowing. You do 5-6 times. The whole field will be plowed very seriously, and then it come down to here. It it come down to here. You can find out that there will be a deep summer plowing reduces the pest population. Now, the soil-borne pest can be easily killed. If you add small amount of water here, that is alternate summer, alternate wetting and drying, you will find you feel because the active nematodes will become more predominant during night time, but as soon as you provide some layer, they will be get attracted and they will get killed. Now, cleaning of crop, refusing, elimination of weed host, crop rotation are the one of the basic. So certain time we record that there will be need of crop rotation, but we don't tell farmer what to do. If I tell you here, we are very much worried about the diamond back moth in the Jhansi region, Bhangra block. But what we replace it, we have replaced it broccoli, cucumber. We find there is no much pest is available at the time for these two crops. They become very, very good. And next we'll come to here green manuring. Yesterday I'm talking about you will have here green manuring, dhecha or moong. And then this is burning of the green manuring after 40 to 45 days. Moong residues after picking and mature puddling will be also there. So keep in mind, green manuring is very, very important in moong. And we have also tried here in the in green menu, in spite of green menu, we say fertilizer. We have tried fair the, you can say, for making effort to print our stories. So therefore we need to this. So sowing of green manuring, dhencha. Now this dose is given to you after picking, after residue. We will find out the port and puddling of the field. Selection of crop variety. I told you yesterday, we must select the crop variety which will be very important. The crop variety which is resistant or tolerant, at least tolerant, will be there. And then we have a seed treatment. We always recommend in IPM strategy, go for good seed treatment. And yesterday I told you, in the rice I am applying the fact. What I am applying, I am taking you can say, the wheat seeds, and then we dip into the salt water, and as soon as certain things will float, we collect and throw it out, because that will be going to play a very insignificant role in our, you can say, pest management strategy. So therefore, what we do here, let's say dhencha or moong, you do it, and depending on then you select the variety which is highly resistant, and the fertilizer micronutrient spacing is very, very important. Learn side by side. So now stage wise, here stage wise you have the before and during nursery sowing. Our aim in IPM is raising healthy nursery. Raising healthy nursery is very, very important, is on target to us. Just move. दो सिर्फ ये वाली यहाँ से Yeah, actually slide is moving, therefore we have just stopped it here. आवाज आ रही है? Okay, so now before nursery, you can have seed treatment. Take the gunny bag. On the gunny bag, spread the trichoderma, 4 gram per kg of seed, and then again place your seeds over there, desired quantity of seed. Put another time of trichoderma. Put the, you can say, gunny bag, slightly wet it and keep it for germination. And before raising the nursery, so what you do, make nursery bed. I try to make it a 10 meter into 12.5 into 1.5 for nursery treatment. Means you can say 10 meter into 1.25 or 1.5 meter for nursery. You go with a sowing gap we have here of 30 centimeter between the two beds. I'm talking about beds. We use the farmyard manure. Sometimes we use urea, single phosphate, then muriate of potash, 20 kg. But here we have 80, 80, 40 gram. But we should avoid. We can have here, you can say 30, 30, 20. Like that we can have, or in last 40 we can have 25 and 15. Like that we can select it. Then comes to during nursery, we find out in Indo-Gangetic plain, you find Bakane, the disease is very, very common. But here we find out during the nursery, you find what you have to do. You take the certified seed. That is very, very important. I told you that soak the seed in salt solution 200 gram in 100 liter for 15 minutes. That's very, very important. And then the seed treatment, I told you carbazendin, but we can apply here with the help of your trichoderma also. Now, during transplanting, I told you transplanting should be done at when the leaf stage is 5. Otherwise, I told you there will be 25% loss of the your yield is there. So based on fertilizer and NPK should be like this, and you have the yellow stem borer, you have the deep you have the clipping of the leaves, and you have 2 or 3 seedlings per hill, and spreading into the, you can say, spacing row to row is 20 and hill to hill is 10 centimeters should be recommended. Now habitat management. This is very, very good to understand. In earlier time, we are thinking that spiders are our enemies. But later on, we could able to find out spiders are our friends. So therefore, what we do here, we do the habitat management. We take a straw bundle, we take a polythene, just like I had shown to you this figure, polythene or the nut, small nut, and then in this we place your, you can say, your seed. You can you can take the spiders, we can take the, you can take the certain your straw bundles, and these straw bundles must be charged with the in the sorghum, charged in sorghum field for 15 days, contain spider adult 20 to 30, egg masses for 8 to 10, and egg masses like this. So therefore we can say that by making the straw bundle available, the farmers can easily manage the, you can say, the plan. Their, now you need to have monitoring. Yesterday I was you have to have visual observation. This is recommended that please visit your field as frequent as possible. That's very by my point of view. Plants also talk. They will also talk and they will tell I'm ill. So therefore you can do this. Pheromone trap, 5 traps per hectare for monitoring. And then we have a light trap. We are using NCIPM or Chinsura light trap. Especially we are using NCIPM light trap. Light trap we are using here. We are using the we are using here the, and then we are using the sweeping nets. Nowadays we are using yellow sticky traps, blue sticky traps also, but here in the rice we don't use this. The yellow stem borer, this is the pheromone we have able to standardized, and now we have the availability of yellow sticky trap, and trap height should be 0.5 meter to 1 meter, 5 traps per hectare for monitoring, and 20 traps per hectare for mass trapping. But we avoid to have a mass trapping. Then dispenser replaced at the third week. That is very important. Now ETL, because your decision is based on economic threshold level, because you are taking care of the you can say economics. Therefore you have to have nursery, or you have to have nursery. In nursery, yellow stem borer will be 1 to 2 egg masses per square meter. Root knot nematode, one juvenile, not nematode, one juvenile per gram of soil is very dangerous. And then you talk about here, what to talk about, the early bacterial leaf blight. 2 to 3 plants per square meter will be infected by this. Early to late tailoring, we can have this. We can have. So these are the ETLs. This is standardized in the whole country. It was standardized, and now we are accepting this in total. So that we say here we can have a early to late tailoring. What do you do? Panicle, initiation of booting, flowering at milky stage, all, or blast, blight, all we can do this one here. So during here, now after transplanting, apply this vitamicin 3% at the rate of 200 gram per hectare, or hexagenol 5%. Okay, so these are the whatever shown to you red. These red show, please don't use it. Though I written for your knowledge, but try to avoid these chemicals because they are not recommended by the other country. So if there are other countries having dangers, India too can have. So therefore we need to have certain technology and certain to make them sure that they are healthy. So we can do this one. Blast. Likewise, the same I told you, the blast is also used. These are chemicals commonly used in the blast. Now, release of parasite. So we have in the morning, you have a lady, Rachna Vashne. That lady has supplied. That lady help us in getting the certain your key pest, which may be enemy to the others. So we request her to again to continue the same and to do. Case form is like the same. Pre-flowering till maturity is like the same. You can have. Now on-farm IPM procedure and methodology for select the area and see what are the resources available. What are the pest problems we have. Then the selection of farmer is very important. Selection of baseline information is also too important. Synthesis of IPM module, there is another one, and provision and the provision of critical outputs that we have to make them sure that people may get it. Collection of data and pest and crop natural enemy and crop yield. Success story what we developed. Now in toto, you can find out we have seen the planting of dhelcha for green manuring, seed treatment, planting, judicious use of fertilizer, and then we can have the other important fertilizers are use installation of pheromone trap for yellow sticky trap. You can have the gum sticky trap also there. But anyhow, this is one spot sticky trap. So we then find it could be done. Now if I show you data, you just variably see the data. Here you can see the data from 2018. We can have 2019 IPM and farmer practice. I just try to correlate to you and tell you that how cost will be making effort. So therefore we can think at our this 18 10,018 810 is given, loss in 2008 which is recovered in the coming time. Now IPM module we are using the Pusa Basmati rice. So what we are doing? Planting thatcher, seed treatment with carbamazepine, seed treatment with the root dip and pseudomonas, planting of 2 to 3 seedlings. So these are what I wrote in the left side. They are our targets. They are actually required. What for we are here is the same. But likewise, we can do have the same for the farmer practice. That is also to be done. Now, if I give you example of Uttar Pradesh, in Sitapur, you can find different pest and different pest position situation is shown to you like this. So now, so here we can find release of parasitoids at the rate of 1 to 1.5 lakh hectare. So drain of water, spray imidacloprid and stay ethofox, and then you can use K-Swami. And so these are our pre and the pre flowering we will do as given here. Now we can find out the total population. This is the way what yesterday Dr. Agunda is also talking to you how to prepare the performer to get the information. By this you can get the performer. Now IPM and success story I already discussed with you. Now in June 2013 to 16 we have a project that is the impact assessment of pesticide on the environment using EIQ tool in the rice cropping system, and we use it and we find out there are certain people, those are here with us, they are also got training from that area. Now what is the objective? Our objective to have a safer pesticide for rice crop using EIQ concept to assess the impact of pesticide on soil and rice crop health. And goal in journal, a mathematical model will be developed on the lab and it may be tested in the field itself. Now, if you see the basic line information is done for 62 people, so in area covered by these people is 67 hectare, and number of villages it is covered is 14. So now you can see the land holding farmers are very small land holding farmers are very large, resources are poor, literacy rate is around 31%. Whereas go to the other side, it become more than 60%. Awareness nil, almost side variety nil. So now you can find out here because of the 1A and 1B WHO class, there will be pesticide. So we are making effort. We are making continuous effort to make our program successful, and therefore we have dreamed ourselves that we will be producing more pesticide food, free food. So therefore we are making effort to making so many farmer field school we conduct. This is the old photograph no doubt we have when Dr. A.K. Singh DD extinction was the chief guest here, and he was the jet PD at the time in Kanpur. So these were the project was formulated. So pesticide dealer training was also given to tell them what for pesticide dealer will do, so there is no loss in the business also. So now impact analysis you look at here. This is the impact analysis you can find out. You can find out the benefit cost ratio is much high in IPM in comparison to non-IPM or in control strategies. So this is the one we need to know, must know. Silent feature of the program I already discussed with you. Knowledge increased here. The person's house, he got the house like that. So many things were in his credited account. So now health benefits. Our health benefits are that. So we are using healthy nursery techniques. Losses minimized up to 30%. Technique is very good and effective. Seed and seed bedded treatment. So now you can find out. You can easily check that the health benefit, environmental benefit, social benefit are making effort to have got. And then I told you yesterday that KVK Sitapur we have framed or guided. We have guided to prepare a FPO. His name is Ozone. Ozone FPO they form, producer they are doing, and through technical support of upskilling farmer community, marketing, safe tomato foods and other vegetable through in IPM Mart we are setting. Now next is this Dr. Anup, who is one of the participant with you, is now making effort of this. So to spread the IPM in Basmati rice in the Bambawad village as well as nearby villages. So therefore he tried to, just like you see here, targeted and improved it. Likewise you can see. So pesticide consumption, application, we can see that here IPM and non-IPM we can what we can do. So this is the NCIPM project done by the Dr. Tanwar, and the cluster of 18 villages he has selected for Gautam Budh Nagar. GPS location is this. Three area like this, farmer is like this we have. So variety grown in like this we can. Now you can see the our achievement 2018 we are shown to 14 to 18. Now we have gone much ahead of this, and in 2020 to 23 we have planned to train more than 10,000 farmers or more in rice crop. So now is horizontal spread. How we make the horizontal spread is shown to you, and it is very simple to tell you that how horizontal spread have took place. And if the if the farmers are benefited with the critical input, they will do much better. But this is again to show you, to impress upon you that IPM play a vital role in reducing the pest population and as well as pesticide consumption. So like that you can say number of sprays is also reduced much high. It's 2.33 average in the farmer practice, whereas it's zero point 0.2 only. The results are almost similar for the other dates, therefore it's not much of shown to you. So cost benefit ratios also keep on increasing. Next is like with the quality of pesticide residue, what Dr. Tanwar recorded here, he recorded all these pesticides he selected and find out the one. I started the I just to tell you that you have to move into remotest area. This is one of the remotest area of our country, East Siang, Arunachal Pradesh, where we have selected 50 farmers, and these farmers were given an intervention, variety, seedling treatment, pseudomonas, nursery treatment, clipping of the seedling, installation of, you can say, lure, alternate wetting and drying, and all these we can do based on our ISA based on IPM. We can do this one. Likewise, we could find out the result. If I see that this is the IPM module is given to you. Now yield is given to you. On an average, you find out the gross net income is 4,35,000, whereas the gross return is 5,37,60 rupees, where net income is 1,000 days, and this will be the total value. So now this is the one to show you. Shepjala also do the same, and Arunachal Pradesh we have. So now we have done in Arunachal Pradesh, we have done in the Karnataka, we have done in direct seeded rice, and all these we have shown it. So I stop here, and tomorrow we'll start with the wheat and maize. If you have some doubt or question, please ask, and I try to find reply here. Please. So I hope now point is clear to you. I stress upon again that we need to see that it should be ETL based. You have to have an economic impact factor is there. What you should do, just like see here. That's very, very important on farmer validation procedure, selection of the area and definition of pest problem, selection of farmer, collection of the baseline information, synthesis of IPM module, farmer field school, provision of critical input, collection of data and pest play a very vital role. And how we can do this is also to shown to you. You can apply these intervention, timely intervention, and then answer. Just like you can put here what is the target pest and what will be our nursery, what intervention we can take. Here we talk about. Likewise, we can have during transplant what intervention we can take, like after transplant what we take, and after your key formation, like that we can have. So I stop here, and if you have some doubt, please ask. Any question?
Good evening, sir. I have a question, sir.
Yeah.
Sir, coming to the ETLs, are we going to follow the same ETLs because since the pest scenario is changing, should we?
Yeah.
Is it the same
You are
the
right.
ETL?
You are very correct. Why I tell you here recently, I really feel very happy to listen from you also, because recently in the you have FCCI, means your foods, you have a sustainability like AI, the one who will tell us how much pesticide will do it. With the rice exporter, we have the meeting. I also in the same view, because I'm doing the rice IPM in Haryana, Uttar Pradesh, Karnataka, and Arunachal Pradesh, Assam, and Tripura, like that. You can have Nagaland. So there's so many states I
Yes.
am working on. Meghalaya. But you are right. My question is same. We are using, no doubt. I just forget to tell you, we are using pest defender ratio, right? So, but this is I given you theoretically point is you are right. But I feel it. There are ETL in different ecologies are different. So therefore, I also recommended to ICR as well as to the other one. You can say to NIHPM, DPPQ, that, and to rice exporter team also, that we must have location specific ETLs, or at least
Yes, sir.
agroclimatic zone ETLs. That I agree. I agree with your point.
Definitely, sir. Definitely, because you are in a institute where you have a lot of experience and you're
Yeah.
moving to different states. Because when you say it, it will have an effect rather than us saying it.
No. That definitely no. It's not me only. Everybody agrees
Yes.
on, because we have scientific data also. I'm not showing you data here. So many data I have availability with me. I really tell you. If I tell you leaf folder in the if I talk about leaf folder, let's say go to here Uttar Pradesh, right? In Bamba, what we don't do anything. We don't do even 5 leaf folder per square meter. We don't do anything, right? But if the same I recorded, if the same you do in Arunachal Pradesh, there will be a loss. There will be loss. 5 to 10% loss will be there. And because they here, the reason is that leaf folder is not much multiplying here faster in the northern plain, but there they multi, because the humid condition are there, they multiply very fast. So, and this one example I given to you. Likewise, we can have, if I talk about Karnataka, Mandya, you are surprised, 2 large juvenile per gram of soil is sufficient for nematode to cause a loss. Whereas if you took the same in Meghalaya, there not be much loss. So because of the different type of soil is there. You are right. We are working on this also. The different type of soil will play an important role, and different type of moisture availability, environmental factor will definitely play a role. So therefore, we are little shifting, and moreover, what about to talk about defender? Defender is also very important. So that's right. I'm pursuing in, like say, I hope in 1 or 2 years it will be finalized, because we need to have a data to tell. They say okay, it is not correct. Then I tell you, rice exporter president IPDA will told okay, this is not correct. And so what will be said to the farmer? So then I have no answer, because I have to work on it. Right. You are correct. Yes, I agree to you.
Thank you, sir. Thank you so much.
Sir, good evening, sir.
Yeah.
Sir, if I want to work on ETL and banana pest, banana pseudostomy wilt, to whom I can contact, sir?
For what?
Banana pseudostomy wilt, that is annual crop.
Yeah. Right, banana. You are working. You are speaking from NRC banana.
Yes, sir.
Okay. So DBBQ is one of the best way to talk about to have this one, right? And we can also put on to the question to Dr. A.K. Singh, DDG Horticulture sometime. That's right. That,
Actually, the banana sulfur will, I couldn't get any ETL.
banana for certain insect pest. I
Yes,
hope
sir.
Sundaraju start working. I hope I'm correct. Sundaraju is the director.
Yes, sir.
Yeah, he was start working. I remember, but he could not make much good stress or effect on this. But he tried. That's one he tried, and some of the pests he identified also, and the key pest. And then actually, first of all, you have to identify the key pest, and then I tell you how we are doing for the ETLs. They first of all you identify the key pest or the area, and then you have to go to hotspots. In that hotspots, now you have to work on. Let's say now what will be the pest given to you. That's very important. Now you cannot ETL, you cannot based on just like you do PhD thesis, right? You got my point. ETL is now you have to in one you cannot work on one place, and you had now what we have to work. Let's say banana grown in the. You have to take the example of Jhansi. We have to take the example of Bihar. We have to take many places,
Okay.
right? And then see that you have three pests. Let's say one pest is there. The three population, four population, five population. What will be the effect? And if the impact are coming almost same, then you can talk about there will be economic loss. You have to work economics also. For ETL, is economics is also.
So from basic, if you want to do means to whom I have to contact, sir? Which organization is best?
For banana, you have to develop yourself.
No, no, no. Only ETL concept actually.
Huh?
For ETL concept, how we can proceed
Right,
for
right. NIHPM, we can do. NIHPM is the one of the best. I hope you have seen. There is some packages there for banana also, na? ISA based
banana is
model,
there, yes, sir, yes, sir.
right?
But ETL, I want
But
to.
what I'll do, I'll to be very frank to you, that I will ask. There is one earlier PPA we have with us, Dr. S.N. Shushil, right? Who
Okay.
is very much concerned about ISA based and that model. I will ask him definitely for banana because banana I have no much of experience to work it out. So I will definitely ask for packages they are developed for banana also.
Actually, I am new to banana also, sir. I was working in lab research.
Yeah,
Just
definitely.
now I have joined.
Because you are ICAR Institute and that I definitely have a good contact with you. So I will ask Dr. Sushil and then we can inquire.
Okay, sir. Thank you.
So okay, friends, if there is no other question, if you have a question, please write in chat box on my mail. We're definitely going to answer. Over to Dr. Rekha now, please. Dr. Rekha.
Thank you, sir, for your elaborate lecture on rice. Dear participants, from tomorrow we will, from 10 o'clock, we will have another lecture on IPM and sugarcane and other sessions. And tomorrow morning, I'll share the feedback link as well as the post-course evaluation link. Kindly complete it before the valedictory session.
जरूर को बुलाओ
That's it for tomorrow.
मैं बुलाओ पीले चाय पीते
I think that's all for today.
Thank you, Dr. Rekha.
If there any other question or query related to the feedback or anything, then they can ask.
Yeah, definitely. What you do in feedback and post course evaluation, please
Tomorrow
post
morning, I'll share
in the
everything.
morning,
Yes, sir.
and then tell them to once all the lecture are over, they
Hmm.
keep track with them as
Yes,
soon as
sir.
like so they will submit by 1:30 or 2 because
Yes,
to chief guest and to manage what we need to tell them that
yes,
will
sir.
right.
Yes,
Okay.
sir. Yes, sir.
Okay. Thank you, Dr. Rekha. Over.
Thank you. We'll close for today, sir.
Yeah, yeah, sure.
Yes.